All right, good morning. Can everyone hear me? Awesome. So my name is Kaeli Swift, and I'm going to be talking about the breeding biology of the Tinian monarch, which I am presenting on behalf of my two co-authors, Fletcher Moore, who's here in the audience, and our PI, who's not here, Dr. Beth Gardner. So before I get started, I'm just curious, how many people here have seen a Tinian monarch? Okay, so if how many of you know that it's a bird and not a butterfly? All right, well, you're doing better than most of my family, which is still deeply confused about the sudden career change. But I'm not surprised that relatively few of you have actually seen these birds. They are what we would describe as biologically rare. They are endemic to the island of Tinian, though there are some records that they may have occurred on some of the other islands. But even on Tinian, they are limited to our native limestone and other mixed forest sands, which means that if you come to Tinian just to go to Pika Fest, you're probably not going to see a monarch. Like other monarch flycatchers, they are primarily insectivores, though we have actually seen them eat lizards twice. So like all of the Mariana's wildlife, monarchs have been tremendously impacted by centuries of colonialism and landscape use changes that have significantly impacted their habitat. So as of today, that's culminated in the reduction of Tinian's primary forest to only about 5 to 7%. And so as you can imagine, this hasn't always been great news for the monarch. And shortly after World War II, a publication came out suggesting that their numbers had maybe dwindled into the double digits. And so by 1970, they were listed as endangered, though no formal assessments of their populations had actually been made at that time. So over the course of the next couple of decades, those surveys were conducted, and actually their populations were a lot more robust than we were expecting. And by 2004, they were formally delisted. As of today, estimates for their population on Tinian is somewhere around 95,000 individuals. So that may seem like a fairly big number, but because these birds are still inherently rare, given that they're an island endemic, there's still interest and necessity in obtaining a deeper understanding of their biology, particularly as it relates to their reproductive ecology. And so that's where we come in. For the past 2 years, we've undertaken the most comprehensive study of their reproductive biology to date. And in particular, we were interested in looking at things like mate and site fidelity, characterizing nest site selection, assessing reproductive success across Tinian's various habitat types, identifying causes of nest failure, and then estimating juvenile survival. So our first step in undertaking this work was, of course, to establish a marked population of birds, and we did that across Tinian's three main habitat types, which are native limestone forests, secondary forests, and then Lucena-dominated plots. And we conducted all of this work in the northern 2/3 of Tinian, which is the military leased land in these plots that you can see in yellow. So once we had our banded birds established, we would go out and we would look for indications of nesting, either through behavior or just direct observations of nests through cold searching. And once we found nests in our marked territories or their neighbors, we would deploy a Reconyx hydrofire camera to help us track sources of nest failure. And then we also conducted regular nest checks every 1 to 3 days. Once a nest terminated, for whatever reason, we conducted our vegetation surveys, and these were in service to better understanding monarch nest site selection. And in particular, we measured 15 characteristics that are commonly associated with nest success. 12 of these were microsite characteristics, so things that are specific to actually where the monarchs are putting their nests and how they're building them. So things like, how tall is it? What's its concealment score? How many support branches does it have? How many access points within a 0.5 meter sphere around the nest are there? And then the other 3 were kind of broader characteristics that related to the 10-meter radius area around the nest. So for that, we were looking at things like canopy cover, describing the understory cover, and then quantifying and identifying all of the trees within those 10-meter plots. Now, in order to contextualize that and understand where monarchs are putting their nests, as opposed to where they're not putting their nests, we did a paired design where once we did our vegetation survey, we selected a random point 25 meters away from the nest and took all of these same measurements in a random 10-meter radius plot elsewhere, theoretically within the territory. So to date, we have banded 304 birds. That's included 151 nestlings, 15 juveniles, 138 adults. We conducted the first lab-confirmed sex analysis for 203 of these individuals. That showed us that our male-female sex ratio among hatchlings is 52% male, 48% female, which is pretty typical of songbirds. And it also validated the very large morphometric overlap between males and females. So when it comes to partner and site fidelity, we found that among our 64 banded pairs, we confirmed divorce 3 times, meaning that both individuals were still alive, but had repaired. In general, what we found was that the males stayed on the territory and it was the females that dispersed. Generally not too far away, on average about 86 meters, which is basically the next territory or maybe the one after that over. So that's just a fairly high level of partner fidelity. Likewise, we saw a pretty high level of site fidelity. So our average distance between our first 2 nests was typically 13.5 meters. But one thing about this that has been a little tricky is, as you can see from this graph, monarchs actually reproduce all year long. There's a dip in the dry season, but typically when we ask questions about site and partner fidelity, we're asking that between breeding seasons. And since monarchs don't have distinct breeding seasons, this has been a little tricky. So if anyone has ideas for how to handle this better, come find me after the talk. And then one thing that we weren't looking at necessarily specifically, but is kind of interesting to report, is the number of times that these birds will re-nest in a given year. And one thing that really took us by surprise is monarchs will re-nest even when their previous nesting attempt is successful, and even when their previous semi-dependent juvenile is still on the natal territory, which is pretty unusual for monarch flycatchers, including their closest relatives. So in total, we conducted 149 vegetation surveys. We found that nest height is typically 185 centimeters tall. In other words, they make it pretty easy for us to find because they're kind of right at eye level, and we are grateful to them for that. They typically build on smaller saplings or on auxiliary branches of larger trees, using 2 to 7 small branches. They're always built under canopy cover, typically about 4 meters high, and very often they would be built near the tallest tree in the plot, and often that was the tallest tree in the area, about 12 meters tall. One of the most important things we found is that monarchs are very clearly selecting native trees as their nest plants. So even though native trees only accounted for 41% of the trees that we surveyed across all of our nest sites, they accounted for 75% of our actual nest plants. Their 3 favorite nest plants are the hoda, the mupunyao, and then one introduced plant, which is the behonia, that's the orchid tree. One interesting thing we found was that even in our Tongan dominant plots, where Tongan accounts for at least 60% of the trees in the plot, they only use it as a nest plant 12% of the time, instead preferring alum, which is another native tree. When it comes to comparing our nest plots versus our paired plots, we found that nest plots typically had taller trees and a higher density of both saplings and trees, which isn't necessarily surprising. We knew monarchs weren't using open fields or agricultural areas, but it just sort of confirms this idea that these birds need forests. All right, nest success. So in total, between 2021 and 2022, we monitored 150 nests reflecting 65 different territories. Of those, 49% fledged, 44% failed, 3% were abandoned, and 2% could not be determined. We calculated daily survival rate using Mayfield and Johnson methods, and collectively we found it was pretty high, 0.97, which translates to a probability of success around 49%. Interestingly, across our different habitat types, we found that there was a slightly higher daily survival probability in our Lucena plots. All right, so while Tinian is fortunate in that we do not have brown tree snakes, we do have rats, a lot of rats. And so unsurprisingly, we found that predation was by far the greatest source of nest failure, accounting for 75% of failures. And of those, of our documented predation events, rats accounted for 62% of them. Look how happy that rat is getting there. Monarch eggs. Likewise, another invasive predator, cats, were the second biggest predator. And unfortunately, cats are kind of the double whammy because they're not only going after nest contents, but we would often see them actually actively attacking the brooding adults. In slightly less depressing news, we also documented 2 native predators, which were previously suspected to depredate monarch nests, but we haven't confirmed yet. And that included Såli and the kingfisher. Now, the fact that they're getting eaten so much is a little less surprising when we looked at our concealment scores. So monarch nests are really visible. They are typically visible in all cardinal directions. They have 85% visibility, like this nest, which these birds nested 3 times and fed the rats each time. They're also pretty easily accessible with typically 7 access points. And finally, when it comes to juvenile survival, we monitored 60 juveniles for 8 weeks, doing weekly survival checks. We found that 35% reached independence, which we based on 56 days, based on a previous Fish and Wildlife report. 42% died, and 23% had unknown outcomes. One interesting thing is we found that their time on the natal territory was a little longer than we were expecting, usually about 65 days. Could be as many as 92, and we actually documented adults feeding these kids as late as 80 days. A small handful of birds have been seen since dispersal, and in general, we find that they are not very far away, only about 55 meters from the natal territory. So just to wrap things up, we found that nest survival for these birds is higher than previous Fish and Wildlife estimates and pretty similar to what we see on forest birds on Saipan, but lower than similar species of monarchs. We've identified that rats are a primary threat to nest success, and that monarchs prefer nesting in native trees and these high density plots, which suggests that rat and habitat restoration may be important management tools moving forward. And finally, independence for juveniles is difficult to establish because there appears to be a fairly large range that they can stay on the natal territory, which suggests both a large level of parental investment and that our estimates of juvenile survival may be a little bit inflated if in fact we need to shift when they reach independence. So we may need to do a follow-up study. I want to just thank everyone who made this work possible, in particular Gilda C. Clark, for really recognizing the value of this work and throwing her weight around making sure it has continued for all these years. And with that, I'll take any questions if I have time. Thank you.
Thank you, Kaeli.
I'm just curious, when they renest, are they abandoning their other nests, and what happens to those babies?
No, so they are not abandoning their nest. They'll just reuse nests regardless of if they succeeded or failed, but we never saw, I don't think we ever documented them reusing an abandoned nest. Yeah, so these are nests that have terminated.
If they have that full nest and they renest, are those babies abandoned?
I see. So the question was, if they renest, why they still have fledglings on the territory? No, it actually looks like we have the parents kind of divvy up responsibility, where one sex will take the older kid and the other sex takes the younger kid. And we couldn't, do that in a real specific way, but that was kind of just based on our observations.
Yeah. There's a Zoom. Hold on. There's a question on Zoom from Sarah Fager. It says, what do you make of the higher nest success rate in Tinian? Was the result statistically significant?
Oh boy, I have some ideas. So we are still working on modeling how to best attribute our different microsite characteristics to nest success. So I don't want to say that it's significant yet, because I want to make sure I get these models right. But as for my hypothesis, my suspicion is that because Lucena-dominated flats have a lot fewer fruit trees, I think there's just a lower density of rats in these flats. They're still there, and in fact, rats were more likely to depredate nests in Lucena flats than they were relative to the other ones. But I think looking at how rat density on Tinian varies by habitat type may be a really important component to answering that question.
You can take one more.
We have another question.
Let me switch it around here, if you mind. Something about.
If not, I might ask a very silly question. Were any of the cats observed pushing nests off, as cats are prone to do with other things that are on high ledges?
No, I yeah. That may have taken seeing something like that may have taken the hurt off a little bit, but no, it was always these dramatic, like LeBron James, boom, just kind of taking the whole nest down.
Well, thank you, Kaeli, for your talk. Let's get Kaeli on that.
All right, so my name is Fletcher Moore, and I work with Dr. Swift on the Tinian Monarch project. But today, I'm here to talk about a different bird on Tinian. So as part of our research, we've been able to learn a lot about the additional ecology of the island. And I'm going to talk about a previously undocumented behavior in one of our other Mariana's endemic forest birds. So for almost all birds, nest building is prerequisite for breeding. It can be a costly and dangerous process. Nest material may be found on the ground, which is could be closer to predators such as cats and monitor lizards. And as a result, some species use alternative methods of nest material collection to decrease costs and risk. Nest material kleptoparasitism is the theft of nest material from another bird's nest. When an animal takes resources collected by another animal, this is known as kleptoparasitism. It's most commonly associated with food theft. So this is a frigate bird, which is a notorious kleptoparasite. They will stay near seabird colonies and steal food from returning seabirds, which saves them the hassle of having to fish for themselves. Nest material kleptoparasitism is considered parasitism due to the energetic cost of collecting more material or repairing damaged nests, cause the destruction of nests or discourage nest building entirely before a nest becomes active. It's uncommon in solitary nesting birds, but is quite common in colony nesting birds like seabirds, where nests can be close together or nesting materials can be a lot harder to come by. This behavior is previously undocumented in any Mariana's forest birds. And from here on out, to be clear, if I reference kleptoparasitism, I'll be only talking about nest material kleptoparasitism, not theft of food or anything else. So white-eyes are a small paleotropical group of passerines. They're considered the most successful oceanic colonizer of any songbird. And there are over 120 species worldwide from the Pacific Islands to Western Africa. There are 3 species of white-eye currently found in the Mariana Islands: the bridled white-eye, which is on Tinian, Saipan, and Guam; the Rota white-eye, which is endemic to Rota; and the golden white-eye found on Saipan, Guam, and the Northern Islands. Some of these species have since been translocated to some of the other Northern Islands, which we will hear about later, but these in general are their main habitat islands. Kleptoparasitism has been observed in 2 other species of white-eye, the Japanese white-eye and the Indian white-eye. So the Nosa, or the bridled white-eye, is a small green songbird found currently on Tinian, Saipan, and Guam. There was a subspecies found on Guam, but it has not been seen in the wild since 1983. It's primarily found in forest stands from native limestone to Tongan forests. They live in small social flocks, but form pairs and establish territories for nesting. They can breed year-round with multiple breeding attempts per year. and both males and females collect nesting material, but males may also use nesting material in courtship displays. So Tinian has a diverse population of native birds, including 4 small native songbirds: the Chicharican tinian or Tinian monarch, which we just heard about, the Naabak, the rufous fantail, the Igigi, the Micronesian myzomela, and the Nosa, the bridled white-eye. So these 4 species build woven nest cups in the undercanopy of forest stands. So they use similar habitats for their reproduction. Each species is territorial, but appears to be more aggressive towards members of their own species than other species. And we have documented different species nesting close together, sometimes within arm's reach of each other, which is not something you would expect from members of the same species. Spinning wheel of death. Oh, there it is. So these are our 4 songbirds that I referred to. So as you can see, they all use woven nest cups. The exact material composition of these nests is not known, but they appear to use a similar combination of leaves, grass, and small sticks woven together with spider webs. The white-eye and the myzomela on the left have the most similar nests in appearance and composition. Their nests are hanging baskets, while the other 2 rely on rigid supports to build their nests, and the nests are generally larger. So you just heard all about our Tinian monarch project, but as a reminder, we use Reconyx hyperfire cameras to document nest fate of active monarch nests. We reviewed the footage after the conclusion of the nest to determine cause of failure or proof of fledgling. And observations of kleptoparasitism were made during the review of this footage, but were incidental to the original monarch project. So to date, we have documented 7 observations of this behavior of nest material theft by the bridled white-eye. The first observation was made in November 2021, and it was a real-time observation in the field of theft from a rufous fantail nest. Soon after that, in December, we collected our first video by a camera trap. Once we observed that, we began carefully checking for this behavior and collected additional observations. Thieves would often make visits to the same nests, and also were observed stealing material during nest building, incubation, post-fledge, and post-failure. And following is the first recorded video of this behavior. So this footage was captured by a handheld DSLR camera. The nest is of a rufous fantail, which was actively being built. After this observation, the fantails completed their nest 4 days later and begun laying. All right. All right, well, that was a little choppier than it was supposed to be, but I think everyone got the idea. So as you can see, 2 white eyes visiting that nest, taking material, likely nesting male and female. So this video was captured by a camera trap. Shows a white eye stealing material from a Tinian monarch nest, which has recently been deprecated by rats. And as you may be able to tell, especially here, the bird can actually cause quite a bit of damage pulling that material away, and seems to pull off more material than they actually are able to fly away with, which suggests kind of an outsized amount of damage for their use of the material. So what are the implications for host nests? In other songbirds, this behavior can cause nests to be destroyed or cause hosts to abandon nesting attempts entirely. It can add energetic cost and potential risk associated with collecting more materials. And monarchs often reuse or rebuild nests after a failed or successful nest, as Dr. Swift said. So even theft of material from a no longer active nest, like in the last video, could increase costs for future nesting attempts. And hosts may spend valuable energy chasing off thieves, as is shown in this next video. So this video is of the same Tinian monarch nest. This was just a little bit later on from the first one. And if you pay close attention, you might see an orange laser come through. And there is the rightful owner of that nest surveying the damage. So this pair of monarchs actually decided to not reuse this nest, and they nested somewhere else with their next nesting attempt. So what are the implications for the wider forest ecology of Tinian? Here are some of the most recent population estimates for the 4 Tinian songbirds we're talking about here. And as you can see, the bright-old white-eye has an estimated population greater than the other 3 songbirds combined. You'll notice that the Myzomela at the bottom, it has by far the least abundant of these species on Tinian. Given what we know, that the white-eye is the most abundant forest bird on Tinian, and that this behavior may have an impact on the reproductive success of several threatened bird species across this range, including the Tinian monarch, and that other songbird kleptoparasites are known to target with birds with the most similar nests in material and canopy placement. We did not document this behavior to the Myzomela, but it seems likely due to the similarities in nests that they would also be victims to the white-eye. The Myzomela has a particularly light, fragile nest, and therefore may be more vulnerable to failure from this nest disturbance. And they're an under-researched species in the Mariana Islands. Their numbers are already low on Tinian, as we saw. So based on our literature review, I believe that we are the first to report on this behavior. However, due to the number of observations we made and the apparent abundance of this behavior, we are certainly not the first to see this. I wouldn't be surprised if even someone in this room had seen this behavior in Mariana songbird. But if those observations are not recorded, documented, and shared, then they cannot be added to our communal understanding of forest ecology. So I'd like to take this opportunity to remind everyone that whoever you are, what degrees you have, or how expensive your binoculars are, that you could make an observation that could be previously unreported and could be significant to our greater understanding of ecology in this area. So if you are interested and you're spending time outside in the natural world and you see something that surprises you or you don't understand, reach out, find someone to ask about what you've seen. It could be really important. And there's obviously much more research to be done to uncover the extent of this behavior and the other many unknown behaviors of Mariana's forest birds. And these interactions are between these species are the backbone of our ecological understanding. So the more we understand these relationships, the better we can practice conservation that supports the entire natural ecosystem. And I hope that future research will shed greater light on these birds and the complex lives that they lead. And I would like to thank the people of Tinian who welcomed us in and have supported us so much with their hospitality and their traditional knowledge of the birds in the forest. And Kaeli Swift, who I would like to recognize as the first person to actually record this behavior, and everyone else who makes this research possible. Thank you.
Thanks, Fletcher. Some questions with that.
That was super interesting. Thank you for that. It seemed like a really dumb, obvious question.
Good morning, everyone. We're from the CNMI Division of Fish and Wildlife, and we're here to talk about habitat use area and selection for the Galga Karisu Nightingale Reed-warbler on Saipan. That is the male colt. No one's ever heard that before. So Nightingale Reed-warbler, it is a federally endangered songbird of the Northern Mariana Islands that currently exists only on the islands of Saipan and Alamagan. Subspecies of it used to exist in Pagan, Goat Island, and Guam. The scientific name is Acrocephalus hiwae. Its Chamorro name is Karisu, which means dwellers of the reeds, and it's found in Tongan forest, wetlands, bamboo, mixed introduced forest, and grass. And these are just characteristics of how the bird looks like. So evidence suggests that there is a population decline for the reed warblers. And what could lead to that is probably habitat loss. So because of that, the Saipan Upland Mitigation Bank was created. The Saipan Upland Mitigation Bank allows landowners to mitigate endangered species habitat that are affected by the developments in other forested parts of the island. In spring and winter months of 1997 to 1998, Steve Mosier conducted a Nightingale Reed-warbler habitat range study. In his study, he found that the mean home range for reed warbler was 4.35 hectares plus or minus 1.10 hectare, and the mean core area was 0.85 plus or minus 0.20 hectare. He had a sample size of 26 and located each bird at 10 to 20 minute intervals during a 1 to 2 hour tracking period. The tracking period recorded 2.23 times per week, so an average of 70 points per bird. Current regulation uses Steve Mosier's home range size as a baseline for the dollar amount to mitigate for silver credits. And then now I'm gonna pass. Oh, sorry. So DFW wanted to update the Nightingale Reed home range size and better understand habitat use and selection with interdaily movement through a longer sampling period. So we captured 30 Nightingale Reed warblers, 4 were recaptured, and each bird had attached a USGS aluminum band and 3 color combo bands, as well as a radio transmitter. So the radio marked birds were tracked using telemetry for an average of 43.3 days at 1 location per day. And then the telemetry was continuous over 2 years through multiple seasons. And now I'm going to pass it on to Brad for the data analysis.
Good morning, everyone. We look here, we have 2 birds. We have bird A and bird B. You can see the reed warblers moving across the territory. Amanda's out there and she's tracking it with the telemetry system. And we can basically triangulate the locations using the observation point where Amanda's standing, and also use bearings. We can take this information and answer our first question, which is what is the territory or the home range or the use areas for each of these individuals. And we originally looked at 5 different estimators. The first 3 are all using kernel density estimators with various smoothing parameters. So what this does is it tries to fit a distribution, an unknown distribution across the point data. The denser the points are, we know that that distribution is more centered around there, and it kind of flows out using these different smoothing parameters. We also looked at some 2nd and 3rd generation home range estimators, such as local convex hull and Brownian bridge movement models. And in the end, after reviewing a bunch of different runs across all the different birds, we decided on 2. So we're going to focus now on looking at kernel density estimators using least squares cross validation, which you can see here in purple, and then using an H plugin smoothing parameter, which you can see in the hatched yellow. The idea here is to use 95% of the actual points to kind of draw the boundary of the home range. And this is what this is like basically what we see. One thing to kind of note is you can really see how the smoothing parameter changes the behavior of how the home range is drawn across the actual detection points. And if we take the mean home ranges for the least squares cross validation smoothing parameter, we get 3.36 hectares with 95% confidence interval of 1.04 or 1.08 hectares. And for H plugin, we see something similar. We see 3.49 hectares plus or minus 1.1. We did remove 2 of the birds from this home range analysis, particularly because they moved quite far distances and they had home ranges over 15 hectares. And what we think happened is either they were searching for new territories, so they We can see them literally move over to another territory for a while, stay for a few days, and then fly back. One was after U2, so we think maybe the resources could have been limited at that time, and it was seeking out other resources and came back. But because they were such extreme outliers, they were not used in this analysis. And also, neither were paired with females at the time. The other question that we want to look at is, what are the core use areas? And these are areas with the highest density of locations. And the assumption there is this is where the reed warblers are hanging out. There's going to be more resources. These are high resource areas, essentially. So we used least squares cross validation again, kernel density estimator. You can see these blue polygons, but we constrained the number of points to either 50 or 70%. 50% was the standard, and if the polygons are still pretty small, we expanded out to 70%. And the idea here is we take these blue polygons and then we randomly place 10 vegetation plots within these core areas, and we use all 30 birds. So we get a total of 300 vegetation plots that we can use to kind of understand what's really happening in these core areas. So we use 10 meter by 10 meter vegetation plots, and these are distributed across forest and grassland community types. Here's an example of the points. So within these 10 meter plots, we're actually measuring species composition and structure for the trees. So we look at strata in terms of shrub layers as well as canopy layers. Amanda's in the upper left-hand corner, measuring DBHs of all the trees. Leroy's to the right of her, taking measurements on substrate cover and things along those lines. And if we generally just look at the distribution of the mean number of trees within each DBH class between the plots, we can really see that overall the plots are dominated by thinner, smaller DBH classes for tongan, bamboo, snags, and hibiscus or pogum. This is a little misleading because we know it's across various different community types. So what we can also do is take all of our plots, calculate what we call importance values, which take into consideration relative basal area and relative density for each species within each plot. It produces an index of dominance of each species within each plot. We use what's called a hierarchical cluster analysis, which groups the plots based on similar importance values within each plot. And we get something that looks like this, which is a little bit meaningless. It just produces 5 clusters, and this just means anything within each one of these clusters is really closely related in terms of species dominance. But what we can do is go back and look at the mean importance values for cluster to get a better understanding of what the plant communities are represented by each one of these. And if we look here in red, we can see red represents cluster 1. We see a high dominance pago or pago or hibiscus. And then you can see tongan is highly dominant in a couple of the other classes or clusters, including cluster 2, 3, and 4. And then we see bamboo was the dominant species for the 5th cluster. So using this information, we can go back and reassign vegetation communities to the actual plots. And we get something like this. So we have 5 clusters that are actually 4 community types, tongan, hibiscus thickets, bamboo thickets, and mixed introduced forests. This is all important because now we can actually classify the vegetation plots. We can look at the distribution of the plant communities within the core habitat for each one of these individual birds. So on the y-axis, we have the number of vegetation plots, which is 10 per bird. The x-axis, we have the actual individual birds. We bring back in the grassland plots, which were excluded from the hierarchical cluster analysis because they don't have DBH classes associated with them. Therefore, they can't be dominated by living species. And we start to see a different story. We see most of these birds are highly utilizing tongan or hibiscus habitats, but they're also using a variety of other plant communities, such as bamboo and more open areas like grassland. We can do something similar if we bring back the home ranges, and we overlay these polygons with the land use land cover classifications, such as this one, which is from Fred Amadon from 2016. And we see a similar story where we can see the mean area for each one of these plant community types from the land use land cover map includes grassland, pine and thickets, and mixed introduced forests. So what's this all mean? So Mosier had looked at home range areas, as Amanda had mentioned, and in reality, he broke it across 3 different plant community types. And we only mitigate for upland habitat. So if we break that out of Mosier's data, we can see it's 3.73 hectares. Our data is pretty consistent with what Mosier had measured as well, observed as well. And I think this is pretty interesting because we should probably reconsider the amount of area that we consider for credit and bring it back down to this upland territory size. So decrease it from 4.19 where it currently stands to something in the 3 range. I think we also show that there's evidence of multiple plant communities that are being utilized within this upland habitat area in terms of both the core areas as well as the larger territories. And what's kind of interesting is it tends to be early successional habitat. If you think back at the distribution of the DBH classes, they're all on the smaller end. So this makes us think that we should probably be considering habitat mosaics when we start looking at the species in terms of a larger habitat conservation management plan, or even in the conservation blueprint. So we need to kind of manage a habitat heterogeneity for the species, which is also likely going to be early successional. And then one more thing I think is important to note is we should probably update the recovery plan. So a lot of the recovery plan for the species is based on previous population estimates, such as 4,225 in 1986 and 2,742 in 2007, based off of distance sampling and point counts. But if you consider Saipan in its entirety is 11,538 hectares, assuming that all of Saipan is habitat, which it isn't, and we use the more conservative home range estimate that we produced, that's 3,434 Reed-warblers at complete saturation across the island. So I'm not saying the previous estimates are wrong, but they just may not be correct. And I think it's worth reevaluating those and seeing if we can validate those numbers using some of the information that we collected. And all this is important to reassess the delisting criteria, which revolve around the stable populations and trend sizes.
So there's still a lot of information that we need to do for the reed warbler. So currently I'm working on another study about reed warbler survival and site fidelity. And next year I'm probably going to begin a diet study to see the food resource available for the reed warblers. Thank you, everyone.
Yes, I have a question. Back in the 1970s, when there were still reed warblers on Guam, they were listed as endangered before the other bird species that we lost. And in those days, the local knowledge indicated that they were always associated with the Karisu, with the wetland reeds. And I'm just wondering, a lot of the protection for them being listed involved protecting those wetlands with the Karisu. How important really is that? Was that the wrong direction for, say, endangered species, or is it really critical that they need the wetland Karisu area?
Yeah. We do see the Karisu in other habitat types, but like we do for us, we don't have people develop on any wetlands. We're supposed to protect all wetlands. So it's important. It's important habitat ecosystem for other species besides the Karisu.
And just to follow up on that, I think it is interesting because the name Gaga Karisu, meaning well, the reeds. with the substantial clearing that happened pre and post World War II on Saipan, it's interesting to think that the species might be queuing in on these thinner Tinian stands, which may be some sort of visual cue that's similar to maybe what they see in the wetlands. And I think, you know, potentially that's something that could be looked at further.
Hi, so I'm from Northern Marianas College. And so what you see up here on the screen, this is a spectrogram. So it's a picture of sound. And this is a picture of a sound of a Mariana Fruit Dove song, a Totut. And so the song, if you don't know it, it's kind of a whoo, 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 whoo. So I want to talk to you about two things today. I want to give you a brief update about some acoustic characteristics of Mariana Fruit Dove songs. And then secondly, I want to talk about some automated identification of Mariana Fruit Dove songs that we've been working on. This is work that I've been doing with the help of some student workers at Northern Marianas College. So Jaden Villacruzes and Alex Tudela are here. Could you just wave your hands so people know you? So they both know a lot about this work. So I encourage you to ask them about this work also. And they've also got some posters about this and some other bird stuff. So that's in the back corner. So please talk to Jaden and Alex about the work that they've been doing on these acoustic things. So we measured a bunch of things in the Mariana Fruit Dove songs. We measured song duration, the frequency, the length of individual syllables, the time between syllables, and the number of syllables in songs. We measured over 800 Mariana Fruit Dove songs. These songs came from over 60 different locations and 4 islands. So yes, we even had a single recording that we measured from Guam. It was recorded in 1978, and that recording is in the Cornell Macaulay Library of Natural Sounds. So we measured that. One thing that we can do with all of this data is we can look for differences in Mariana Fruit Dove songs between the islands. So with birds, we normally assume that song has some function in breeding. So if there were differences in the acoustic parameters of the songs between islands, that could indicate a reproductive barrier or reproductive isolation. So my student, Jaden Villacruzes, had a poster investigating that for the islands of Saipan, Guam, and Rhoda. So I'm not going to tell you right now what she found, but please go look at Jaden's poster and talk to her about those island differences. Right now, I want to show you some preliminary results from our study of the number of syllables in songs. We classified syllables. We labeled the types of syllables by letter. So we called those long, slow syllables at the start of a song B syllables. And we called all the syllables in the second part of the song, which are generally shorter, faster, and decelerating. We called those C syllables. We counted the number of each type of syllables in songs. We found a few things. So first of all, look at the top right. Generally, as the number of B syllables in the first part of a song goes up, the number of C syllables in the second part of the song goes down. So Totut seemed to be trading off B and C syllables to some extent. Despite that, there's still a positive relationship between the number of C syllables in the later part of the song and total song duration. So if you look at the graph on the lower right, the x-axis is the number of C syllables, the y-axis is the song duration in seconds, and the color of the dots shows the number of B syllables. So you can see that songs with more C syllables are longer, even though those songs tend to have the fewest B syllables. There's also something funny going on with the B syllables, though. So we found a lot of songs that had 2 B syllables at the start of the song, like the example shown in the top spectrogram. And we found a lot of songs that had 4 or more B syllables, like the bottom spectrogram example. We found almost no songs with 3 B syllables at the start of the song. So if you look at the histogram, the graph there, in all of the songs that we measured, I think we only found 2 or 3 cases where birds sang only 3 B syllables. This dramatic lack of 3 B syllables in the hundreds of songs that we've measured suggests to me that the B syllables at the start of the song serve some important function. I don't know what that is. Are there different messages conveyed by 2 B syllable songs versus 4 or more B syllable songs? Is there some sort of selection pressure that prevents 3 syllable songs because it's trying to avoid confusion in that message? Or is this related to sex? So as far as I know, I don't know whether the songs are sung by males, females, or both in Mariana Fruit Doves. So if anyone here has insight about that, I would very much like to hear about that after the talk. So maybe what we're seeing here is a signal about the sex of the bird. I want to follow up on this issue, and I would like input from all of you as I get ready to do that. So we're about to start a playback study in which we will play Mariana Fruit Dove songs through a speaker in the field, and we will be trying to assess how birds respond to those songs. My main goal for the playback study is to test for differences in responses between 2 B-syllable songs and 4 or more B-syllable songs. I've never done a playback study before, and so I would very much like to hear any ideas or suggestions that you have. I'm also curious about whether you've noticed any differences in how the Totut use the 2-syllable or the 4-syllable songs. So, and even more generally, have you ever observed any responses of birds to song? Have you ever seen singing lead to a fight or to copulation or anything like that? If you have those kinds of observations, please come talk to me. I'm very interested. Okay, so now I want to talk to you about a slightly different topic, an automated identification of Mariana Fruit Dove songs in sound recordings. So the goal here is to train a computer to automatically identify Mariana Fruit Dove songs in sound recordings. This would let us analyze thousands of hours of sound recordings, for example, from automated recording units placed in the northern islands. We would be able to monitor Mariana Fruit Dove presence and abundance. So I used the analysis program software developed by Michael Towsey and others at the Queensland University of Technology in Australia. The analysis program software has some general machine learning methods that users can fine-tune to search for specific types of sounds. So along with another student of mine who's not here, E.J. Guire, we've been tuning a classifier to search for Mariana Fruit Dove songs. We're using a pre-built whistle detector that's in that analysis program software. And we can tune various parameters to search specifically for Totut songs. So for example, we can tell the algorithm what frequency range to search in and how long we expect the syllables to be. We now have a functioning classifier. It does a decent job of finding Mariana Fruit Dove songs and recordings. And I'd like to show you some measures of how that classifier performs. Our training data contains 480 one-minute segments of sound recordings in which a human has checked to see whether there's a Mariana Fruit Dove song. These recordings came from 3 different locations and span the time from 6 a.m. to 6 p.m. Overall, there are Totut songs in about 37% of these 1-minute segments. For each 1-minute segment, I want the computer to answer a yes or no question. Is there a Mariana Fruit Dove song in that segment of recording? And then we compare the computer's answers to our human answers. I trust the human data much more, and so this lets us evaluate how well the computer is performing. So here I'm showing 2 summary statistics that show different aspects of the computer's performance. True positive rate, which is the purple line, tells us what proportion of the minutes with a Mariana Fruit Dove song were found by the computer. False positive rate, the green line, tells us what proportion of the minutes without songs were incorrectly labeled as containing a song by the computer. So true positives are good, we want a high true positive rate, and false positives are bad, so we want a low false positive rate. So let me explain the graph. I can adjust the sensitivity of the algorithm by adjusting a volume threshold that the algorithm uses when it's searching for songs in a recording. The decibel threshold is shown on the x-axis here. When that threshold is low, the algorithm has high sensitivity, so it has a high true positive rate, which means it finds most of the songs, even the really soft ones. When the decibel threshold is high, the algorithm's true positive rate is low, which means it only finds the loudest, most obvious songs. As you can see, there's a trade-off between these 2 types of performance. To get a high true positive rate, we have to accept a higher false positive rate. And if we want a low false positive rate, we have to be willing to accept a lower true positive rate as a consequence. So depending on the research question, you can adjust that trade-off. For example, Kaeli Swift from the CNMI Department of Fish and Wildlife has hundreds of hours of sound recordings from the islands of Guam and Alamog, 150 miles north of Saipan. The Department of Fish and Wildlife translocated fruit doves to Guam a few years ago. They really want to know whether the doves are still there and how they're doing. To the best of our knowledge, there are no Mariana Fruit Doves on Alamagan, but survey effort is very limited there, so it's hard to be sure. So we use this automated detector to search for Mariana Fruit Dove songs in the recordings from Guam and Alamagan. In this case, we want a high true positive rate because if there's a Mariana Fruit Dove song in those recordings, we definitely want to find it. And we're willing to accept a higher false positive rate. So this means that my students and I have to look through a bunch of false positive segments in which the computer claims that there's a Mariana Fruit Dove song, but in fact there's not. But it's worth doing that if it means that we find even a single song from Alamagan, for example, which would be a big risk. So far, we have not found any Mariana Fruit Dove songs from Alamagan, but Alex and Jaden and I are still searching through a bunch of segments flagged by the algorithm. So far, those are all false positives, but we will keep looking. The main point that I want to make about this is that we have a functioning classifier that automatically identifies Mariana Fruit Dove songs in sound recordings. This is ready to use for answering biological and ecological questions. And the classifier is adjustable so that you can prioritize a high true positive rate or a low false positive rate, depending on your needs. We are continuing to improve this classifier, and I'm certain that performance will get even better. The software is free to use. I'm happy to share the parameter settings, the configuration files that I'm using. So if you have a use for an automated Mariana Fruit Dove song identifier, please come talk to me. And I'll just note, I also see people here from other islands, including Palau. I think this classifier would work for the Fruit Dove on Palau. The song is very similar to the Mariana Fruit Dove song, so I think we could use it for other birds as well. I want to thank my students who've done a great deal of work on this project. I also want to thank Northern Marianas College and Mrs. Velma De Leon Guerrero, who's the head of our science department, who's been very supportive of this. My student research assistants have been funded in part through the Lewis Stokes Alliances for Minority Participation or the LSAM grant. And I'm grateful to my partner, Ellie Roark, and her colleagues at the Department of Fish and Wildlife for their help. So now you can ask me questions, or you can answer my questions. What's going on with those B syllables, and what can we do with a fruit dove classifier? Yeah.
Great talk. You have to forgive me. I don't know if I heard correct at the beginning of your talk, but I was wondering if you mentioned, and if you did not, could this be used to track these recordings to use as a proxy to track speciation between populations of the fruit dove?
Yes, I think they could. And so my student Jaden has a poster looking at this. We looked at exactly that. So for example, if the fruit doves on Rhoda sing a song at a higher frequency than the fruit doves on Saipan, that would sort of be an early warning clue to us, like, whoa, maybe these populations are not sharing genes and they might be kind of beginning to diverge. So that is, the data can definitely answer that question, and you should talk to Jaden. She can show you those results later. Thank you. Yeah.
Andersen, thank you for your talk. I just wanted to thank you for using your time and your platform for really promoting the work of your students, Jaden and Alex, and thank you all.
Hi, great talk. I just noticed that you did not sample for Tinian. I was wondering if there's a reason for that.
The main reason is that I just haven't gotten there, and Ellie hasn't gotten there, and Haldre hasn't gotten there. I think we had some recordings that Haldre let us use. But the other thing with Tinian is I've been there. There are Totut fruit doves there. They're less abundant in my experience than Saipan and Rhoda, and I'm less familiar with how to get to where they are. So we need to get recordings from Tinian eventually, but I think it'll be harder to get large sample sizes because it seems like there's fewer fruit doves there. Do it. Yeah, send it to me. Let's do it. Go back yet.
Sorry, yeah, great talk. And I just have just a quick question because I might have missed it. I step out for a second, but is there any evidence that there might be like competition for sound space or frequency or bandwidth between the different species out there?
Yes, that would be most prominent, I think, with the smaller kind of tweedy songbirds, you know, bridled white-eye, rufous fantail, all those things are about 7 kilohertz. The reason I started doing the classifier for the doves is that they're in a very different frequency band from most other birds. They're much lower pitch. The only other things in that frequency band are the island collar dove, the Philippine collar dove, the Streptopelia. And the white-throated ground dove is slightly lower frequency, but there's some overlap. And then the last thing that's in that frequency band that's actually giving me lots of false positive problems are chickens. So roosters are right about this kind of 400 hertz. So I think there's less competition in this frequency range than there is in the higher frequency ranges. In higher frequency ranges, for sure.
There's a question from Zoom that from Rick Camp that says, interesting presentation on Mariana Fruit Dove vocalizations. Do you also record the strength of the signal, which could be used to determine distance and compute abundances using Q-rate distance sampling methods?
That's a good question. I thought about that, and no, we don't. So I have talked to some people about how to estimate the distances, and that is more difficult than I can get into at this point. I've talked to people who place multiple microphones and try to triangulate. The other thing is that we're using multiple different types of hardware to collect these. So some of these recordings are from shotgun microphones. They're very high quality. Some of them are from automated recording units, audio moths or things like that. And so we don't know the distance. We weren't there in person, and the hardware varies. So I have not tried to measure the signal strength yet, but I'm interested in that if you know how.
Thank you, Victor, for the talk. Rick Camp.
Right. Thank you, Wilson. I appreciate it. And as Wilson was mentioning, there are the student posters, so take your time to go through there. One of my favorite findings that confirmed a thing that I had dreaded my whole life in growing up on Saipan is in data form, and it's so relieving. I'm not going to tell you what it is so that you have to go figure out what I'm talking about. But next we have Martin Kastner presenting. Martin.
Good day. Can everyone hear me? All right. Yeah, I'm happy to bring us a bird talk from Guam, but great talks everyone so far for the CNMI. So I'm presenting on behalf of my co-authors, Kayla and Haldre from Virginia Tech, Malia, who's in the room, Scott and Eben from USDS, and Shane from USDA, and talking about Såli fledgling survival in areas where we did brown treesnake control. So why study Såli? Well, first of all, because they're the best. But second of all, they're one of only 2 native forest birds remaining on Guam. And also they happen to nest and roost in areas where there are brown treesnakes present, which is something kind of different and interesting for native birds, and has a lot of potential for conservation. And why study fledgling survival? I mean, the fledgling stage is somewhat understudied in general in birds. And with Såli, we know fledgling survival is extremely low. We've had previous studies on that, somewhere between 5 to 25% survival in general. And most of the mortality is due to brown treesnake predation. And so because of that, we think that Såli fledgling survival could be a good sensitive indicator of the success of brown treesnake control. And some additional background, brown treesnakes in urbanized areas could present a very high biosecurity risk because the movement of cargo, of people, of vehicles, all that kind of thing is mostly from urban areas to other urban areas, including between islands. And then the lessons of, studying a bird in an area with brown treesnakes could be applicable to future work on reintroductions, for example, on Guam. And no one has ever actually used bait tubes specifically in urbanized areas before because it wasn't approved yet by federal agencies. And so this is the first study using bait tubes in especially a residential area specifically. Urbanized areas, I mean, if you define ports as urbanized, and maybe bait tubes have been used there, but they haven't been used in areas around homes, which is, has some really interesting potential for expanding avian conservation across the island of Guam. And yeah, this has been an interesting, multi-agency collaborative project, the kind of projects that people like to see. So 2 main research questions. First, does brown tree snake baiting produce a detectable increase in Såli fledgling survival? And will brown tree snake ban control in general lead to a reduction in brown tree snake caused Såli fledgling deaths? So you can see our study plots across residential areas of Andersen. And so it had a factorial design, so close to forest and away from forest, and then with presumed high brown tree snake density and presumed low brown tree snake density. So that gives us 4 main plots, and each had a treated area and an untreated area, so 8 total subplots. So if that's not enough to kind of wrap your head around, we also had 2 different brown tree snake control methods, but they were separated in time. So first we had baiting phase that was from October 2020 to July 2021. And then we had traps on the landscape from April 2022 to October 2022. So somewhat complicated experimental design and definitely a headache to analyze, but we're doing our best. And so just some pictures of bait tube on the top and a brown tree snake trap, kind of a newer design on the bottom. So yeah, shout out to the Såli crews. I mean, the Såli crew in the room, and then the crew that did most of the fledgling survival work. Zia, Kayla, Ray, Gary Dotamona, some of you in the room, and yeah, a very photogenic crew for sure. So it's at 62 transmitters in the pre-treatment phase, so before any brown tree snake control at all, and then 240 in the post-treatment phase. So I should underline that because that's like a massive amount of work and a whole lot of birds transmitted. And for every single bird, we track them daily for the first 15 days, and a little bit less frequently after that when we know that the survival tends to be a little bit higher. And so we track them when the birds were alive, and if we found the bird dead, then we would try to figure out what the cause of mortality was. Sometimes it's quite obvious when you find a transmitted bird inside a brown tree snake, and so there's some pictures of that, and that's always exciting. And then sometimes we always also find different. So I've presented before on sliming by brown tree snakes, which is when birds are killed by brown tree snakes, and presumably the snakes are too small to actually swallow the birds, so they just drop them semi-regurgitated. That's about half of the brown tree snake caused mortalities. And then we also, you'll see later, we have some cat caused mortalities. And so just there's a whole bunch of different results, but these are some results from USDA on the bait tubes. So take rates, you can see in the graph on the top, were highest in the higher density brown tree plots. And so somewhat logically, the take of mouse bait was higher than the take of chick bait. And Shane hypothesizes that this is because the birds tend to decay a lot faster than mice, and not necessarily a signal of preference of mice over birds. And there were about 300 bait takes overall, and with a known kind of correction factor for non-target take, meaning animals besides brown tree snake taking the bait, that's equivalent to about 270 brown tree snakes removed from the plots. The trapping phase, and I'll remind you that trapping happened after the bait tubes in the same plots, so the BTS levels could have already been suppressed. 81 snakes removed in that phase. Most of them, there was trapping ongoing in the treatment plots as well as the non-treatment plots. Most of the snakes trapped, 50 of them were in a non-treatment plot that was somewhat close to the forest, so it seemed to be kind of a major kind of sink of snakes into traps, but there were 23 snakes removed from the treatment plots. So Såli fledgling survival. So in the graph on the left, you can see that we had a slight increase in the proportion of fledglings survived, so the blue dots, from the pre-treatment phase to the post-treatment phase, and a slight kind of relative decrease in mortalities. Whereas in the untreated plots, we had a jump in mortalities, so the reddish dots, and a decrease in the proportion of survived fledglings. But proportion is a little tricky to analyze. There could be a lot of factors that kind of affect those proportions. But if we look at the survival probability of fledglings, there is no significant difference across phases or between treatments. Looking at cause-specific mortality, and I presented on this at the previous MTCC, and that was kind of more, it was during the baiting phase. And we had actually seen kind of like a relative decrease in brown treesnake cause mortality in the treated plots. After the whole 2 years of post-treatment in the snake control areas, so the third and fourth columns in the graph on the left, BTS cause mortality was about the same relatively, whereas there was an increase in BTS cause mortality, so the green section in the untreated areas. On the right, you can see in areas that didn't receive control, in 3 of the 4 plots, we had an increase in the proportion of fledglings that were predated by BTS. And in the snake control areas, 1 plot had an increase and then the others were stable or a decrease. But overall, the main kind of result is that we didn't see a significant difference in fledgling survival probability between phases of treatments, despite some kind of promising indications in terms of the proportion of birds surviving. And there were proportionally fewer brown treesnake caused deaths in treated areas compared to untreated areas. We definitely saw continued brown treesnake caused mortality, even in treated areas, especially in those kind of high density brown treesnake areas. Some of the BTS might be live bird specialists and just not really interested in taking dead bait, whether mouse or birds. And it's also worth noting that some of the largest brown tree snakes may actually survive a single dose of acetaminophen, which is on the baits that were provided. And the large ones tend to be snakes that do go after birds. So, you know, despite some mixed results, I mean, it's a complicated study. Our study does have kind of positive indications that suppression can be possible even in relatively bird-rich areas on Guam. And I say that because this is a relatively bird-rich area, and we still had, you know, a big chunk of snakes that were taking baits in that area. 270 snakes removed with bait tubes, additional snakes caught in traps. So it is possible to control snakes even in bird-rich areas on Guam. And the current trapping and baiting practices can be useful in urbanized environments, albeit with some limitations and a lot of room for improvement. And so, you know, hopefully in the future, those tools can be used and they could be fine-tuned. And so in areas where aerial delivery isn't an option, these tools can be useful in combination potentially with other tools like visual searches, even putting transmitters on fledglings can be a good way to remove snakes. And you know, these types of control in bird-rich areas could be the difference between a declining population and a growing population, a stable population. So it's worth pursuing this further, most definitely. And with that, I'd like to acknowledge all our collaborators, our college ARM, everyone at Andersen, and then all the different crews from all the different agencies. You know, your work is important, and thank you for your efforts. And hi, Nora. Oh, they're gone. Oh, bad. Yeah, happy to take any questions.
Hey, Martin. Do we know enough about snake movements to add to our list of speculations that maybe snakes are from outside or supplanting the snake that are being taken from the bait tube traps? And basically, should we just put more bait tube traps everywhere?
Yeah, good question. I mean, so, you know, like there's a couple of indications in that direction. I mean, there was some ongoing telemetry work on snakes in that area, so there'll be a lot more results kind of coming out, especially from USGS in terms of BTS movement. But yeah, from what we can see, I mean, there's a couple of things. So first of all, clearly snakes were in the areas closest to the forest. Snakes were probably coming in from the forest into the urban areas. In the urban areas, you know, it's a lot more broken up. There's a lot more barriers to BTS movement. But I mean, you're right, like our plots were relatively small. And anecdotally speaking, after the baiting phase, you know, given enough time, it seems like snakes were kind of moving back into areas where for, you know, quite a while we weren't seeing any predation. And so having kind of like a long gap after the control, it's almost inevitable that eventually some snakes will move in. So yeah, I mean, it does need to be sustained.
Great, thank you.
Hey, Martin, thanks for doing this. This is good questions and great answers. And I'd like to know, are you going to be expanding this research, continuing it maybe in other locations? Or I think it'd be really neat to do something like this where even the cocoa are present as well too. So anyway, just thank you for putting together this research project.
Yeah, thanks, Suzanne. I mean, so this specific project, you know, not sure exactly where it's headed, but yeah, I mean, I appreciate the comment. And I mean, I do think that, you know, this type of thing needs to be done more in the future. So, you know, happy to talk and kind of brainstorm ideas. And I think it would be, you know, really, really good to just kind of figure out how we could have birds, you know, breeding on Guam, native birds, and, you know, how to kind of fine tune the brown treesnake control.
Yeah, it's because some of the that project that we did back in 1997, where we had cocoa released in an area up north called Area 50, and the birds were still reproducing with their we had removed a whole lot of snakes in the area, but they were reproducing at the same time the snakes living in the area. So it'd be great to have work like that presented. So good.
Let's make it happen.
Thank you, Martin.
So this next one is from Eben Paxton, who's on Zoom. But Eben, do you want me to use your recording or do you want to go live? And let me unmute you first.
I mean, I think you just do the recording, but I could try to find I could pull up the presentation, give it live.
Totally up to you. Recording's right here, ready to go if you want to do it.
Let's, yeah, just do the recording. That'd be quicker, easier.
Oh, good.
Off a day. My name is Eben Paxton, and along with my co-authors Marcos Gorison, Paul Cryan, Megan Parker, and Frank Alig, presenting our talk on Jajaguac population dynamics. The Jajaguac, or Mariana swiftlet, is a cave-roosting species of the southern Mariana Islands and was once considered abundant across these southern islands. But beginning in the 1960s, there were significant declines, and today they're only known from 3 islands, Saipan, Agiguan, and Guam. On Guam, Jajaguacs are once common from both the North and South Island, but declines began in the 1960s and 1970s and was listed as an endangered species in 1984. They disappeared from all known caves until until the mid-1980s, when they were discovered in 1 and then subsequently 3 caves within the naval base lands in southern Guam. At the peak, there was an estimated of 1,500 individuals. But recent counts indicate a sustained decline from 2016 to 2021, with the most recent count being the lowest population size in over 15 years. These declines are reflected in all three caves. So this is a population-wide decline and not any issue that may be specific to one cave and not the other. If we look at this trend over time, we can break it down into three distinct periods. From 1985 to 2005, there was a relatively stable period with populations fluctuating, but between 200 and 600 birds. From about 2005 to 2015, the population increased by around 7% annually, peaking at its highest levels, around 1,500 individuals in 2015. But from 2016 to present, the population has been decreasing by about 12% annually. And if this current decline continues at the rate that we've seen over the last 7 years, the Jajaguac population in Guam will fall below the 1985 to 2005 historical average within 5 years. Before we delve into possible reasons for the decline of the swiftlets in recent years, I think it's valuable to also understand what may have caused this increase from 2005 to 2015. This was a period of initiating BTS control in and around the caves. Morn and Amidon in 1996 searched Moloch Cave 126 nights, capturing 33 brown tree snakes, with 6 of them having swiftlet remains in their stomach. In 2000, a BTS trap perimeter surrounding Moloch was established, and between 2011 and 2012, a total of 112 BTS were captured in those traps in the perimeter around Moloch. And in 2012, bait tubes were added to the perimeter. So a lot of snake control being conducted around the caves during this time period. In addition, from 2011 to 2017, USGS did in-cave searches for brown tree snakes and removed 194 during that period. 28 out of 43 that were captured, about 2/3, had swiftlet remains in their gut. So clearly, brown tree snakes are present predators for these birds within these caves and taking a number of birds as well. Other reasons that may have contributed to this increasing population size is that BTS populations island-wide may have peaked in the 1980s and have declined subsequently, so predation pressure isn't as high as it was in the 80s and early 90s. It may be that the population is recovering from a heavy period of cyclones in the 1980s. It also could be recovering from a period of pesticide use, such as DDT, that were heavily used in the 50s and 60s and early 70s. So what are some of the reasons for declines? Well, the BTS removal program is not as intensive as it was during the earlier years, both in-cave searches and the perimeter trapping and baiting. And so it may be that brown tree snake levels are increasing, which is causing higher mortality in the swiftlets. Other reasons could include food resources are declining, which would lead to lower productivity and survival. Disease and pesticides are always a possibility when we see declining populations. There's no evidence for either of those, but it's something to consider. Natural populations fluctuate, and so we may just simply see a return to normal, that sort of 200 to 600 level from 1985 to 2005. It's possible other threats such as disturbance and invasive insects could be contributing as well, though again we have no evidence of higher levels than what there was in the past. Interestingly, swiftlets are declining on the other islands where there are no brown tree snakes, and so there may also be regional factors contributing to this decline. So to unravel this mystery of why this population is declining, we need to understand the vital rates of these populations, and this includes survival. What is the adult survival rate? Does it vary among caves? Is adult survival at a sustainable level? Productivity. What is the average productivity across the year? Does it vary among the caves? And is that productivity level plus the survival rate sustainable? And importantly, recruitment. Are juvenile birds surviving and returning to these natal caves? Or are there other occupied caves that may be drawing some recruitment potential from these NAVMAG caves? When we think about managing for long-term persistence of the Jajaguac, while multiple factors may contribute to their decline, BTS are clear predators of swiftlets, and effective management of BTS at these caves is certainly going to benefit the long-term prospects for these populations. There's still many unknowns on BTS threats and predation rates at these caves. And so evaluating different approaches for sustainable control of BTS in and around the caves would be fruitful, as well as developing methods to detect and track the BTS in caves. Jajaguac are the only native Guam forest bird species persisting in these forests, and management of BTS and habitat around the caves could have the double benefit of helping recover this endangered species and be a focal area for forest restoration and restoration of other Guam native forest bird species. And the last point I want to leave everyone with is a lesson hard learned from many other areas, which is that the smaller the population gets, the harder the recovery will be. We've seen a sustained decline for a number of years, and if this decline continues, it'll become harder and harder for us to pull these birds out of this population decline. I want to thank the generous funding that we received from NAVFAC Marianas and Joint Region Marianas, and we really appreciate the field assistance that we've had over the years at these caves. Mahalo.
Hey, Eben, thanks for the great talk. This is Noah. I'm just curious if you're tracking what the swiftlet decline is on the other Mariana Islands and how that compares to Guam.
Yeah, I don't, it's a really interesting question, Noah, about the declines regionally at the other islands. You know, that is, you know, that is unknown, basically. I think there's been some recent work, Marco Scorison and others working with CNMI folks of trying to recensus the Jajaguac as well as the bats on Agiguan, and you know, there may be more swiftlets there than some past counts have indicated, so that may be hopeful, but that is definitely something worth I think collaboration across the islands to try to get at why we might be seeing a large regional decline.
Are you looking at any other species that taking the bait out of the bait tubes in these areas?
There's been, you know, that's not my thing, but I know there's been a lot of work with folks doing cameras to look at, you know, what's called incidental bait take from these tubes. You know, my understanding is a number of different things. Ants are probably the biggest sort of stealer of dead mice from these tubes. You know, certainly the swiftlets wouldn't be interacting at all with those tubes, and there are no other native birds out in these areas. But I do know there's been a number of studies looking at that, just making sure that there isn't any concerns on that. As far as I'm aware, they're not.
Hi, Eben. This is Ellie from CMI DFW. I am wondering, do you know what the cave monitoring effort looks like outside of these three kind of known occupied caves? Or are you searching for other potentially occupied caves or monitoring the occupied caves at all?
Yeah, that's a great question. So we're actually doing a lot of work at these caves trying to understand, well, a number of things. I mean, we're working on a lot of different, trialing different cameras to be able to quantify numbers. So a lot of these counts that we've rely on to look at changes over time are based on observers outside of the caves, you know, in the evening when swiftlets are returning. Which, you know, is the best sort of way we can do it in terms of an eyeball survey, but you know, it's often getting dark. It's hard to see the swiftlets. You can sometimes get a whole bunch that come in at once or leave at once. We're now finding with cameras as well as some telemetry we're doing that a number of birds won't even come back until well after dusk, and in the middle of the night. Some birds even skip a night of coming back to the cave. So there's obviously a lot of dynamics in terms of when they're coming in and out of the caves, which have implications for counting. Like I said, we are doing some telemetry work to try to see where these birds are going across the landscape. That's really challenging because the transmitters we put on them are very small, have very limited ranges, but that's something that we're working on. And yes, we're very interested in seeing if there's potentially other caves that are being occupied. I mean, people do see swiftlets in other areas that may indicate other colonies, but that's not clear at this point.
Hey, this is Suzanne, and Ellie also said, carry on with that conversation. I know with Noah, and Eben, we spoke with you as well too. There could be some other cases out here that we're patiently waiting for the dry season to arrive, so we can hit some rivers and hopefully check out some new locations, and hopefully at DFW.
Thank you for the presentation. It was great. I recall that there were populations of the swiftlets in really suitable cave habitats that were Tinian and Andersen Air Force Base in the past. And I'm just wondering if, besides monitoring for their return, if there are any proposed action to perhaps transfer some birds and reestablish populations where they used to be employed. Thank you.
Yeah, I didn't catch all of your message, but I think you were talking about previously occupied caves. A number of them up in northern Guam on Andersen. That's something that we're very interested in and trying to figure out ways to reestablish swiftlets, jajaguacs on other caves. I think there's a lot we can do if there's an unoccupied cave in terms of trying to snake-proof it, if you will. And then try to figure out ways to either encourage sort of self-colonization of those caves or actual do types of translocations. There is the possibility of social attraction. It may be that these birds, as they fly around the landscape, do visit these caves from time to time just to see if they're potentially suitable. And so I think there's a lot of work that can be done on that. I think certainly the ultimate goal for long-term stability of these birds is to get them in more caves over more areas. And that's certainly within the recovery plan guidance as well. So there's definitely, I think, a lot of fruitful work that can be done to identify good prospect caves for future colonies and then figure out ways to get birds into those colonies once we've been, but first making them snake-proof before these endangered species occupy it.
I have a question. So with how Guam's forests are moving into more of these more like non-native forests, could the swiftlets not be getting their preferred food? And what is their preferred food?
Yeah, diets could potentially be a very important factor. They are believed to mostly be eating small midges, termites, flying ants. It may vary. Through our banding, we have, recovered some what are called bolus, but little balls of insects are bringing back to nestlings, and those seem to be mostly, small flies and midges. Which would suggest sort of that they would be spending a lot of time over aquatic areas. But again, this is something where there's, a lot of potential for more research on this. I mean, there's underneath the roosting areas, there's the swiftlet guano that could be analyzed for diet, as well as, as we get a sense of where they are foraging on the landscape, we can then start looking at, possibly available insects in those areas and maybe get insight into why they're in some areas and not others.
Thank you, Eben.
So that concludes the first part of our session. So thank you for being patient and waiting for break. So we have some refreshments in the back.
Half a day, everyone, and Jesus Maasi for having me here today to talk to you about the impacts and outcomes of AGA rear and release. Today I'm in Oregon, and I wish I was in Guam with you guys because it actually just started snowing, which is rather crazy. So the AGA rear and release program on Rhoda is run jointly by the San Diego Zoo Wildlife Alliance and the University of Washington. Our goal is to raise AGA eggs and nestlings at our facility in Rhoda and then release them back into the wild to increase the population. Through this program, we have now raised 6 cohorts of 8 to 15 birds, and we've released 4 cohorts back into the wild. Today, I'm going to summarize the survival and reproduction of captive reared AGA and also try to answer the question, how do these nest collections impact the reproductive success of wild AGA? So why rear and release as a recovery strategy for this species? The AGA has relatively low reproductive success. 50% of pairs of breeding pairs produce young each year. And that's not to say they have 50% nest success. Their nest success is more like 30%. But since they can re-nest, 50% produce by the end of the year. 30% of fledglings die during their first year, and those that survive have an 8-month period of parental dependence, such that a pair can usually only rear 1 clutch per year despite their ability to nest year-round. This year-round nesting and propensity to re-nest after failure are key to rear and release. Theoretically, we can double the reproductive output of pairs we collect from if we collect their eggs and nestlings early in the season, and then they go on to reproduce at a normal rate by the end of the season. So let's ask, do these pairs actually go on to have kids at a normal rate after we collect their first clutch? With 5 years of data, we can begin to answer this question. Before I get to our answer to that question, let's look at this forecasting model made in 2013 by Fred Amidon of the U.S. Fish and Wildlife Service using program Vortex. This model was foundational for the decision to use rear and release to try to recover the AGA on Rhoda. First, I'd like to point out that this is a 30-year forecasting model that explores the impact of rear and release, as well as mortality reduction in the wild and captive breeding in a variety of combinations. I'll ask you to ignore all of the lines except for the yellow, the blue, and the black, which I've made bold for you, because the other lines represent scenarios that include captive breeding, and I won't be discussing those today. We'll be focusing instead on scenarios that include 5 years of captive rear and release, as well as mortality reduction in the wild of juveniles and adults. So these 2 measures for encouraging population growth in the wild are different but complementary. Rear and release is a tangible program that we can achieve, that we are achieving. Mortality reduction is more difficult because we don't know why the birds are dying or how to stop it. So let's look at this model. The black line is baseline with no intervention. The prediction's dire. The blue line, slightly above it, includes 5 years of captive rear and release at the onset. So this bumps the population up a little bit, but then the system falls back to baseline after rear and release ends. You know, neither of these scenarios is going to work. If we look at our yellow line, this is a combination of rear and release and mortality reduction in the wild. So here we see a positive trend, increasing numbers of wild AGA on Rhoda. And if you wonder about mortality reduction on its own, that red line beneath the yellow is mortality reduction alone. So still a good outcome, but you can see that rear and release sustains the population at a higher level. So now after 5 seasons of collections, let's see how things look on the ground. So our program, I'm happy to say, is really excellent at hatching and rearing AGA for release. Aside from the utility of comparing our numbers to the vortex model, I find it most useful to look at our success rates from the standpoint of viable eggs, since taking infertile eggs from wild nests has no cost to the wild birds. So here you can see that from 6 collection seasons, we took a total of 80 viable eggs or chicks, and 74 of those, or 93%, survived to be released or to be considered releasable. Many of these birds are still young. Now, the 2013 vortex model assumed that approximately 25% of collected eggs and 90% of collected nestlings would produce potentially releasable birds. I need to explain that this egg rate in the vortex model represents all eggs, not just viable eggs, which is a really important difference because 41% of all aga eggs are non-viable at collection. Most of them are infertile. So if I were to use the same metrics as vortex within our program, 57% of all eggs and 94% of nestlings produce releasable birds. The vortex model also assumed that the collection of eggs and nestlings would result in a decrease in wild fledglings during those years, which is of course true if those pairs couldn't compensate by renesting. So I'm finally going to answer that question. Is it true, or can they compensate? Here we are comparing seasonal reproductive success of pairs that we collected from in pink versus those that we left alone, blue. Most adults in the population are color banded, so we sorted our data by pair ID. And you'll see that the proportion of successful pairs on the y-axis and breeding season is on the x. You may notice if you're looking closely, the 2018 is missing. That was Typhoon Mangkhut, no collections that season. We see there is no consistent trend, no significant difference between the groups in the proportion of pairs that successfully fledged young from wild nests by the end of the season. Here is a bar graph showing cumulative proportions. Again, this is success of all the years combined for pairs that we collected from on the left and those that we did not collect from on the right. And there is no statistical difference between the groups. Another question I had was regarding the number of fledglings produced by these two groups. In this graph, the mean number of fledglings produced is on the Y, and the breeding season again is on the X. We see, once again, no meaningful difference between groups in the number of wild fledglings produced by the end of the season. And remember, the fledglings that we produce in captivity are not represented here. Here's the corresponding bar graph, again showing no difference in number of fledglings produced by the end of the breeding season between these two groups. So back to the vortex model again briefly. Remember that one of the assumptions here was that the post-release survival would be lower for captive reared birds than for wild birds. Of course, it makes sense. In the vortex model, 45% first-year survival rate was set for the first year post-release. So let's look now at how many of our 46 birds that we have released so far on Rhoda, how many survived their first year. So first year survival is 70%, and cumulative survival for all the birds we've released to date is 65%. Both of these are much higher than the inputs used for the model, and so this could significantly change the population forecast for rear and release if we redo the vortex model. And since we're talking about survival, it's only fair that I touch on causes of mortality for released birds. The primary necropsy findings for all released birds are listed in this table. The range of causes is similar to what we see in wild birds. The majority are dying of inflammation, chronic antigenic stimulation, and often with no lesions that are severe enough to explain death. This is extremely frustrating. We have also had 5 due to predation, a lot easier to understand. 5 to predation. This was 3 by feral cat and 2 by monitor lizard. And while there's a lot more to say about this topic, I'm low on time, so I will move on to a summary of nesting for the many birds that survived. We have found 28 nests built by released birds and their mates. Some pair with wild birds and some with other released birds. Here is our first fledgling from last year, produced by a captive reared female and her wild mate. We actually didn't find her nest. She snuck this kid out after making a long distance dispersal when she was no longer radio tagged. You can see her in the background. Her unbanded kid is in the foreground. He was too nimble to catch by the time we discovered him. Here are some summary stats on nesting by release birds by release year. And this is one area where we need to see improvement in the coming years. The 2013 Vortex model assumed that surviving released birds would breed at the same rate as wild birds. However, our data here on the ground indicates some level of delayed breeding or lower productivity than expected. I would say don't pay too much attention to the bottom two rows. Those birds are still young, they're still settling in. But the top two, we would have expected more nest with contents, which means eggs or nestlings, and more fledged young. I will leave you today with a quick summary. In my opinion, it's incredible that we have been able to create 46 extra adult aga at negligible cost to the wild population. And remember, this is 46 extra adults in a population that has less than 200 birds total. This result, along with the high success rates of captive and released birds, are hugely positive signs that this program can make a difference in species recovery. That said, we're left with a few challenges. Why are reproductive rates of released birds so low? Perhaps they need more time. Perhaps we'll learn something useful in our upcoming nest camera study. As we speak, one of our captive reared females has just produced her second fledgling. Another pair has eggs. Others are thinking about starting something, but hey, maybe they're millennials and they would have rather stayed home and gotten free food a bit longer. Or maybe they're Gen Z and they're like, have kids. What about the future of our species? Whatever the reasons for delayed breeding, the wild population appears to have stabilized, partly due to the survival and recruitment of captive reared birds into the breeding population. I would like to end this presentation by thanking the numerous partners and individuals who have made this work possible: U.S. Fish and Wildlife Service, CNMI DFW and DLNR, Rhoda Office of the Mayor, and finally, Guam DAR. Guam DAR folks, we would be literally unable to feed our captive birds without your help in procuring food for them. We would not have been able to quickly rebuild our aviaries when they were all destroyed in the typhoon. I hope that you are here today and listening to this, and I hope that you consider the successes of this program your own as well. Thank you.
Yes, your question.
I'll ask again. So, what is the natural lifespan of these birds? And do you know if wild birds learn and get better at fledging young as they get older?
So the lifespan of these birds, we believe, is in the 20s, maybe up to 30. Our oldest breeding bird was, I think, in the low 20s. And certainly the wild birds are still breeding like crazy through their teens. So these birds do have time. And we don't have enough data on known age birds yet to say whether they improve over time, but I can certainly say that like young birds during their first attempt often will kind of play around with nesting and not seem that serious about it. Maybe they don't even lay eggs. And then the following year, they'll get more serious. So I hope that partially answers your question.
There's a question from Zoom from Lauren Stump. Is there a particular reason captive breeding was not considered an ideal recovery strategy for the aga?
Yeah, so captive breeding is extremely expensive and resource intensive. You need to have incredible infrastructure, large aviaries to hold pairs that are separated from each other because they're territorial. So it's kind of a last resort. And the hope was to do something a little bit less resource intensive and have that paired with, eventually mortality reduction be enough. But captive breeding is still on the table if it's needed, and there's enough resources to support it.
Hey, Sarah, great talk. I think you might have mentioned this and I just missed it, but how did your survival rate for released birds compare to the survival rate of your wild population for your fledglings?
Yeah, so the release birds survived at a higher rate than wild fledglings. First year survival of release birds is about 70%. First year survival of wild fledglings is more like 60%. We did expect it to be higher because we're releasing them as adults. So there's certain vulnerabilities that the fledglings have that, these birds have overcome by surviving to age 2. We release them at about 2 years of age, but they have a higher survival rate than fledglings, but a lower survival rate than wild adults. Wild adults survive at about 90% rate per year.
Great, thank you, Sarah. Appreciate it.
Thanks.
All right, how about now?
Yeah.
Good, sweet. All right, so I'm Henry. We have heard a number of excellent data-driven talks this morning in the bird section. I want to just tell a story. This is a story that is personal for me, as I imagine it is for many others in this room. The best way to start here is to admit that when I first came to Rhoda in 2013 to begin studying the crow, I could not imagine that there could be widespread apathy within the community of Rhoda towards the recovery of the species. This was it was plainly naive on my part, but I think it's helpful to out myself here because perhaps being value system blind is a common bias among researchers in our field. Like most issues, it can only be solved once we admit it exists. There's much more to be said here about colonization, the history of the Marianas, decolonizing science and academia, but these topics are going to be a bit beyond the scope of my story today, so I want to keep it more narrowly focused on the crow. So, shortly after settling in on Rhoda, I made a friend who asked me, "Why do you care about the crow?" After I gave my answer about fragile island ecosystems, global biodiversity loss, endemic species, and just why crows are really cool, my friend, he wasn't impressed. He used a really interesting turn of phrase, and he told me, this is a quote, "That's a nothing bird." I thought, that was something that had not been sort of directly communicated to me, and it really stuck with me. And it was a sentiment that I would hear repeated many times on Rhoda. The general concept here is good for nothing. Can't use it, can't eat it, isn't pretty. For my friend, the value of the aga and perhaps all wildlife lies in its utilitarian value. I know for certain that not everyone on Rhoda agrees with my friend. I've heard stories from elders who demonstrate both intrinsic and ecological services-based value systems. But the point here is that the public perception of an endangered species should be really important for conservation biologists to understand and to incorporate into their work. There are many hypotheses for why the aga population declined on Rhoda. On Guam, the decline of the aga is well understood, but we're still trying to answer this question of why the crow declined on Rhoda. This graph shows the decline over time. In 1983, there were roughly 1,300 aga on Rhoda, and today there are just around 200 individual birds remaining. That represents an 85% decline. So researchers have proposed habitat loss, predation by cats and monitor lizards, nest predation by rats, inbreeding, competition with other introduced species, disease, and human persecution are all factors that have, contributed to the decline of the aga on Rhoda. Of these, human persecution is a factor for which we have the least data. You'll notice there's something that most of the aga decline hypotheses have in common. They focus on the nuts and bolts of the aga's population and its habitat. They involve biological research. They want to diagnose biological problems. This makes sense because biologists are going to biology. That's the tool in our toolbox, right? But this focus on biological issues conveys a value system. A critical hypothesis yet to be explored requires engaging human beings on what matters to them. And with that in mind, let's consider what matters to the community of Rhoda by looking at significant events in the relationship between the community and the aga. So first, the species is federally listed in 1984. In 1990, the CNMI government passes the Rhoda Agricultural Homestead Act. And this act sets the rules and procedures for a program in which the government gave applicants 1 hectare of public land for their own private agricultural use. Given the history of the CNMI and geopolitical context, this program was extremely popular, and there was a high demand for agricultural lots. But this period in red here indicates a period of human-wildlife conflict, which began in February 93 when CNMI TPW recorded incidental take of the aga while someone was clearing their agricultural lot. In response, CNMI Coastal Resource Management placed a cease and desist order on the homestead program, and residents were prevented from accessing 600 hectares of agricultural lots that they were now told had been released early before proper permits were in place. Just 1 month later, another instance of take occurred on the Road Resort golf course while the golf course was being developed. Both instances of take involved aga nests interfering with development. Later in 1999, aga were confiscated from the locally owned Rhoda Zoo. And it wasn't until 2014 when the final 244 hectares of the 600 hectares originally released were actually given back to residents, and after the CNMI government and U.S. Fish and Wildlife Service signed a memorandum of agreement. And this agreement added 167 hectares of crow habitat to an existing conservation area in exchange for allowing homesteads within aga habitat elsewhere to be developed. So for 21 years, 244 hectares of land were tied up in the name of aga conservation. Living on Rhoda for 10 years has afforded me the opportunity to hear many sentiments characterizing aga conservation. By far, the most common thread in these discussions is a rhetorical question. What is more important, the people of Rhoda or the bird? And put a different way, we just frame this slightly differently. It seems the manner in which aga conservation has been conducted has demonstrated to the people of Rhoda a set of values. And in this set of values, there's no room for their own or for any compromise. And the years following the suspension of agricultural homestead lots, the slogan "Kill the Bird" became popular. Some invested in the slogan more intensely than others, but the phrase was the ubiquitous signal that rages against the set of values demonstrated by conservationists. So, how to address this question? To what degree did human persecution factor into the decline? The question poses a serious challenge for ecologists hoping to collect decades-old mortality data. Many of you have probably already heard of this emerging field that's been labeled human dimensions of conservation. This field leverages social science to answer a simple question: what do people do and why in regards to endangered species? It recognizes that now in the age of the Anthropocene, humans are the most powerful factor impacting wildlife populations and the environment for better or for worse through both intentional and unintentional actions. It recognizes that social and ecological approaches to conservation have not been well integrated, that understanding human value systems enables effective conservation decision-making. For examples of how this is getting applied in other contexts, please see these two excellent articles or come talk to me about them later on. Earlier, I said that nobody has studied the human persecution hypothesis of aga decline. This is true. However, in 2010, one group of biologists from UW studied the way the people of Rhoda relate to endangered species. This study, led by UW's Adrian Sussman and Dr. Renee Ha, asked 72 questions to 573 respondents. Taken as a whole, the results of the survey were a strong indication that more attention needs to be put towards addressing human dimensions conservation on Rhoda, and that more work is required to quantify human persecution's impact on the aga population. Sussman and colleagues found that almost half of 573 participants agreed that they would be happy if there were no more crows on Rhoda. 38.8% agreed that most people think crows are a bother. 52% condoned chasing crows, 12.9% knew someone who shot crows, and 16.4% agree it is very acceptable to shoot crows. The most significant actions taken to address human-wildlife conflict around the aga are the 1999 production of an educational video titled Rhoda, Our Island, Our Future. The University of Washington's own public perception survey, which I just mentioned, the 2013 establishment of a landowner incentive program, which paid landowners to leave aga habitat untouched on their land. The 2014 signing of the MOA that released homestead lots back to the people of Rhoda and set aside additional land for conservation. These efforts deserve to be lauded. However, UW's public perception survey, along with our conversations with the public on Rhoda, indicate that sustained long-term effort needs to be focused on human dimensions of aga conservation. We would like to repeat Sussman's survey with additional questions that characterize the impact human persecution has had on the aga population. After all, public perception is a dynamic property, and there's an ever-present risk of its conflict escalating. We'd like to acquire funding specifically for addressing human dimensions of conservation. We perform education and outreach every year, but there is just a really wide gap between what needs to be done and how much we can do with funding that is primarily intended for population monitoring, augmentation, and research. And finally, we'd like to be joined by our partners in adopting a guiding philosophy that recognizes the ways we can do better to tailor our conservation work to Rhoda's unique history and values. This is important for the recovery of the aga, but as I stated in the beginning of my talk, we want to ask the entire community within the Marianas invest in this idea that human dimensions are important in our work. The impact of negative sentiment does not just contain to the endangered species, a single endangered species, right? It spills out and is inevitably applied more generally to values that are demonstrated by conservation as a whole. This dynamic is demonstrated by a common joke told amongst the elder generation on Rhoda, including my friend who called the aga nothing bird. In this joke, anyone who is arrogant, closed-minded, or comes across as a know-it-all is jokingly accused of being a scientist, regardless of their occupation. On the surface, it's really funny. It is a good joke. But if you think about it for a second, it really does offer scathing evidence that we can do better to carry out our work in ways that include human dimensions and expand the idea of what it means to carry out conservation. I hope the story of the aga and this pitch for human dimensions inspires everyone from students, all the way up to the regulatory level to expand their vision of conservation, not just to a field limited to protecting biodiversity, but rather a field that protects both biodiversity and the way our community relates to it. Thank you. And that's my talk in one slide. That's my talk. Thank you to everybody that, provides funding, the people of Rhoda who grant us access to private lands in order to conduct our research, and all the partners who make our work possible. Thanks. Yeah. Thanks.
I had a quick question regarding attitudes toward the Marianas Pro. How much do you think attitudes about conservation and federally listed species in general might be linked to the CNMI's political status and the relationship with the U.S. in general? Because we know there's a very long history surrounding that.
Yeah, quite a bit. I think quite a bit has to do with that. In fact, so much that it's hard to even know where to start to answer that question. But I think you're definitely hitting the nail on the head. And I wish there were formal ways in which we could, as a conservation community, address that very problem. Ways that we can, expand our focus beyond just, measuring, reproduction rates in endangered species and nest failure, nest success, survivorship, et cetera, but to dedicate attention and funding to addressing that type of political and social issue, because really, everything's connected. Yeah, I wish I could be more eloquent. Yeah. I mean, economics also plays into a huge role there and plays a big part and is part of that very question as well. So.
Hi, Henry. Thank you for that talk. I was just wondering if anecdotally, if you noticed any difference between generations and their attitudes towards conservation.
I do. Yes, that is a great question. The generations that feel the most negatively towards the crow are the generations that were most impacted during the 90s in conflict over land. The younger generations are bright-eyed and bushy-tailed and are ready to learn and are excited about wildlife. So there's a very big difference over age groups that I think is important in to keep in mind while we address this topic.
Yeah, I think that was going to be an issue for a lot of people, and I know myself as well, where our views are in direct conflict to our parents, and trying to wrestle that, might really affect how you with people and how you respond to questions.
Yes.
I just wanted to ask, what is the case that you usually make for the pros when you run into those people that are for the killing of them or would rather see them gone?
The first thing I always do is recognize that the conservation community can do better. That recognize that there are things that went wrong, and without pointing fingers, recognize that we're aware of that and that we aspire to do better. The next thing I move to is just the value of biodiversity and the idea that we don't have the power to choose what wildlife populations are convenient to us, and that we prefer over others. everything's connected again, yeah, out there, and we need to be aware of that as we're getting our hands dirty and trying to save some species and trying to actively suppress others.
Quick question, well, maybe more a comment. This actually explains a lot for some of the work that we saw for parallel conservation efforts and at the same time for other people in Rhoda. So really thank you for that retrospective analysis. I was wondering, with respect to like those surveys, is there a sense that with some of the questions are asking what are some of the economic benefits that maybe sort of the federal footprint might be having for the local economy over there?
Would you just repeat whether you asked economic benefits specifically, or?
Yeah, the economic benefits of, having basically people, conservation biologists doing work there, and money in the economy, or more importantly, probably, this kind of cuts, I think, what you're talking about here. Is there a perception that maybe there's job opportunities, capacity building for the local community?
Yeah.
So
yeah, that's something that was not directly addressed in the survey that I do think is important. Firstly, the economic benefits of having wildlife research done on Rhoda is not really recognized. It's not really talked about. I think generally it's seen because of this issue and issues with Panihi, is seen as more detrimental and as a hurdle barrier to overcome. But to that end, a major part of solving this problem is going to be to make sure we demonstrate that wildlife biology, that conservation is a viable career within these islands, and to really on every level in the education system, early career, young adults, to make sure the opportunities are there for them and to make sure that we are inspiring interest and dedication to the field, because that ends up feeding back to the community economically and also in terms of biodiversity.
I'm gonna go for just a tag onto that. There are two human dimensions researchers, one Dara Wald, who's worked out here, and another Ashley Dare, who is on that paper, who would both love to take on students from the island to work on questions like this. So if you are an undergraduate or thinking about graduate school and are interested in this, definitely come talk to me and I can connect you with them, because I think it would be amazing to have more human dimensions researchers from the island trying to figure out how we get through the tough social, kind of how we do a better job of conservation in general. So.
Hey, Henry, great talk. So a few years ago, I had a colleague that did a human dimension study looking at the relationship between wolves and ranchers in Eastern Washington, which for those not familiar with that area, is an incredibly contentious relationship. And one of the questions they asked was looking at an incentive program. And to her discouragement, what she found was that a lot of ranchers wanted no part in it because they were just already so mistrustful of the local government that they didn't want to work with them at all. So I'm just curious with your landowner incentive program, what was the reception? And I think the reason that this point is so important is because sometimes we have tools that from our perspective as conservationists seem really effective, but if we don't address these underlying relationships, we can't actually use those tools.
Right. Yeah, so I think what we run up against is the rigidity of a system that isn't really designed to handle a wide berth of contexts and situations, right? When things get really challenging, often it's hard to apply a general model to a bunch of different specific situations and have that model fit. And so I think it's really important to keep in mind that as things grow more unique or more challenging, we need to try harder and put a greater effort into just reacting in a way that's appropriate to that unique context. And so with the landowner incentive program on Rhoda, there were challenges in interest, there were challenges in trust, and you know, there were challenges also in matching the things that we could offer within the program to the things that work for the landowners themselves. And yeah, so I would just advocate for greater flexibility in finding solutions to really meet problems rather than taking models from elsewhere that have, you know, maybe worked for one context, but.
Okay, hi everyone. So my name is Ellie Broark. I work for the CNMI Division of Fish and Wildlife. And today I'm going to give you an update on the progress and future directions of the Mariana's Avifauna Conservation Program or Plan, also called the MAC Plan. And some of you might be familiar with this program as the CNMI's Bird Translocation Program. So the program arose out of concerns about brown treesnake extirpating bird populations from Guam and the threat of brown treesnake introduction into the CNMI. The goal is to create redundant populations of our most vulnerable forest bird species in case of brown treesnake introduction in the CNMI and extirpation on Saipan, Tinian, or Rhoda. So in order to do this, the DFW, CNMI DFW partnered with U.S. Fish and Wildlife Service, the Association of Zoos and Aquariums, and Pacific Bird Conservation to figure out how to develop these redundant populations for our native birds. And what they came up with is a three-pronged approach to bird conservation. But today I'm just going to be talking about the translocation portion of the MAC program, specifically summarizing which translocations have been completed so far and how we've been following up after translocation. So the idea of the translocation program is to capture individuals from Saipan, Tinian, and Rhoda and bring them into captivity and then release them in the northern islands. So that's everything in this orange box there, north of Saipan. And the thought is that the threat of brown treesnake introduction is significantly reduced in the northern islands because there's far less traffic of people and goods moving around up there. There are some homesteading settlements in the northern islands. They're not all completely unpopulated, but they remain relatively undisturbed in terms of anthropogenic threats and human development compared to southern islands in the archipelago. So far, we've moved species to 3 islands, Surigan, Guam, and Alamagan. So between 2008 and 2013, we moved 4 species to Surigan. And then we moved on to Guam, where we also moved 4 species, and we have moved 2 species to Alamagan. So that's 5 species total that we've moved so far. We started with non-listed, not endangered or threatened bird species to kind of provide proof of concept before we move on to endangered and threatened species populations. So the important question here is, how are these populations that we've moved doing? And we have been back to do some manner of follow-up surveys on 2 out of these 3 islands. And this is what we found so far. So in follow-up surveys to Surigan in 2016, we found that the bridal white-eye, golden white-eye, rufous fantail, and Mariana Fruit Dove were all still present. That's what the P indicates there. And we also found unbanded individuals of bridled, golden, and fantail, indicating that they successfully bred on the island. So we know that they bred. What we don't know is whether the populations are increasing or decreasing. Did follow-up surveys on Guam in 2022. And we found that the Tinian monarch and rufous fantail were present and detected very commonly on our surveys. We released fruit doves in 2017 on Guam and in 2022 again. So on our 2022 follow-up visits, we couldn't really evaluate how the fruit dove population is doing. Bridal white-eye was absent from Guam in 2022. We have not yet been back to Alamagan to conduct follow-up surveys, so we can't really evaluate the persistence of those bird populations yet, but we did have a DFW team there for an unrelated project last year, and they anecdotally have reported unbanded golden white-eyes on the island, so that's indicating that they're breeding there. But the biggest story here is that we have evidence of bridal white-eye nosa on Alamagan, which is an island where they have never been detected before now, and they were not intentionally introduced. We translocated them to Guam in 2015 and 2016, but in 2022, we did not detect a single bridal white-eye on Guam, and the team on Alamagan did detect them. So in terms of answering the question, how are the birds doing up there? We've proved that we can move these birds successfully, and they can survive and breed in the short term on most of these islands. The difficulty is that these expeditions just take an enormous amount of resources, huge amounts of money, time, labor to put together. And a single follow-up survey on these islands really can't tell us much about how the population is doing because it doesn't allow us to estimate the normal population variability. Or in other words, how much the population just kind of naturally fluctuates over time. So we need to be able to estimate that population variability in order to figure out population trends at any scale finer than just colonization and extirpation. So right now we can say whether the birds are present or not present on those islands after translocation, but we really can't tell whether the populations are increasing or decreasing. So to combat the data scarcity situation up there, we are working on developing a monitoring program to deploy automated recording units, or ARUs, in the Northern Islands. So these are little audio recorders that we can program to record at certain times and leave in place to collect data over really long periods of time and then return later to retrieve the data. This is a photo of an ARU on Guam last year. The amazing advantage of ARUs is that they let us massively increase survey effort by having kind of ears on the ground, so to speak, without actually having to have biologists physically present. So this is going to let us increase the resolution of our data to account for population variability and be able to track trends over time, which is great. But it does present some additional challenges in terms of data processing time. As you can imagine, recording for months and months is just going to produce a huge amount of data. And we are going to need manual or hopefully automated ways to process the data. So as Willson was talking about earlier with those automated detection algorithms for fruit doves. But another advantage I particularly want to point out is the ability of ARUs to provide verifiable documentation of species occurrence. So here is a spectrogram of a nosa calling on Alamagan. The field crew on Alamagan thought that they had heard the bridal nosa while they were there, but they were not focused on bird surveys. They had a lot of other stuff on their plate, a lot of other stuff to do, and they really weren't 100% sure whether or not they heard it. It was unexpected up there. But they had deployed an ARU so that we could collect at least a little bit of bird data while they were on the island. And sure enough, they got home and we had confirmed documentation of the bridal white-eyed presence on Alamagan, and that's the first documentation of this species on this island. So what this tells us is that it's likely that the bridles moved from Guam to Alamagan. We can speculate about why they weren't happy to stay on Guam, but the important thing that this points out to us is the meta-population dynamics in the Northern Islands and in the archipelago in general. So even populations of our very smallest forest birds on these islands are not isolated and restricted only to the islands where we put them on. The movement of populations between islands is part of the bigger picture here, and it's part of the population dynamics for these species, and we're going to need to consider that when we're thinking about conserving populations of our native forest birds. Another important consideration that I want to discuss is the species area relationship for these islands. So here we have land area on a log scale on the x-axis and the number of terrestrial bird species on a log scale on the y-axis. Each island is a different color dot, and that's showing the number of terrestrial bird species found there pre-translocation. So we can see something here that pretty closely adheres to this straight line relationship that we would expect on a log-log scale, where the number of species increases with the land area available. So we see that strong positive correlation between land area and species richness. Now I'm going to add points to the plot to show you what the species richness looks like on the islands where we have translocated species. So the black dots that I've added here show post-translocation species richness. So Guam, for example, which is all the way on the left there, went from 5 species present before translocation to 9 species present afterwards. So that's a huge jump. That's almost double the number of species, and the island is just about 4 square kilometers. So eventually, we would expect that the number of species on these islands is going to come back down towards the mean number of species that we expect based on the species area relationship rule. What we don't know is the time scale for this or which species are going to be extirpated. So if any of you have thoughts about how to estimate or predict the relevant time scale, I would love to talk to you more about it. But generally, from a conservation perspective, we need to be aware that we just don't know how long some of these islands are going to hold inflated numbers of species. So in terms of what this means for the future of the MAC plan, as we work on long-term plan development, we're going to need to account for metapopulation dynamics and species area relationship concerns. Our immediate next steps are going to be to increase our follow-up monitoring survey effort using ARUs and to develop processing methods for the resulting data. And we're now at the stage of preparing for the translocation of listed species. So we're planning to conduct a full environmental assessment. And this spring, we're going to deploy ARUs on Alamagan to collect data for the next year. And then in 2024, we're hoping to deploy on Group 1 and Surigan as well. And we're beginning to develop workflows for data processing, but we're not going to be able to come up with automated detection algorithms for all species completely on our own. We're going to need partnerships with people like Willson or any of the rest of you who are also working on this kind of thing. So the proof of concept phase for the MEC plan and translocation of these initial species have been really successful, but moving forward into a monitoring and reassessment phase is really going to be critical for us before we start moving listed species. So this program, as you can imagine, is just a tremendous effort for conservation of our birds. It takes a lot of work, and many people, including some of you in this room, are involved in it. So thank you so much to all of you, and particularly to our Pacific Bird Conservation partners, one of whom you are about to hear from next. We would not be able to do this work without them. So I'll take your question.
Hey, Ali. So are you guys putting species back on island, back in places where they occurred historically, or are you putting them back where they didn't necessarily occur?
Not necessarily where they have occurred historically.
And then the idea is that they'll sort themselves out?
I think the idea is that we just have some population redundancy in case of brown treesnake invasion, so that if, bridal white-eye, let's say, which is found in the southern islands, and as far as I know, has never historically occupied the northern islands. If the brown treesnake were to invade, we don't want that species to be extinct. And so having a kind of redundant or maybe reservoir population in the northern islands would prevent extinction. The question is just, what's the time scale on which the populations we've established in the Northern Islands are going to be there to provide that redundancy.
Good talk. Thanks, and thanks for this great effort. I'm just wondering if you do risk assessments to understand the ecological and trophic effects to the native populations that are there, the islands that you're referred to.
Yeah, I think the environmental assessment that we are about to conduct is focused on kind of future listed species translocations, but it's also going to be retrospective, focused on the previous translocations as well. I think there was a real sense of urgency and even emergency when this project began, because the threat of treesnake introduction into the CNMI just felt like something that was extremely imminent. And so there was a sense like we just have to move the birds so they're gone. And now I think we have a moment where we're kind of trying to take a breath, reassess, figure out what the impacts of the work we've already done are and what the impacts of our potential future work are.
Hi, Ellie. I had a question, it's kind of related to the first one, but more specifically, how do you decide what species go to which island, specifically in respect to the species within the cohort and their relationship with food dove?
That's a great question. That's something I've been thinking a lot about. Historically, the map, I wasn't part of the development of the original plan, but there was a lot of talk about kind of like niche availability, habitat availability on the Northern Islands, and prey species availability, that kind of thing. I'm not sure that really competition between cohorts of species was necessarily considered, but I'm not 100% sure. I wasn't part of those discussions, but I think that's something that we need to be considering moving forward. Absolutely. I think there are some like competing flycatchers up there and other species that might be, potentially affecting viable population levels for both species.
Hi, Ellie. Great talk. Thank you. I was just wondering, have you looked into, well, first, how many individuals are you releasing when you're releasing a species into the islands? And then have you seen any evidence of migration between the larger islands and the smaller islands, as in Rhoda, Saipan, and Tinian and the other islands? And have you thought about the possible impacts of bottleneck effects in the future?
Yes, so the cohorts of birds are typically about 50. It depends on the species. The Mariana Fruit Dove cohorts have been 25, and most of the smaller passerines have been 50 individuals at a time. And we try to release 2 cohorts for 100 birds total that are released on the island. Sorry, what was the second part of your question? Is there migration between larger islands? Thank you. Yes. So, yes, we have seen some evidence of like independent colonization of the island. So, for example, Sarreguin, they detected a fruit dove before they ever released fruit doves up there. And the island doesn't have an established fruit dove population, but it seems like at least one fruit dove arrived before we released our cohort of fruit dove. So, yeah, there is some movement. And I think, on Anatahan this year, we haven't translocated birds to Anatahan, but that's kind of the largest northern island that's closest to Saipan. And this most recent set of surveys that DFW just did up there found a couple of new colonizations on Anatahan of birds that had never been reported there in the past. So it's happening. There's definitely movement from the southern islands.
Thank you.
Yeah, good morning, everyone. It's going to be, well, my name is John. I'm a PhD candidate at University of Missouri. I'm also in Haldre's lab and a member of Pacific Bird Conservation. It's kind of great that I'm following Ellie's talk because I'm just going to go through basically like a fun methods talk of just how you move all of these species. They have very different requirements. So, keeping them alive and trapping them and bringing them up on these boats, a whole lot goes into it. I'm going to be talking about the Canario, the Chichirica, and the Totut as my main examples because all 3 of them are pretty diverse. We've also moved Nosa and Chicharican Tinian. If you have questions about those after the talk, I can talk to you about those 10. So yeah, I'm just going to go over this really fast because Ali kind of talked about it, but here's a big chart of the species that have been moved, the number that has been moved, the years, and the source island versus the island they were brought to. For Pacific bird conservation, we mainly partner with AZA zoos. So in a typical translocation year, we'll have about 30 to 35 individuals come onto the islands. It's mainly Saipan from about 8 to 12 different institutions. This includes veterinary staff, education staff, and biologists who handle a lot of the collection and husbandry work. Zoos are just like really good at keeping animals in captivity for long periods of time. They can deal with all the different needs that these different species have. And they're good at getting funds for on-the-ground conservation work that are needed. We also have local students and biologists that work with us closely on all aspects of this work because this is their project, their species, and their island. They just asked us to assist them in reaching their conservation goals. So yeah, here are the 3 main species, the Totut, the Canario, and the Chicharica. We're going to go through capture and misnetting, do holding prior to translocation, and then the moving and translocation of these species. So the first thing to talk about is just the netting logistics. In order to move the birds, you have to catch them first. We use 2 different sites for the smaller passerines. And for the Totut, we use a bigger, like a ranch style field. All the birds that are caught on Saipan are in the northern region in Marpi. We use private land that we've got access to from landowners and relationships we built with them. DFW helps us access the land and make those connections. The main nest site for the Chicharica and the Canario are right before the Calavera Cave. It's in a Tongan mixed limestone forest. And the Totut is right after Calavera Cave, if you're familiar with that area, in a much larger ranch field. We usually have to worry about getting a lot of birds in a very short amount of time so that we can get them on the boat and up to the islands. So that's kind of what makes the decisions for where we're picking our land. The Totut field, unfortunately, there's a invasive plant in there that we haven't identified yet. It's a green, hard fermented berry, but they all congregate and eat it. And we can catch about 20 birds to 24 birds in 2 to 3 weeks. If you're netting Totut in like really nice limestone forest, it's really hard to get them in that amount of time. And we usually go April or May based on DFW's needs, with the boat trips, typhoon avoidance, and just better wind and weather. On the left here is just a single high mist net. It's up with a fishing, it's an extendable fishing pole. We use those for a lot of the passerines. And then on the right, you can see the double to triple high, more intense conduit steel poles that you need for the Totut as they're higher in the canopy and they're cruising just kind of faster and harder to get than in this dense forest. Here's some individuals taking out a Totut and Chicharican. The CNMI birds aren't really hard to misnet. They don't present like major challenges. They're not very tangled. The hardest one to get out is the fantail a lot of the times. A lot of times they're chasing each other. They're very territorial. They'll spiral into the net. They're kind of high stress birds, so we want to get them out very quickly and into the holding cage that we have out in the field. So after we net the species, the next step is to bring them into captivity before the translocation occurs. And depending on when the bird is caught, it can be with us either 1 to 14 days. So if you're caught, when the boat's about to leave, you'll probably only be in our care for 2 to 3 days. If you're caught right in the beginning, you might be with us for 2 weeks. Before they can go into the holding room, they need to have an examination by the veterinarians and the vet techs. They take all vitals, screen for parasites, take blood and fecal samples, and they kind of have to okay every individual bird before they're allowed to go into the holding room and up to the islands for translocation. As of 2022, there were no local vets on Saipan. There were no local vet techs, and we have a goal to try to have the project eventually be led totally by local veterinary staff and local vet tech. So if there's any students in the room that are considering vet school, please come talk to me. We also do corticosterone testing on the birds, so just testing their stress. This is Chicharica from 2018. You can see that on day 0 and day 1, the stress levels are kind of they're not as baseline as you see in day 2 to 7. It takes them a little bit to get acclimated. Here is a Canario. It's a little different. Day 1, you can see they have more variability in the stress levels that they go through, and then it kind of tapers off. You can see the Canario are showing a little more variability in the corticosteroid levels than the fantails were. And then this is just a health assessment table of Canario from 2018. You can see that we record the band numbers, the exam dates, the weights, the fecal parasite tests, the blood parasite tests, body condition scores, and just any exam notes. So NSF just is standing for no significant findings. So after they've passed all the veterinary exams, they can go into the bird holding room. And this is typically what it looks like. We have these conduit pipes, racks holding all of the bird boxes. There's 1 bird per box. And you can see that folding table is kind of in the middle. That's where all the staff prepare the diets. The veterinarians do anything they need to do, any cleaning work that needs to be done. And on the left here, you can see the smaller boxes are for the smaller passerine birds, and the right boxes are for the Totut, are much larger, and they have these cloths hanging off of them. The cloths we need to use because we have to actually grab the Totut to feed them because they have a special feeding requirement, which I'm about to go into. So all 3 of the species I'm talking about today have vastly different requirements. The Canario is mainly sustained on a normal diet of papaya, mealworms. They start eating out of the bowl really quickly. They're very, like on Canario years, people are usually really happy and they know they're gonna have a pretty normal trip. The Totut and the Chichirica have pretty extreme diets that we need to make up for. The Totut on the left, you can see is being tube fed. So they consistently go off food when they come into captivity. That is just a response that they have. It takes them about 2 to 4 weeks to start eating out of bowls. And because the holding time for the translocation is shorter than that period, we need to tube feed them the entire time. So they're tube fed about 4 times per day with a supplement that's mixed with a lot of local papaya. It keeps fat on them. It helps them maintain their weight. They do very well on this. We've moved about 80 fruit doves over 4 years, and this has always been very successful. I work with the Ecology of Bird Loss Project and have held fruit doves on Saipan for many, many months, and they really do start eating about day 15, 16 out of the bowls, and then they're fine. The methods to successfully translocate the Chichirica are a lot more intense because they're just an aerial insectivore that presents a lot of problems because they're just flying around the woods all day and eating insects. So when you have them in a box, you need to solve that problem. And we do that by these fly buckets that you can see here on the right. This is not everyone's favorite job. We have to basically catch flies the entire time we're here and supplement their diet of mealworms that we bring from the mainland with the fly diet. So we take these big serving platters, we put mesh on them. Underneath the mesh is a water and dead fish mixture that we get from local fishermen. You put that out in the jungle and you get flies very quickly. It smells terrible. It's everyone's least favorite job. We consistently have to replace the fish, so it's very expensive. It's very time consuming. But you basically get all the flies to attract to your bucket. They kind of go into this inverted cone. When you need to feed the fantails, you invert the cone. We have little petri dishes with holes in them. You place that over the cone. The flies go in there. Oh, I forgot to talk to you about an important part of our box setup. The petri dish goes on the boxes have these removable floors, so we never have to interact with the birds after we've caught them. So you can kind of crack the door a little bit, move the floor out, replace the bowls and the food sources, and then put the bowl back or put the floor back in. That way the bird never sees you and it helps keep those stress levels down. But with the petri dish, you kind of have to make sure you don't lose all your flies, slide that floor back in, and then the flies go into the box and you can hear the fantail hunting all day long. Here's just a quick photo of fruit doves. We weigh them before and after their feedings. There's another photo of a crew member feeding a juvenile fruit dove. And then here's just a chart of Totut post-capture body weight in grams. Each color is a different Totut. They kind of oscillate back and forth, whether it's AM, PM, plus the feeding schedule that we put them on. And then after all of this, it's time for the translocation. We put them in different holding boxes for translocation. They're a little smaller so they don't get banged up on the boat. Each box holds 5 birds. You can see us leaving on the boat here. We usually use the Super Emerald. And then when we get to the islands, on the left here is Guam. There's a little skip there. The birds need to be loaded safely onto the skip and then onto the island, and then, you know, off the rocky shore, which sometimes it's very difficult. If there's big swells, you have these boxes of birds and the skip is going like this and banging against the rocks, and you're trying to get the birds safely. We've had some people spill over before, so that's once that part of the trip's done, it feels good. And then we bring them to the release area. The release area is often kind of heavy cover with plants and trees, and it's difficult for the birds to, you know, birds like to get elevated right away. So we have to stack the boxes like this so the fruit doves don't have to just kind of take off from the ground. So we kind of give them the most elevated space that we can. But that's pretty much the translocation methods in a very quick nutshell. Before I take any questions, I wanted to talk a lot about PBC's education and community outreach. We have a mission to serve local communities where our work's being done, and it is a core component of our project. And I would say that as of 2018 or 19, we do just as much education and community outreach as we do research. We have a team of 4 to 5 educators that are solely dedicated to doing education out in the CNMI. We work with the CNMI school system, which is our main partner is Asap Ogamoro. So thank you to him for helping us reach our education goals. We typically can talk to 12 to 1,500 people in a 4-week trip. We go to the flame tree festivals, libraries. We provide buses so that the students can come out to the field sites and see the mist netting opportunities. We provide buses and field trips so the students can get experience with the veterinarians, the animal care team. And we can just kind of show everyone all of the different career opportunities that you could have, not just field biology. Here's another last photo of elementary school we started doing this year. And something really great that we did this year as well is we mostly work on Saipan, so we don't get to go to Tinian and Rhoda a lot. And we wanted to make sure that we're providing high school students on those islands the opportunities as well. So this year and the years moving forward, we're using all of our funds to fly students and their teachers to Saipan from Tinian and Rhoda so that those students can also get the opportunities. That's pretty much everything I have for you today. Thanks for listening to my talk, and I can take any questions.
Hey, great talk. So I noticed you mentioned that you guys take blood samples. Do you know, and this might also be a question for Elliot, were you guys determining sex ratios of these birds before you were releasing them, or even after the fact?
Yeah, so we can determine the sex ratios after the fact. We don't have the technology to do it as quickly as possible before the birds leave for the translocation. We do a big report after the fact with our veterinary team, and we can know how many males and females went up to the island. As the technology is getting better, we might be able to do it on island someday, and that'd be really important if we move listed species. We definitely want to know more of the sex ratio before you move them, especially if you're moving individuals in low numbers. But yeah, we can't do it on island right now before the translocation.
For the next question, can I just ask all the speakers from this past section to hop up to this table? And let us know if you need more chairs, so we know there's a bunch of you. But you can keep asking questions.
Great talk, John. I'm just wondering how where the funding comes for Acoustic. Are the zoos like paying, like they pay to send people out, or do they pay even if they donate? Still don't they don't send people out because they prefer? How's the funding? How do you guys get the funding for the?
Yeah, that's a great question. So PBC is very small. I'm the only paid employee right now. So we're funded from a grant we get from the Disney Fish and Wildlife, Disney Conservation Fund. That's kind of our big grant that we have. All of the zoo folks pay their own flight and their own, they pay a fee to Pacific Bird Conservation, and that pays for their hotel, rental cars, helps pay for all the resources that we need, food. So it is from this combination of zoos and grants.
Yeah, given Ellie's previous talk about a bit of a pause in translocations, how will PBC's role change? How do you stay engaged and stay relevant?
Yeah, that's a great question. So we dealt with that last year. We had a big meeting with Division of Fish and Wildlife. Ellie and I were kind of talking back and forth on this. As Ellie mentioned, we're going to, it's not, announced yet, but we are moving into the stage of working with listed species. So our plan right now is to make sure that we can bring those listed species into captivity with little to no mortality events. If you're talking about reed-warblers and rhoda wide-eye, you want to make sure that we're doing the best possible things we can for these animals. So we are doing method years. So before we actually move them, we're taking them into captivity this year. On rhoda, we're taking rhoda wide-eyes into captivity. We're seeing how they respond to captive environments. We're doing basically everything that we always do, except we're not doing a translocation. So we're going to do that with other listed species in the future. The next couple of years, we might do reed-warbler, so that we basically are, just getting better at translocation, better at our methodology before we start moving these very low number species on the island.
Right, this is going to be our last question before we jump into the panel. So if you still have questions for John, feel free to ask him and generalize it to the whole panel.
Thanks, John. Awesome talk. I'm just curious, based on your experiences, are there any species that you would not recommend for translocation where you feel like you've had too many failures? Or are you feeling confident that all of these species are still on the table for future translocation?
Yeah, I think that Pacific Bird Conservation is working very carefully and making sure that we are good at translocating all of these species with special needs. The fantail is a species that a lot of folks do not want to work with, institutions refuse to work with. Something that PBC's, we've had some mortality events with fantails, but it's something that PBC is very proud of, that we've been able to keep a 6-gram aerial insectivore alive in these holding boxes for weeks on end, put them on these boats, bring them up to these islands and keep them alive. So I would say the fantail's the trickiest one, but we've shown that we can successfully move them.
Thanks. Thank you, John.
All right, we're gonna start the panel, and I think we'll get Eben and Sarah up here on the screen in just a second as well. And this has been a long morning of talk, so with lots of bird research in the island. So and we have a lot of panelists here. They probably won't be able to hear from everyone. But I want to hear from maybe a few people. We think a lot about individual bird species one at a time, and I'm curious what community, like how we might, if there are things we're missing, by not thinking about the community as a whole. Like we often do like a monarch study or a reed-warbler study or a moving another bird species. Are there things that we might be missing by not thinking about the community as a whole? Or where would you start if you were going to start? Or what things maybe have we done that have been really good at, like, kind of thinking about bird community interaction? I'm not thinking about plants here. I'm thinking about
Yeah, I'll think of first. First of all, I think as Henry well illustrated, the community includes the human community as well. So that's like, step one. But then, thinking about it on Guam, I think it's a pretty interesting question because, in some respects, we'd be pretty much starting from scratch in a forested area. And so, as I think it was Kaeli who mentioned, Såli can be nest predators. And so there's a lot of interest in bringing Såli to kind of different locations. But is that, necessarily the best idea if you want to have, maybe some of the smaller forest birds established first? Definitely thinking about, the whole ecology of seed dispersers, insectivores, and the overlap is, something really important. So I think, those are really important questions. And then, when it comes to, getting to translocating to the northern islands, getting to potential carrying capacity and stuff. Ellie touched on it pretty well, but that's like, another kind of level. So maybe you want to expand on that a bit.
I think there are actually some really interesting questions about community assemblages for birds in the southern islands too. Like, Tinian monarch and golden white-eye, not co-occurring, even though golden white-eye is present on Saipan and on a Guam. The question of like, are they competitors in some way? Are they like excluding, mutually exclusive, or is it an accident of extirpation and then they just haven't managed to recolonize? Tinian monarchs haven't managed to colonize the Guam or Saipan, and vice versa. Golden white-eye hasn't managed to get to Tinian. I don't know, and I think like thinking about those things in kind of a larger context is important. I'm not entirely sure how you would structure a study to like go about trying to figure that out.
Yeah, we've attempted to look at species distribution models based off of the island survey data, particularly for Saipan. I built out a whole series of species distribution models. There is a data, there's some data limitations because we're looking at essentially either temporal or spatial limitations in terms of how these samples are collected. It makes it really difficult to either validate the outputs of these models, but it's possible to get some better data that we can take all the various distribution models, overlap, and then start looking at assemblages in some sense. But it takes a lot of data to develop that.
Yeah, just to add kind of one more maybe point on this. I think, there is a question of kind of shifting baselines. So, we think of maybe, and Ellie kind of mentioned it, like we think of the Northern Islands as relatively pristine, but I think all of them have some number of invasive species on it, including rats. And research from other places has shown that if you get rid of rats, then the carrying capacities can go way up. The forest can recover a lot. So that's something I'm sure you guys have thought about at CNMI DFW, but maybe something to kind of look forward to in the future is maybe increasing that overall baseline for species of carrying capacity in the whole archipelago.
So one thing that we did not share during Fletcher's talk on parasitism, that it's actually not just the white-eye. We have seen that but we don't have as many observations for some of the other species to make as good of a talk. But I think that's a really nice example that, sometimes when we get so hyper-focused on these individual species, we sort of miss the forest through the trees. And I also think the reason that's important, particularly here, is we have a really cool opportunity because these communities are really small to ask more tangible questions about evolution in these cohorts, right? Because it's not as complex a question as if you're trying to do some of this work, say, in Costa Rica, where you have much higher biodiversity. So, I'm going to relate this to kleptoparasitism because that's what we've been spending a lot of time thinking. But I think we have a really interesting opportunity here to look at evolutionary histories of how something like that kind of behavior can evolve and spread potentially across the communities, and then kind of retroactively look at that to help understand this behavior in other parts of the world.
One more question for the panel, and we'll open it up to the audience. Something now expanding to think about the human community. I think that was a really powerful talk, Henry, and it's something that we certainly need to recognize a lot more. And so I'm curious from the panel's perspective, kind of other examples that might be bright spots, places where the community has really been like a key partner in helping to with birds, or opportunities that we haven't taken advantage of, or other strategies that we could use. I think Henry identified a few, but if you have thoughts of like how to do a better job of not just being scientists doing science and ignoring the fact that you're embedded within a human community. Anyone?
Yeah, I know I talked about it briefly, but I think providing just like simple logistics a lot of the times when you're out here working on research projects, whether that's providing buses for schools to come to conferences like this, or go out in the field and do field work with scientists, or have internships, flying different students to different islands, just anything like that that can bridge the logistic gap and get more boots on the ground of the students that are there. I think that really improves it. And I didn't have time to talk about this in one of my examples, but Amanda was one of our interns that went through BBC with us, and then she did a college of bird loss internship, and now she's a biologist at the Division of Fish and Wildlife, and it's just, great to see full circle. I wanted to say about that.
I know this is a bird panel. I want to recognize that the subject that I've spoken about, it's not a novel concept. It's not, this is something certainly that is happening in the community. It's attention to human dimensions. This is not a new thing, and there are bright spots in the community. It's easy to point out, on Rhoda Forestry is taking volunteers to Totut to fight erosion. Rhoda Forestry disseminates seedlings, native trees to the community. The CRB project on Rhoda is doing a lot of education outreach to inform the public on the impact of CRB invasion. Here in Guam, like tapping into citizen conservationists that remove snakes from Sundano. I think the photo, very bright spot, a lot of potential there. Again, specific examples with birds are not coming to mind right now, but generally, we've seen a lot of allusions to the value of attending to the concept of human dimensions in the talks, on Tuesday, in talks today. The outreach that CIS is doing. There's a lot that we have to be excited about. We still have a lot of work to do. It'll be really great to get more focus on marrying our interest in protecting bird species with developing or investing.
I just want to say that, Henry and others have thought about this a lot more than I have, but there's a lot of birds that people have really positive feelings about. One of the reasons I really enjoy working with the Totut is that if you've been to the Tinian Island, Saipan, you hear that bird everywhere around the island. Like every single person knows that bird. And it's kind of very embedded in the culture, it's a mascot of the schools. I see people come in, there's sports teams named after the Totut or the Kingfisher. I think there's a softball team that's Kingfishers. And I just like to focus on those positive things. I've never met a student yet who dislikes Totut, Fruit Dove. So that's just a sort of a way to get them involved, but they might have very little knowledge. And so we can sort of highlight those birds that people already like and are already part of the community.
Yeah, I've got just a couple more examples to add. First of all, shout out to Olympia for the Migratory Bird Festival. That was a huge success. The other day, half the groups here had tables there. Probably more than half, probably everyone here was there. It just kind of was a great celebration of birds on the island. The migratory birds here, half of them spend more of their time here than they do anywhere else. So they are very much native birds as well. And then also, in terms of the CHEC and work with that, all the work that Department of Ag does, and a lot of public outreach, a lot of kind of social media and community outreach. I think there's a position opening up this summer, so that'll be pretty exciting to have just a lot more kind of engagement on the CHEC and its recovery. So that's something I'm looking forward to.
Yeah, just putting all of that, I just want to say that community outreach is really important for Fish and Wildlife. We do our annual Christmas bird count, so the whole public's been invited to that, and we go birding around all over the island. You guys can be new to birding. We'll help you guys identify the bird by sight or sound. Yeah, we also go to a lot of schools and talk to students, and when we also did the map station, we invited a lot of students to come watch us catch birds, and same when John invites everyone. Students to watch them catch birds too. Just having them involved hands-on is really important because they get to see what we really do.
I just want to quickly observe that most of the shout outs that we're giving to groups that are doing this work, tapping into human dimensions, focus in there. Are groups that are starting to ground up. These are people that are recognizing the value of this work and finding ways to make it happen, or using resources and that already exist and finding ways to do like more education, more outreach. I would like to see more of that leadership coming from the top down, rather than mainly from the bottom up. And that's a conversation that I would love to have. I'm not policy maker or regulator, but I think there's a lot of work to do from the top down.
All right, one more thing, and this is just to build off of what everyone else here said. But coming from Tinian, we've been doing our own very small scale outreach campaign. We've spoken to schools, there's camps, we've done land birding trips and stuff, but at a very small scale. And compared to the other large southern islands, I feel like there is much smaller representation of what we're talking about here. So I would really like to invite every one of the agencies that's represented here. I appreciate Don was talking about bringing Tinian students to Saipan to be part of that, but obviously that's not available for everyone. So anyone who would like to come to Tinian and expand your organization here, there is plenty of need for it, and we will really appreciate it.
I think we have some questions from the audience. That's a great topic. Yeah, go for it. See, do you have a microphone?
Sorry. I know Mike has a question that is going on.
Okay, go for it, and then can we get a microphone?
Thanks a lot. I have several questions, but I really like your all the others. Maybe two quick questions. Years ago, when I visited the Northern Islands, there were residents on Assumption, Modern, even. Of course, they've left. But it seems like there was a family on Alamaga that's pretty long-term still there. Is there any attempt to enroll them in monitoring the birds that are there?
So we have not asked them to help us do like formal survey assessments, but I will say the folks who live on Alamaga are really helpful to us in a number of ways. We have collaborated with them. We often, just are like, hey, yeah, do you want to ride up north? Do you need to get on our boat? Because they're amazingly helpful folks, and they have really facilitated a lot of the work that we've done there in terms of trail cutting, data collection, navigation on the island. Their local knowledge is phenomenal.
Related to that, the population of could that be a potential area for some of the transportations too, and using the local people there to monitor? As well as I think it's interesting people wanting to go there kind of time.
Yeah, I think there's a lot of possibilities, and we're hoping to have a formal meeting between DFW and the new mayor of the Northern Islands. There's just been a new administration elected, and so I think, yeah, we're hoping to facilitate those conversations in a formal way through the Northern Island Mayor's office. Pagan had initially been kind of off the table for translocation discussions because of the high probability of DoD development of Pagan. And kind of as that situation has unfolded, I think there are certainly ways to reopen that conversation at this point.
I had a question. If you guys have ever observed any of our native bird species being a candidate as a predator or a form of biocontrol for like the the coconut rhinoceros beetle?
Yeah, so the straight up answer is no, except for in my wildest imagination and dreams. Recently the CRBs dispersed en route from Lizo area, the peninsula across Sassananda Bay towards Igani, and they have now entered sort of into the crow's range, crow habitat. I'm talking with CRB team about seeing if we can figure out in a more controlled way what happens when Aga and CRB meet. It's kind of an interesting idea right now, the crow population is so low that I don't think like it's reasonable functionally to expect the aga to be sort of a to help us out there. But theoretically, crow population is really strong. Knowing crow biology, crow behavior, crow diet, like it does seem reasonable that would be a really popular thing.
Yeah, I mean, if we're speculating here, maybe my top candidate, I mean, and some of the CNMI folks might correct me if I'm wrong, but maybe like the Sasangat, the megapode might be a strong candidate for bird that would dig around and eat out those tasty grubs. So I mean, let's bring it back to the Guam.
Hi, I just have a question for the translocation folks, so John and Ellie. You mentioned that the kind of the goal of these translocations is to provide a redundancy in the population should brown treesnake get to the other islands. Wondering, with all of the resources and funds that are being put into developing new methods for control and inspections and preventing those invasions from happening in the first place, what do you see as your end goal for these translocations? Do you have like a defined stopping point where you feel like the populations are secure enough?
This is something that has been coming up a lot recently, and I would love to talk to Trey about it. Actually, we'll hear from him later. But I guess I don't have a good sense of like what our sense of security should be on Saipan from brown treesnake invasion at this point. I think the kind of stated goal of the MAC plan is to try to have redundant populations of literally all of our vulnerable forest bird species. So right now, the species that we just haven't translocated and don't have redundancy for are, rhoda white-eye, the reed-warbler, and the swiftlet. We have the swiftlet's on multiple islands, but there are no northern islands that would be appropriate for swiftlet reintroduction. We've been talking about reintroduction to rhoda for that species. Anyway, I guess I see the question is really about some of the issues that I was talking about in my talk that I don't have answers for, in terms of like what is the timeline on which the northern islands populations that we've established can be redundant or reservoir populations. And I think it gets back to this 3-tier approach. I think that the idea of the map would be to be constantly reassessing the security of populations through both the redundant populations in the northern islands, the status of the in situ populations that are already established in the southern islands, and then our captive population. There's a captive breeding program associated with the map plan stuff too. I feel like that was kind of a convoluted answer, but I hope I got at your question a little bit.
I can just add thankful that Saipan doesn't have a, persistent brown treesnake population if there is a small one. It just seems, just in the 8 years that I've been there, whether it's super tycoons hitting in and a lot of logistics and FEMA coming in, or unchecked developments with, casino and things like that, it seems like the island is getting, it gets developed and things flow in like crazy. And I think it is hard to keep all of those brown treesnake security checks on that island when things like these events happen and ramp up, and not all of these containers are getting checked. We have measures in place, but sometimes Saipan goes through these peaks of extreme funds coming in or development happening, and it hasn't happened yet, but it definitely could still. I think with how much funds and just things happen sometimes. I guess that's my short answer for being so slightly nervous.
And I'll just clarify, there's no evidence of a population on Saipan right now. I just want to make the clarification. It is very possible that it could happen in the future.
Thank you. There is.
Hi, I have a question over here. I too am a little nervous to ask questions. I don't know if I'm like asking it correctly, but concerning like everyone who is doing like bird studies, has there been any evidence of like urbanization or any microplastic waste or human waste being involved with nest making? And is it an issue? And is it comparable between like Guam? Because I can imagine there's more urbanization here than the other islands. Or is there any waste chemicals within the birds that can be hindering their reproduction? Thank
you. Yeah, I'll just start real quick. I mean, well, so in terms of plastic in nests, I mean, with a Såli, we see a little bit, not much really, but I mean, you know, semi-regularly we'll see some plastic in it. So that's definitely does happen. Yeah, I mean, on Guam, it's really interesting because on Guam right now, like the urban areas are the hotspots for birds. So in terms of, I mean, Såli, Kakak, Tungi, they're all really using the urbanized areas as their kind of nesting and roosting areas because, you know, that tends to be where they could find little pockets away from brown tree snakes. Not to say that's like a solution or necessarily like even a great thing, but it's just kind of like the situation on Guam right now.
That's a really terrific question. To answer for the monarchs and other forest birds on Tinian, we haven't seen that, but we do see plastic in our naughty nets. Even up in the MLA, they take big strands of like tarp and I don't know, big fishing material. And I think that is a really important question because we do know with some birds of osprey, for example, and some seabirds that can cause nestling mortality. And I don't think anybody is tracking that, but it would be worth looking into. And then likewise for microplastic consumption, we wouldn't expect to see that in the forest birds, but we really need a seabird person up here to better address that question.
So I also do wedge-tail shearwater birds, and
we have a seabird person.
So a couple years ago, someone found a shearwater, and it was decomposing, and like in its stomach, there's like old plastics, like all this plastic waste in it. So we have a colony up in Managaha, and every now and then I'll see some trash, but it's not something very noticeable. But most of the time, the area is like kept.
We have two more questions. One is over here first.
Thanks over here. Great talks from all the panel. Back to Ellie and John, just back to this idea of introducing the birds to non-native birds to islands and moving forward, like what Mike said, so maybe Hog and other places. Moving forward, is there going to be an effort then to do the risk assessment beforehand? And this is all just to couch this, right? This is sort of in research that we know from Aldrin's group and others, right? That the massive trophic effects that these birds have. Just looking at Guam, we know that with the birds gone, there's effects to all the communities. So the real assumption then, I think that introducing a non-native to an island that's going to have all kinds of trophic effects.
Yes, I think it's exactly right, and that it's a really substantial issue that I think we need to grapple with. And in my ideal world, we would revisit the same island year after year and be collecting kind of ecosystem scale data about the effects of translocation. The reality of the pressures to maintain monitoring effort on all of the islands and the limited resources and funding and personnel to make those things happen just makes it difficult to do that tracking. But I think it's extremely important and relevant to be considering those trophic effects. And I think we've made some dramatic changes to a couple of the Northern Islands and the species communities on the Northern Islands, and we should expect that there will be some effects from that. So it's really, I mean, with all the translocation stuff, it really is a like risk and reward cost benefit equation. You know, we care desperately about conserving the bird species, and at the outset of this project, you know, the kind of feeling of emergency of trying to establish those redundant populations was outweighing a lot of the other concerns about what might happen after introduction of these species. And I think now we're at a reassessment phase. And that's an important component of the reassessment.
I just want to add on to that, and this might answer Fletcher's question from before as well. So prior to translocation, there is a bioinventory surveys that happen on the islands to look at recently prey availability, certainly the species that are already present on the island, as well as vegetation, habitat suitability. And that is taken into consideration. So no bird has been moved to an island where it didn't already coexist with, you know, the species that are there on the southern island, at least. Whether or not that still may answer the competition question or not is to be looked at further. But I think moving forward, we will initiate some efforts to potentially look at food availability on future islands, either in terms of a number of trees that are available for certain species that are frugivores versus also insect prey availability.
And just to add on one more thing about the Northern Islands, I feel like we also believe that the species that are present on them are constantly kind of oscillating through time. So just because a species isn't there when DFW is doing a survey doesn't mean that there was a small population that could have been there that, you know, they're very susceptible to. If a typhoon event hits one of those Northern Islands, it could wipe out a really small population of NOSA, and that, you know, in that time, they would maybe naturally get back up there, but it just takes a little bit more time. There's a lot going on in the Northern Islands that we don't know. And I think the species are kind of fluctuating like that, and there's extinction events and immigration events happening kind of naturally. And we have seen species, DFW has done surveys and seen species on certain islands, and then they go do a survey again and it's not there anymore. And as Ellie said, the NOSA got to Alamagan, you know, assuming it came from Guam, but that is also, we don't know. Those NOSA could have come from another island as well. So there's a lot of stuff going on there.
Last question. I want to commend you guys all on your science, it's been great. But one of the human dynamics that really hasn't been broached in this room that I've heard is we're going to soon have an inundation of tourists. We're going to have millions of dollars spent on tourists, and they're going to come in. And do you think that as a human dynamic, that's a group we should also be addressed? I know the CNMI, almost every one of the rental car maps has a bunch of pictures of birds on it, and that's really fantastic. But do you think that there's room that as a conservation community, we can engage tourists or get basically money from them to, you know, or to increase the value of our birds and other wildlife through engagement with them, which then would also be conveyed to the residents like us that live here and deal with us. So there's a lot of people there. There's a lot of dollars coming in. Any thoughts?
Just as a very quick anecdote, one of those tourist maps that gets handed out at rental car agencies has a bird from Hawaii on it and not my bird on it. So yes, I think there's absolutely an opportunity to improve our engagement with tourists.
Yeah, on Rhoda, the tourism isn't quite strong. Make an understatement that it is but it has been very encouraging to see more leaders in the community putting forth the idea of ecotourism as a viable, possibility for Rhoda's future. And I think you're really hitting on something that we as conservation community need to be part of participating in that and engaging the subject in a way that ensures that the community is going to be receiving on the receiving end of this and benefiting and as will ideally the biodiversity of the islands there. So for sure, even though the possibility of that occurring on Rhoda is not as imminent as it is on the other islands, it is definitely in the forefront of our minds, and it seems like it's an idea that's gaining steam on Rhoda. Super encouraging.
Sorry, they left me with a microphone, but do you think there's room for ecotourism? I mean, I haven't really seen any ecotourism here.
Yeah, yeah, I do. If anybody has not visited Rhoda, I really encourage you to visit Rhoda. It's unbelievable, the degree of beauty that the island holds relative to how well visited it is. There's just so much potential. Ancient Treasure Island. It's called that for a reason.
Yeah, I mean, like I could only imagine that. Well, first of all, when it was still running, Islandano, Takatsukasae, was like a pretty strong tourist draw. Debatable how many were going there specifically for birds versus other reasons, but yeah, I think there's a lot of potential there. But you could only imagine that a site on Guam with native forest birds would be quite the draw for tourists, and that that there could be possibilities for corporate park sponsorship from hotels, that kind of thing. I think it's definitely worth exploring. And then yeah, Rhoda, I'll just and yeah, if you haven't been there, go check it out. Check out the bird sanctuary. Nothing better. If you haven't been to Palau, you could just see the potential for ecotourism. The idea of visitor levies, that kind of thing, and it's all, yeah, I think worth thinking about.
Awesome. Well, go for it.
Shout out to the Puntos again. From Rhoda. Back in the day, he had a nature trail on Rhoda that featured the native endemic species on his property. He cultivated as as an attraction where people who were not living on Rhoda could come and be inspired by what we have in the jungle and in our ecosystem here in the Marianas. It's something that has been done in the past. It's not. There's a model for it, and we can very easily. Maybe not easily, but it is possible to tap back into that.
Thank you. And let's thank our panel. You can all go sit down. We have like the poster presenters. There's 5 poster presenters. They each have 1 minute. So I'm going to keep you for 5 more minutes, and then we'll break for lunch. The 5 poster presenters each have 1 minute to just briefly tell you what their poster's about. And then after they do that, we'll have them grab a plate first and then stand by their poster, so you all can kind of meander around and eat lunch. But 5 more minutes. All you know. So first, can I get all 5 poster presenters just come up here because it'll be pretty fast. All right, So we're going to start with Alexander, and then Jaden, and then Jonai, and then Doug, and then Nation. Okay. Oh, I need to share screen again. And go.
Good afternoon, everyone. My name is Alex Cudella, and I am a student at the Northern Marianas College. I am assisting in a research aimed at discovering whether or not birds in the Sinai have a dog forest.
And if they do, what time that downpour exists. So the downpours is a natural phenomena that typically involves male passerine birds, and it describes their vocal activity as starting from a peak at dawn and shows like a noticeable drop throughout the day. And the overall purpose of this research is to improve the efficiency and surveying of bird communities on the island. And yeah, so if you have any questions, just feel free to stop by and check out my post. Thank you.
Good afternoon, everyone. My name is Jared Lucas. I'm also a student at the Northern Marianas College, and I'm assisting in research on the Mariana Fruit Dove, also known as the Totut. And we are specifically looking at their songs. So birds use songs to communicate with each other to defend their territories and attract mates. And so because songs are a big part of their communication, their songs must be similar enough that other birds of the same species are able to recognize them. And so that's why we are looking at the Totut songs across the islands Saipan, Guam, and Rota. And for more information, you can check out my poster where I can share my results. Thank you.
Today, my name is Tinian Island, and I'm a graduate biology student at the University of Guam, working under Dr. Ross Miller. And my project focuses on identifying the plant species that honeybees forage on Guam. And this is a really important study, a really good foundation study for honeybees. And the aspect of beekeeping, it helps beekeepers understand what their honeybees need to thrive, and as well in our agricultural community. It allows us to understand the basic role of honeybees as possible pollinators to both native and non-native species of Guam. Thank you.
Hello, everyone. My name is Jack Christie. I work for the Institute for Wildlife Studies, and I'm also a student at Humboldt in California. My project is looking at the population of Hilitae on Island Dano. The cocoa were reintroduced, and kind of as a part of that, control efforts began on the Hilitae as a method of kind of controlling the amount of predation that occurs on the island. So my project is basically trying to assess the population, like now over a decade later, using spatial mark recapture methods and as well as photo identification and pit tags. Please come by and visit me in my poster if you're interested in talking about basically any part of that project, you know, be it Dano, reptiles, spatial mark recapture. I'd love to talk to you. Thank you.
Hi everyone, my name is Aisha. I work for the CNMI Division of Fish and Wildlife. I had a little slide, but it's lost somewhere in Google. In 2022, we conducted an extensive island-wide survey on Rota, which if any of you have been involved in island-wide surveys, it's a tremendous amount of work. My poster summarizes our avian and habitat survey data. However, we did survey for herps and crabs, and a bunch of other surveys were conducted at the same time. So if you'd like to come over and talk about the bird surveys and habitat surveys, or any of the other surveys that we conducted, please see.
All right, so what I'd like to have happen is all of the speakers, make sure you get on the food line first so that you can get a plate of food and you are before everyone else gets up there. We have the same, it's a different caterer today, but the same setup. So we have, I think, 2 tables here. And do we have one in the registration room too? Yep, and so we have 3 tables. I think is there a vegetarian one? there's vegetarian tables in the registration room. And the lumpia is vegetarian. And then I think there's some water and drinks and such on the sides. We're going to try to both meet back at 1:15, so that gives us not quite a full hour, but hopefully we can have enough time to eat lunch. So enjoy.
Thanks for everything there.
You're great. Sorry.
And it relies on 2 host plants for the entirety of its life cycle, Acrocephalus and Alatus semicalcareum. Okay, so these are the life stages. On the far left, we have the eggs. We found that the viable eggs are green, and when they're parasitized, they're black. You can find them on the underside of leaves. They're typically found around the edges of the leaf, although not always. The second picture in from the left are the larvae. These are about mid-stage larvae. They get very much smaller than this and also very much larger. Their bodies are full black and they have orange cycles emanating from them. Another species of Hypolimnus, which is also found on Guam, has red face capsule, red faces, and you can tell these ones are Hypolimnus octocula by the black face. The third picture in is the chrysalid, golden brown in color and spikes on the back side of it. And then we have the adult butterfly on the far right. The underside is typically golden brown, and the wings, which you've seen, are black with orange scales. So, as I mentioned earlier, they've been found in Guam and in Saipan. On Guam, literature cites about 10 different locations, and that number is increasing the more surveys are done. I think nowadays we're up to about 14 sites. As you can tell, most of them are in the northern end of Guam. These are the 2 host plants. On the left, you have Alatus semicalcareum. On the right, you have Purpus pedunculata. They are both mostly found in pristine limestone forests. The more pristine that the forest is, the more likely you are to find them and to find bigger, healthier patches of them. Okay, so the current issues that Hypolimnus octocula marianensis currently faces. There is a severe lack of critical biological information for the species. We don't know how far they can travel. We don't know how many there are. And before this rearing, we didn't really know how long they survived. Now we do. There's inadequate reference surveys for these species. Before their listing, there were 2 major surveys done, and both of them were not expansive as enough as we'd like them to be to cover the entire populations on Guam. looming habitat loss, I'm sure we all know about that. There's severe deforestation, development, and competition from invasive species. Parasitism rates in naturally occurring populations, a really big one. We found that eggs in the wild can suffer parasitization rates up to 86%. And that's just the eggs. They have multiple stages after that before they become able to reproduce again. And as I mentioned before, there is a lack of population clarity. We don't know how many there are. We don't know the extent of where they can be found. We have a general sense because of Guam and their limestone forests, but we don't know specifically. Okay, so the objectives of this study. We were hoping to gain a clearer insight to the species' basic biology. Importantly, their lifespans, their survival rates. And this was the proof of concept check on this species to see if it was even possible to be reared in captivity. And from that, we can inform future conservation efforts for this species. And we would know what it would take for a successful rearing facility to exist. Okay, so captive rearing. We started out with a small, this one was 6 by 6 by 6 foot cage, which we snapped some shade cloth on. The shade cloth acted as a barrier against predators, as well as preventing them from escaping, as well as an attempt to mimic the limestone habitat, which they are typically found in. And on top of that, we created a 10 by 20 by 20 frame and slapped some more shade cloth on that to act as double protection, as well as increasing the shade for the environment. Procris was used as the host stock, and we found we sourced the Ptilinopus Acoustic from the University of Guam's forest right next to the mainland. So from the captive rearing, we're hoping to gain more insight into their life and see if this method was even possible for establishing populations in the future. Okay, so let's get to the rearing. So this was the rearing facility that I constructed at the University of Guam. This was the 6 by 6 by 6 foot cage where we hosted the adult butterflies and some host plant in case they copulated, which they did. This is a picture of the two 6 by 6 by 6 foot cages. We kept the life stages separate. On the left here, we kept the larvae and chrysalids. On the right was where the adults reproduced. And this was the first generation of base-spot butterflies that we reared in captivity. From these three, we had 1 female and 2 males. And this is the video I took when I found the female ovipositing on purpose. I told you at the beginning of the presentation, typically the eggs are found towards the edge of the leaves, as you can see at the top left up there. But throughout this process, we found that if the environment is healthy enough, they really put them anywhere, really. Although we did find a higher percentage of them were found on the edge of the leaves. And here are some of the adults that we reared. So from this effort, we found 333 eggs. The question is that a lot of eggs? Compared to references of naturally observed populations, the most found at one time was 180. We produced 38 adults from this effort. 23 of them we were able to release back into natural populations. And here's more of them. And thank you. I have a slide missing from this. I apologize. I would like to thank the Department of Agriculture. This funding was provided by Wildlife State Grants. And thank you. Yes, U.S. Fish and Wildlife Service for providing those fundings. And thank you, G, for providing me the space to do this. And my chairs, Dr. Wheeler, Dr. Demilanare, and Dr. Moore. Thank you. If you have any questions, I can hopefully answer them.
So we have a few minutes for questions, and sorry about that, Matthew.
It's okay.
It was downloaded earlier.
I should have not forgotten to thank them.
Hello, great talk. So in vertebrate ecology, when we think about doing releases, often we have to consider things like naivete and doing things like predator training. And so I have no idea how it works with an insect. Do you like soft release or hard release, or is there any kind of training that happens, or is it just kind of you let them grow up and then they float away?
So this was the first time this has ever been done for this species. As far as I know, we did a hard release. We released them at 2 different times and released all of them, as many as we had, as pertaining to the permit that we had at the time. For future causes, if this were to continue, that's definitely something we'd have to consider more, as well as providing enough pre and post surveys to prove that it's actually viable.
That was a great question. I wanted to ask that too. To add on to that, I know that there's been a study demonstrating this is the right phrase, negative conditioning in butterflies from stimuli during the larval phase that they responded to negatively in the adult phase. So what would be your way of moving forward? What's one cool way you can do exactly what she said, condition or train these butterflies to be less friendly?
That is a good question. I'm to be very honest, it's not something I've thought of before. But as far as training butterflies pre-release, it's a good idea. I'm not well enough informed about it. They're very heavily predated upon in the wild, so it's definitely something to consider.
I was nervous. So if you were allowed to have as much funding as you could, and if you were to do this sort of study, what would be the next steps after viewing them?
Okay. So as far as conservation efforts are, the hopeful end result is that downlist them and then eventually delist them, correct? So for this species, we're looking at establishing, it actually says in as part of the recovery plan for the species, they want 14 established resilient populations for downlisting. And then for delisting, they want 20. So if I was allowed an incredibly ridiculous amount of funding, I would probably establish a facility to eventually hit that goal. My goal, I don't know, ballpark, probably 10 to 15 years to be able to prove that they're established and resilient.
Hi there, great talk. I just had a quick question. If you considered releasing them back in Saipan, or if there's any place, if there's any island where maybe the predators are at a lower level where they might be a little more successful?
So yes, I have actually asked that question a whole lot to the people above me to release them back to Saipan. It's not something that's being considered right now. Right now, we're looking at establishing those populations here. From what I understand, and don't quote me on this because that's not a lot. There's a whole lot of hoops to jump through to be able to do the same type of releasing on Saipan. But don't quote me on that.
You mentioned the relative scarcity of the vegetation that they rely on. Have there been any associated outplantings of those plants in relation to these butterfly releases or habitat creation?
So, yeah, yes, as an entity, I'm sure there are a couple of entities as well that have been trying to do that. Crocus and Elastem are incredibly hard to outplant. I don't want to say as we found difficulty, but there is a lot of difficulty. They don't take very well. And as far as they go, they also have a very specific habitat requirement that's not very easy to replicate. It is something that is being tried, but there's a lot of difficulty with it. That's why preserving the habitats that we do have, where they are found, is increasingly important.
Okay, thank you so much, Matthew. So our next speaker is Dr. Kurt Fiedler from the University of Guam, and he will speak on mollusks and plant surveys of the islets of Guam.
How do I advance?
Should just be left or right.
Hi. Hi. I'm not used to using a microphone, so forgive me if I walk away from it on mistake. My name is Kurt Fiedler, and I'm going to talk today about some, I call them the small island surveys that we conducted over the last few years. My co-authors are Dr. Alex Kirk from the Marine Lab and CJ Paulino, who's here today, who now works for TTT. A little bit of background. We have at least 60 small islets around the near shore areas of Guam, and some limited survey efforts have been conducted in the past. There was a really good study on reptiles on a few islets, and there's been a few studies here and there on plants, but virtually nothing on invertebrates, including terrestrial snails, which is something that I work a lot with. And Dr. Kerr and I did some small surveys on Cocos Island, and published 2 papers, one on Cocos Island and one on Alupad Island on the mollusk fauna there. And we decided, hey, let's do it everywhere. So we had a few objectives. One is to see if we could get to every islet available around Guam and do terrestrial mollusk surveys. And because we like plants, try to do some plant surveys as well. And that's where CJ came in and helped us identify the plants that we had difficulty with. We also wanted to get some idea about distribution patterns. And we also, in the back of our minds, were thinking about maybe some suitable candidate sites for future translocation of endangered species like our Mariana's tree snails. And I'll get to that a little bit later. So all those little green pins, including the yellow pin there, these are the places we surveyed, about 50 islets. We started in 2015 on Cocos Island, and then eventually we went to Alupad Island. They occur from 2,000 to 2,500 meters offshore from Guam, elevations up to 26 meters, and the size of these islets varied from 1 square meter to 91 hectares. So quite a variation in size. So as I said, we started with Cocos in 2015. We published another study on Alupad Island, and then we started expanding to some accessible islands in 2017. And every once in a while, when Alex and I had time, we would go to some other places. We got some funding from DAR to do terrestrial snail surveys from Fish and Wildlife, and this allowed us to make a concerted effort in the last year, and we brought CJ on board. So surveys were conducted by 2 to 5 people, and here you can see 2 people, and one of our bigger small islets, let's put it that way. Most of the islets were accessible from shore, either walking, wading, swimming, trying not to drown. A few we accessed by boat. Generally, we would start with the margins of each islet and then work our way inwards where possible. Sometimes vegetation was very thick, it was difficult to get inside. Other times, the substrate was difficult to navigate. Some of the larger islets like Cocos Cabris, yes, that's an island, and Alupat we visited more than once because we thought we needed to cover those areas a little better. And we, of course, we took lots of photos and a few specimens here and there. And we recorded both live and dead snails. So here's some of the general results. And this is a graph showing, a table showing the relative area of some of the larger islets and the number of species of snails and plants. With some exceptions, the larger islands had higher numbers of snails and plant species. And I'll show you another graph that illustrates that with all the data. But one of them, Alupat, I'd say punched higher than its weight class because Alupat had quite a few snails for its size, even though it was very small compared to some of the other islands. Neje is another island that had quite a few snails and a good number of plants, but that is an island we didn't get to spend much time on, and we need to think about some other island characteristics as well. So here's a scatter plot, or sorry, not a scatter plot, a plot showing the relationship between species richness of snails and plants, and you can see it's a positive relationship. Don't ask me for the arc. Let's look at plants first before we start talking about snails. We found about 150 different plant species among the islets. The one that's most common, as you can imagine, is Penthus, and that was on 44 of the islands. The other plants that we saw are typical species you would expect in coastal areas. And we did see some larger trees, Pisonia, but mostly on the larger islets. These two graphs show kind of the same thing, but a little bit different perspective. One shows the relationship of plant species number and islet area, and the second one shows plant species and the vegetation area. I don't show the elevation here. So as you can see, generally a positive relationship once you get past a certain size, and these are log transformed areas. Now it's freezing up. Why is it freezing up? It's not going. I mean, it's that's just no, something else going on here. Okay. We did find a few rare and listed plant species, Heritaria, longipediolata on Cabris and Ani. Of course, our Micronesian cycad, one instance of Eugenia, and Initia bajuga, sorry, I can't pronounce it. And I'm not saying where that is because I don't want people to harvest it. Snails. Generally, as I said, the same pattern, and I'll show you a scatter plot here in a second. We found 37 different terrestrial snail species over the survey. The most species occurred on Cabris, where we had 23 species, and some of the other large islets. You can see the next was Cocos, and after that was Alupat, Nedze, and Aluton. There was one island that didn't have any snails or plants. And the mean number of snails per island, the snail species per island, was about 5. And scatter plot showing the same positive relationship that you would expect for island area increasing, and then with vegetation area similar for elevation. The most commonly recorded snails were this guy here, Tahisha marianarum. We found that on 28 islets, and the old name was Truncatella, and it's a family called the Truncatella because they actually snap off their tips. You can see a younger one that hasn't yet snapped off its tip. And here, these have snapped off the apex of their shell. Amphilochropus submaritima was the next most common. Let's see, I've got a couple others here. Melampus luteus up on the top, and Alopeus gracile, which is not native. The other 3 are native. And then some of the other ones probably you wouldn't recognize in a dark alley, so I won't go over them. Most of these snails are really, really tiny. I want to make that clear. So it's not something that you would see and call me up and say, hey, is that a tree snail, Dr. Fiedler? These are tiny, and a lot of them are in the leaf litter. We did find 3 possible undescribed species or subspecies. One Paulina here on the left, we think, we're not sure, at Cabris, and then a couple Ampelotropus that look similar to this, one at Cabris and one at Aluton. We did find evidence of past tree snail populations, but no live tree snails. We found old worn Partula radiolata, we think at Cabris. These are fragments, old, so it wasn't from a recent population. And Partula gilla we found at Nedze. Both islands are relatively close to Guam. Here's Nedze here, it's called Turtle Rock, off of Big Navy. And then here is Cabris, which is maybe no longer even an island now. I'm not sure how you would define that, but it used to be an island. We didn't find any of the major snail predators, Phytodemus manaquari. And as many of you may know, this is the terrestrial flatworm that has decimated our native snail populations. And we only know maybe what the effect is on tree snails because they're big and pretty and all that. But we have no idea of the impact on these small, tiny species that most people don't even know occur. We did find an introduced predatory snail called Gulella bicolor on Agrigon, but it's unlikely that it would have an impact on larger snails. And when we've seen it in other places, it's been pretty uncommon. We did find brown tree snakes, on Cocos. We were the first ones to find the skin there in 2020. And we caught a live brown tree snake on Anai in 2022 in August. People told me, though, that they've been known from Anai for a long time. And no other signs of brown tree snakes were seen on other islands. So are islets important biorepositories? Well, this survey documented well over 30% of the 110 known terrestrial land snails from the Marianas. And most of these were examples of living. We did find some ones that we did not find live examples of. And a lot of these were specialized for coastal habits. They're on these small islands. Comparisons with species richness on Guam itself will be interesting, and we're in the process of doing that. Many of our native snail species have not been seen alive in decades. So when we find shells of something, we don't assume that we're going to find living ones in most cases. So can islets be used as refuges? Some of the larger islets, I think, have relatively high biodiversity of plant life and other things, and habitat complexity. They don't have manaquari, as far as I know, the flatworms. So maybe we could try to establish partulid snails on one or two of the larger islets. We could combine these with efforts of habitat restoration. Some of these islets, obviously, people have been living on there and brought plants there. Maybe we could remove some of the invasive species. And of course, another thought we had was, Anai's not that big. It's much smaller than Cocos. Maybe you actually could get rid of them on that island first as a proof of concept, as a precursor to scaling up for other islands. What's left to do? Well, we need to survey. We'd like to survey some of these little islands we couldn't get to because of DoD access. And we'd like to go back to Nedze and some of these other large islands, even Anai. We didn't have much time. We're going by boat. We'd like to go back and hit those really well. We'd also like to do reptile and bird surveys. I proposed that to some herpetologists, but we had a pretty ambitious schedule. We hit 50 some islands, in a relatively short amount of time. We could also do snail bioblitzes on Guam. I'd love to do some collaboration with CNMI folks for these surveys. Acknowledgements, my co-authors, CJ Paulino there on the right, holding his phone, showing me his Instagram account, I guess it's number of followers. And then Dr. Kerr catching his lunch on Anai. I'm not sure what he was doing there. Field work, we had a lot of help with field work. Alex's family helped out, Roy and Joni. Carla Dizon, one of my former students, who just got her PhD. Joni, I misspelled her name, Villarreal from Weary, and Heather Jinko, who is now on Snail Team 6. And of course, we had some administrative support from folks at Department of Agriculture, Diane Weiss and Jay Gutierrez, and then folks at RCOG. Our funding came in part from McIntyre Stennis grant I had, and then a state wildlife grant to Alex and I via Department of Agriculture. Thank you.
Do you have any questions?
Thanks for, very interesting. Do you have actual plans or what would it take to, assess the survey?
Money. No, I'm money.
Oh, yeah. No, I'm thinking at, Aurora, DOD, or what steps do we have to take? to get to?
That's a good question. I probably could work something out with NAVFAC.
Maybe some mitigation.
Yeah. Yeah, I don't know. That's a good question. But we just finished this and we're wondering, are there other big targets we should hit? But yeah, that's one we definitely would like to do. And there are a few up north we just couldn't get to because of shore access. Other questions? Yeah.
Thanks for the presentation, awesome. Curious about the lack of the flatlands on the islands and what it says that just that they're not going to swim across the water, and are there bugs or risks in terms of people accessing those islands where flatlands might get earlier?
I don't know how flatworms get moved except maybe in soil with plants. So I would have a hard time believing they could get there easily. And it's saltwater. These animals aren't going to do well in that kind of environment. They're not going to swim like the snakes are probably, for example. So that's a good question, but and we don't know for sure that they're not there. We just didn't observe it. But I can tell you, we've been there in a lot of wet days and a lot of places, lifting up leaf litter, digging through leaf litter. So we think we would have seen them if they were present. Yeah.
Great talk, Kurt.
Thank you.
So they seem like largely saline environments. So do you think these snails you kind of got at this, you think they're adapted, or they can somehow regulate salt better than snails on the mainland?
Some species probably so, like the Truncatellus, they seem to be, you find them on coastlines and things like that. But not all of them. Some of the ones we found further inland, may be a little more tolerant or a little less tolerant, but they're in pretty decent habitat. So I'm not sure if I answered your question.
It's kind of a lame question. I'm thinking of the Moya takatsukasae that lives on these islands. It's the same tolerant skiing. And so atypical in that regard is because of these types of habitats.
Well, that gets to the question is, how do snails move between islands at all? A good question. That one of the dominant theories that is out there is that they ones that are associated with plants took a ride on bird legs, but I don't think that could happen for all of these species.
So as he's walking up here, this is my former colleague here at Guam Community College. I'm glad to see he's not shy about coming back. This is Dr. Frank Camacho, who has now moved over to the University of Guam, and he will speak on the effect of a dam on streamfish assemblages in the Gaius River Peninsula.
Thank you, Jody. Can you guys? And I know this wasn't what you were expecting to see at a terrestrial conference, right? So, and I was kind of doubtful whether or not the abstract would be accepted, but through, I won't lie, blatant pandering, I threw in the words invasive and bamboo. So I put a few people in. Okay, so I'm going to talk today a little bit about, I am going to talk about bamboo, but I'm also going to talk about kind of a project that we've been doing for a while that was started in 2016 and focusing on the effect of an impoundment on the movements and distribution of sort of our native insular stream fauna. And recognize my co-authors here, Dan Lindstrom, Nicholas Camacho, and Kate Moose, from the University of Guam. Cool. And so, we tend to think of we tend to think of rivers, I guess from people that do terrestrial conservation, most of the time we're thinking about rivers as basically conduits for sediment that are basically transporting, silt and sediment out onto the reef bed, right? And as a product of upland erosion. And what I would ask us to consider is that these systems also host a diverse array of native stream fauna, right? With rather unique histories. I'd also like to call to attention to the fact that, These riparian areas that bracket these streams are actually oftentimes have more species diversity and species richness for plants than the adjacent savanna areas. So they can be viewed almost as kind of refugia for some of these plant species. The usual suspects we have that we deal with in our work here in Micronesia are the dominant sort of group are going to be the gobies. All right. For most people, you know, your frame of reference that you might recognize gobies might be like mud skippers, right? And so what unifies gobies are the presence of these fused pelvic fins. And these pelvic fins allow gobies to ascend steep inclines, high grades, such as waterfalls and cascades. All right. Other common conspicuous organisms include freshwater prawns, macrobrachian larvae, probably the largest decapod crustacean that commonly occurs in these streams. And then we have some predatory fish that are aggressive. These include freshwater eels, Såli and Gila marmorata on the bottom, but also flagtail perch, Kulia rupestris. And Kulia rupestris plays an important part of the story here in a second. Now, as I said earlier, kind of the life history of the stream fauna in Micronesia, not just the Marianas, not just Guam, but basically throughout most tropical oceanic islands that are able to support surface waters. The dominant life history amongst those organisms are going to be this amphidromous sort of life cycle in which adults mature and spawn and live out their life in freshwater, but their eggs, when they hatch, the larvae are transported out to the open ocean, where they complete sort of this pelagic phase of larval development. And the larvae are distributed and undergo metamorphosis out of the open ocean, and eventually currents sort of distribute them around to other islands, and they're able to, these post-metamorphic juveniles are able to recruit to these river mouths and migrate upstream. Right. So that's an important part of the story, because what that means is whenever you go out to a river, whatever island you're on, whether it be Palau, Guam, Pohnpei, we've done work in all those areas. Just about every organism you see in the stream, whether it's at the river mouth or almost at the very top of these headwaters, essentially came from the ocean, right? So that's, in some cases, it can be a quite spectacular migration. Now, as I said, we've been doing a lot of work in Micronesia since the early 90s, doing freshwater stream surveys, starting in Palau, then Yamaskan, and also Yap, and then continuing on to places like Pohnpei, Kosrae, Rota, and of course here on Guam. And kind of a recurring theme that we've seen in these sort of natural systems are that there's sort of unique sort of disjunct distributions in the fish, in that in the lower reaches of streams below usually the first major waterfall, we typically find an abundance of predators, particularly Kulia, that flagtail perch that I was showing you on the upper right. Okay. Sort of dominates those lower reaches of the stream, and then to go above that first major waterfall, and that's when you start seeing sort of more of the gobioid fauna. All right, and it turns out that this is largely due to the fact that Kulia lack these adaptations to scale these waterfalls, scale these cascades, and so that first major waterfall and natural barrier basically is an important sort of force in sort of structuring at the whole river level kind of what the streamfish communities, the streamfish assemblages are like. Okay. So that said, we know very little about the role of man-made impoundments on structuring streamfish communities. Right? So that's kind of where we're going at with this. There was some work by Georgette Conception looking at the effect of the spillway there at Fena, and a little bit of work out there in Hawaii, but for the most part, we really don't know whether or not these impoundments present sort of a barrier to the upstream migration for these native stream assemblages, okay, or the streamfish. So we're going to talk today about the Gaius River, which has a small man-made dam about 2 kilometers upstream from the river mouth. It's a small stream. It's only a third-order stream for those that have taken a limnology course to do stream work. The mean slope is about 33%, but really that overlooks the fact that most of it is sort of relatively shallow grade, and then just really starts to get rather steep, back towards the headwaters and the base of those hills. Importantly, this river was a significant source of potable drinking water for the surrounding community of Molesso. I've tried to figure out when the dam basically was no longer used anymore. I think people in the community told me sometime around the 90s, but for the most part, it was still diverting water for human consumption, at least well into the 20th century, right? So we have the dam. This is kind of what the dam looks like back in '74 when Harry County did some surveys. Some of you might remember him. This is the dam on the left when it was still functional. You can see there's ponded water behind the face of the dam. And on the right are sort of more recent photos. The dam is in the back. A lot has changed out there in this Gaius River Valley. And so there's been a lot of slumping, a lot of erosion, a lot of faulting. So kind of much different today than it was 30, 40 years ago. In fact, more so that impounded area, that pond behind the face of the dam has been silted in. So we're no longer talking about a dam that's still functional and impounding water. Rather, we're talking about really a dam that for all intents and purposes is really like a waterfall, okay, because of that silted in backside. All right, so real briefly, our objectives really were just sort of basic, kind of what we've done in other places, just trying to figure out what are the densities of these native streamfish, and also characterizing the physical chemical characteristics of the river, and then determining whether or not there's an effect of the impoundment on streamfish or something. And so, you know, the way you've usually approached this are just basically visual surveys, mask and snorkel. You might recognize for those the known Brent Tibbetts right now, floating log out there in the stream. And what he's doing is basically counting fish and inverts within basically a frame out there, kind of a non hypothetical frame, because we can't really use a quadrant because it'll scare the fish away. So instead, we basically use reference points and then measure them after the fact. But regardless, we're able to get densities of streamfish. We also collect a number of different other variables, information such as pH, dissolved oxygen, canopy closure, and things like that. So real quickly to the results. On these bubble plots, for lack of wanting to show p-values and stuff like that, almost all of these relationships are different. These are comparing the densities above versus below the dam, which is symbolized by that yellow diamond sort of on these graphs. This is lat and lon on the x and y axes. And what you find is that, you know, there are definite differences in the distribution above versus below the dam for a number of these different species, okay? The Maculia, which is that predatory perch that I was talking about earlier in the bottom right panel, is only found in the lower reach along with a few other predatory species. In contrast, a lot of the smaller gobies are found almost exclusively in the upper reach, with only sort of a few incidental occurrences below that dam, right? The obvious question is, well, this is great that you're finding these patterns, but does this appear as if these are different from what you might expect from a natural system? Does the dam have an effect on those distributions, even though we're seeing distributional differences? Does that necessarily mean that there's nothing going on with the dam? And in fact, when we compare this to the previous work, so this is a work by Jim Parham. This is his thesis that he from 1995. He surveyed the Aspenus River and he found very similar patterns. So it looks like the distribution of these streamfish are very similar in this one impounded river compared to natural to other streams here in the region. Okay, so same sort of distributional patterns between them. More of the same Stiphodon, Cystiopterus, all these gobies tend to have very similar distributions. All right, so real briefly, I'll just say that, you know, just thank you, Haldre, for going over NMPS for everybody. But basically, this is real quickly, the one on the hull on the left represents sort of lower reach sites. The hull on the right represents upper reach sites. There's a significant difference when you test it with Andersen. When you map out some of the environmental variables that correlate with those axes using NDFIT, you find that a lot of proxies for basically elevations such as conductivity in the lower reach, et cetera, and things like that, basically all are significantly correlated. So just real briefly, I only have a couple minutes. That said, when we were doing these surveys, we started to notice that, you know, the riparian communities, well, it's a beautiful river valley, but there seemed to be sort of a lot of non-native species comprising sort of riparian gallery. So just a few images from those areas. And so we did line transects. And real briefly, you know, what we find is that starting from the lower mouth of the stream up towards mid reaches, that about half of the half of the cover is comprised of non-native species. And basically the species richness of native species are quite low. It goes all the way up. Species richness of native species increases, but generally speaking, what comprises the bulk of the riparian cover is actually bamboo. And this is a real problem, and I'll just, last couple of slides. This is a real problem because in these areas, clumps of bamboo that sort of fall off into the river modify the river channel. And so water has nowhere to, in addition to basically forming a natural dam, we also encourage the water to go around these big clumps, modifying the width of these stream channels. We also, it seems to be associated with kind of slumping in some of these areas. This was a major fire that occurred about a year ago. And last but not least, we really don't know what's going on with leaf litter and how all that litter input, like you see in that bottom right photo, how that's sort of influencing the the in-stream dynamics like primary productivity and things like that. So in summary, we found differences in density of stream fish above the dam and versus below. It seems to be structured by Kulia. The dam's not a barrier to native gobies and prawns. It would be nice to kind of expand this out to other areas that already have impoundments to try and see if this pattern holds. But there's still a lot more questions. And in particular, I think there's a lot of opportunities to investigate how something like bamboo and other non-native species kind of might be affecting the in-stream dynamics of what's going on in these stream fish communities and assemblages. Okay. That's many of the partners here. Don't have time to go over every one of them, but some really supportive folks from the mayor's office, the community molested for allowing us to work there. Of course, funding our funding partners, and then people that have helped us out in the field. And I'll take any questions. Thank you.
Right, first of all, it's totally great. Looking aside the marine side and the western side better than the culverts, right? Which are so near and dear data from the Nightingale River is very near and dear to me. Earlier career back on Guam. So what I wanted to, you covered it, some of the vegetation. I need a better photo though of that slide.
I know, I'm sorry.
So we can share that, that'd be great. But more talks and more information on desired riparian habitat. We do so much work upland, inland, but what we haven't put enough resources in, even though the Bureau of Statistics Plan Coastal Management Program, many of you, well, some of you might know Patrick here, we've been doing work in these rivers for a while, and so what we need to see is the preferred habitat type for the riparian. I remember walking through there and watching the walls just sloughing off of the riverbank. So it'd be great to see more of the data and where you would like to go with more research, because forestry definitely wants to put that.
Thanks. Yeah, it's a good question. Thank you, Christine. And regarding the preferred riparian habitat, it does. We didn't survey the headwaters, but it looks like bamboo is not as abundant in the headwaters. Whether it's because bamboo just hasn't had enough time to spread up into the upper part of that valley, or whether or not there's issues with water and soil water and what is that slope angle and things like that, which I'm a freshwater person, right? So totally out of my league there. But it'd be interesting to kind of see what else is occurring up there in those headwaters. Maybe that might be informative for the restoration. Yeah, sure.
Frank, when you showed the slide of the occupants of Kulia with different gobies, there were 2 goby species that were down in Kulia, right?
Yeah, there were some that seem to be doing just okay. Some of them are ambush predators that are in this sediment. So there's a lot that we haven't. I didn't show. I didn't want to push my luck with the terrestrial conference, but yeah, there are some that prefer soft sediment, and they're in the sediment. That's kind of how they're able to escape predation, I think. A good question.
Well, real quick, Frank's aware of this, but those that are interested in this topic, there's annual surveys of the Andersen Stream and the National Park Service. That's collecting all the same parameters pretty much.
Yep. we've been fortunate to participate on some.
Right.
Frank, great talk. So you noted that the flagtail distribution was kind of the same in a extreme without a dam. What do you think may be driving that distribution?
Yeah, I think it's just the dam itself is a barrier, so they just can't descend over that dam, just in the same way they can't descend over large waterfalls. They can get over small cascades, large waterfalls for food.
Okay, so you think it's just kind of mimicking a natural sort
Yeah.
of cliff line feature, right? Yeah.
Okay, thank you.
All right, thank you, Frank.
Right, up to date, everyone. You might notice I'm not Robert Fisher. He was originally supposed to give this talk, and I got to step in for him. So a big shout out to him and everyone else as a co-author here. I might have done the least work of all these people, so definitely fitting that I get the presentation. But it was a great time. I'm really happy to be included in this work. It was a lot of fun, a lot of good field work in Rhoda. So it's just some preliminary results of island-wide surveys that DFW and USGS were working on. If you saw Dacia's poster, a lot of the same sites and overlap there. And Robert wanted to be here, but he hurt his knee and couldn't travel, and so he definitely missing this for sure. All right, so Rhoda has a pretty good diversity of native and invasive reptiles. There's also a toad. It is notably missing Moist Slevini and Lithinia, which have been found in other places. So here's some pictures of our team out there doing these surveys. We had a big group of folks, went for maybe 3+ weeks of solid surveying, day and night. A lot of good work went in. It was a big effort, I think, and everyone here was actively involved and did a lot of great work. Just shout out to Becker, Robert, Josh, Thomas, Levi, Logan, Scott, Charles, Dacia, and Bronson for all their help there in the field. So what kind of reptiles do we have in the Marianas? It's definitely not an all-inclusive list, but it's just kind of the main things that we were thinking about. So there's lizards, mainly made up of geckos and skinks, most of the diversity there. There's a monitor, maybe two, and an old. There's a couple snakes in the Marianas, notably blind snake we found one in Rhoda, but we did not find any brown tree snakes, and there are not any brown tree snakes that I'm aware of in the scene of mine. Those are the things that we kind of expected we might find or were sort of looking out for. So how do reptiles get to these isolated islands? There's a couple of ways naturally that the reptiles can get to islands, either through oceanic transport on a vegetative mat, through eggs or juveniles, or even adults possibly. And then through something that we don't have here, but islands that are become attached to other large land masses at some point in the past, and then things can migrate over and you lose those bridges. That's not really an issue for us. We are so far away from everything that we don't get a lot of influx that way. So most of our stuff came through oceanic dispersal or through artificial means through humans. That's been well documented. So a couple of the questions that we were kind of looking to answer through this and some other general Pacific-wide research is just kind of what species of reptiles are present on islands, what are native, what's introduced, what may be a new introduction that hadn't been seen before. Just kind of getting some baseline for distributions and abundance, kind of looking forward to climate change or invasive impacts from newly introduced things. Also just sort of determining some trends for elevational transects and recovery of the forest over time. Also sort of planning for some biosecurity work. So this Rhoda survey was sort of modeled after a similar survey that took place in Saipan before 2020. And so we set it up kind of the same way. and tried to compare some of the findings just to see how those two islands compared. And I don't know if you can see this table, but you kind of note here that a lot of the main things that we found a lot of inside, or that they found a lot of in Saipan were introduced. Parlia was one of the top ones, and Knowles, some Lamprobeltis had some decent numbers. So that's just something to note for later. So here's kind of a map of the island of Roto with all of the survey sites from all of the island-wide surveys. You can tell it pretty much encompassed the whole island, all over the place, every habitat. And here's sort of the parsed down version of what we did for the reptiles surveys. We took point from most of those transects throughout those habitats. I will note that we added a couple of wedding cake sites down there, WC1 and 2, and a DFW site behind the office there that I don't think were initially planned for. So I went through all the habitat types from like right next to shore all the way up to the native limestone forest. And we went pretty much everywhere. We even went to the dump. It was actually a pretty good site for lizards. So we left no stone or refrigerator unturned. So our survey methods, we had daytime and nighttime surveys. For the daytime, we did sticky trap transects at 10 sites each station, I'd say. Each station had a ground, a log, and a tree. And then we also did visual surveys over a set time in that transect. For nighttime surveys, we just did the visual surveys, we not do sticky traps at night. And so once we set our sticky traps and came back after a set amount of time, we pulled off all the lizards and we took measurements and weighed them and took some samples, some biological samples. We also kind of noted anything else that might be on our sticky traps. We got a lot of yellow crazy ants in certain spots, and that's something that we counted and took note of, that we can kind of look at that data later to see how that might relate to some of the distributions and abundances of those lizards on those sticky traps. So for the visual surveys, we just walked the transect over a set amount of time. We noted all the reptiles that we saw. We also noted things like coconut crabs and some rats and stuff as well. So here's some of our kind of preliminary results. If you look, you will see that we got a lot of lizards, mostly. And so the skinks found during the day, geckos at night, but we found a lot of Parlia. We also found a lot of Cerulocata. Cerulocata is native, so that's a positive thing. And for the geckos, it was a little more evenly distributed, but still kind of keep in on a few specifics. So when you kind of look at it in terms of what's native and introduced, we have sort of it's more skewed towards native fauna. All right, there are some invasives. Parlia was one of the heavy hitters in the Saipan survey, also found it in Rota, but we also found more Cerulocata in Rota than Parlia, so that's nice to note. So we found almost 1,500 lizards, and through observations and captures, and about 2/3 of those were native, which I think was notable. So it's also an opportunity to look at some of the state wildlife action plan species and kind of know what we found there. Two of the specific ones were Acrocephalus and Karisu, a skink and a gecko. And we did find both of those targets, so that was really good. The Karisu we found at several sites. I think folks were pleasantly surprised that we found a few more of those than we thought we might. Acrocephalus, we still found some, not that many though. So I mentioned those wedding cake sites earlier, and I just wanted to touch on that. We did not find any Slavinae at those sites. We kind of set them up for that. We didn't find any there, but we did note that the habitat there was really nice, and just kind of note that. Might be a place to look in the future. So another really notable finding here is that at Penetang Park, which if you've been to Rota, it's an abandoned water park right north of town there. And it's kind of an interesting place. I definitely recommend walking around. But we found Amoya cyanura there, which had not been documented from Rota before. And as far as I know, it's only found on Islandano in Guam and nowhere else in the Marianas. So that's really interesting. And it's very similar to Cerulacata, so it could have gone unnoticed for a while in previous surveys. I think you can tell them apart with the blade-like lamellae that cyanura has versus the kind of more rounded lamellae on their toes there. They also have a continuous dorsal stripe in the cyanura. We did also note a new invasive snail from Taiwan, natively. It's been found in Guam for quite a while, since the 80s, but no one had noted it from Rota or anywhere else in the Sinamai. I am not a snail person, but I think maybe I've seen this in Saipan as well. That's just very anecdotal though. So yeah, general outcomes of this. In some positive news, there were some possible invasives that are found in Guam and Sinamai that we were kind of hoping we didn't see, and we didn't see them. We didn't see any green tree skinks, we didn't see greenhouse frogs, and we did not see any brown tree snakes. Hooray. So some things that we were looking for that we did not find were Slevini and Lavinia, and that's a downer a little bit. We did find also 2 new invasive species, the snail from Taiwan and the cyanura. We did find a new population of tree snails, which that was good. That was a great find. And then just overall, compared to Saipan, Rota has a more native dominated herpetofauna than Saipan. So that's what we found there. And with that, I'll take any questions. And if I don't know the answer, I will try and direct it to someone who does.
Questions for Grace?
Did you see any Guam rails?
I did not actually. I think that other folks did, but I did not.
For the native species that you were hoping to see but didn't, you have any instances for why they're present?
For Slevini, I don't think anyone had seen them in a long time. There hadn't been like any real surveys that I know about that had been like hardcore looking for them. And still, I would say that this survey, while we added sites to sort of include some habitat we thought might be good for them, we didn't go scour that habitat and say we're going to find Slevini today if it's here, right? Like, and that's sort of thing that I think with all of this, it's like the snapshot of what's going on with the island, and like even bad things, right? Like brown tree snake. Like I wouldn't say that our surveys were geared towards finding brown tree snakes. They were geared towards just seeing what's there during our survey period. If like we went back with the specific goal of finding specific species, we might be able to do that, either good or bad.
Hello.
Hi.
I thought the cyanura was native. Why did you guys look at it invasive?
I do not know the answer to that question. That's what Robert told me.
Any other questions?
I was wondering if you could elaborate on the green anole a little bit. I know it's native to the southeastern United States.
Yes.
I was wondering, when did it get to the Marianas, and what kind of impact do you think it's having on native lizard species?
I do not know when it got to the Marianas. I don't know. And like native impact, it in my mind it fills a different niche than some of the native lizards. So I think it could be not having a huge impact, but I don't know. I might ask Malia if she's around, if she could answer either of those questions. I don't see her in there. What issues?
It was used in the
They were really pretty.
Oh, that's me in 1993. I thought they were really cool in 1993. Yeah.
Yeah, I don't get into that, the interaction between those species, but, they have pretty different people on the community, rest of the echo, so the probability that they're directly transmitting is, I think, low, but it might be interacting, right?
Yeah.
So they're smaller global.
Nice. Malia, before you give up the microphone, do you know the Sayonara question about whether it's the situs introduced or not?
I think that was just like a big topic of debate.
Okay.
Everybody goes back and forth as to whether or not it's native or not native, and feeling a position that
So I also believe that the Zihaira oceanica is definitely native, right? But what used to be called Mutulata, I thought was native as well, but Rhodolus isn't introduced. I do not know enough to have a strong opinion about that.
So the Sayonara thing is just Robert has really loud for it.
Yes.
So that's his opinion, but there's only one record on Guam.
Yeah.
And then they're known from East Montana. And then this new recent record.
Yeah.
Moved all over the Pacific.
Yeah.
So the question is, why wouldn't they be all over Mariana?
Yes, that makes sense. And I mean, I think the thing that I think about is something I touched on is that they look really similar to just even a generic biologist to a Sierra licata, right? It's a small skink with maybe a like bluish greenish tail sometimes when it's a juvenile, and it like has some stripes. Like, I mean, honestly, before I did this survey, I could not have told you the difference between the two.
Sure, unidentified.
So I think that's from the visual surveys. I think it's just that's yeah, it was just, oh, that was a skink and it went under that leaf and I don't see it anymore. And I saw its leg, you know, like I don't know.
Okay, so what we're going to do now, if you can bear with us, we'd like to get a group picture of everybody. You know, it'll also be a good way to show your boss that you're actually here. So I think we can move to the front, and then after the picture, then we'll take about it. Oh, yes, I'm so sorry. All right, so then, so I think we're going to try to take the picture up at the front, and then after the picture, we'll take a 10-minute break, and then we'll come back for the rest of the session.
Sure. Do you want to the table?
Yeah.
Great.
Thanks, Joni. Thanks, everybody, for hanging in there at the last minute. So I'm Jonathan or John. I work for the USGS. My duty station is in San Diego, California, but I'm one of the lucky few people at my duty station who have the privilege of working on Pacific Islands, and some of that work here is in the Marianas. And the work I'm going to talk to you about today is part of development of an ungulate control program in the Northern Islands. But before I begin, I just wanted to acknowledge my co-authors, Adam Backlund, Scott Getz, and Levi Gray, who are also USGS people, and then Bronson Curry, Emily Kohler, and Jose Atzaleg, who are CNMI DFW. And working on the Northern Islands is a huge privilege, and it's a huge endeavor, and it's an impossible thing to even think about. Many steps, many players involved, and so these are some of the people I just mentioned, but at the top is Mario Sanchez. So this is the Sanchez family that you actually mentioned that owns the land on Alamagan, and so Mario actually spent his youth on the island, and then Ken Sanchez is his nephew, and then Dane and Aru were members of the Super Emerald, which is the boat that we took up there that I'll talk a little bit about later on. But yeah, they were, I think Ellie said it the best. I mean, these guys were incredible. They taught us so many things. They guided us through the forest. They shared their local knowledge. They hunted for us. They fished for us. They were insisted on expanding my Chamorro linguistics in ways that I can't share with you right now, but insisted that I learn these phrases and terms. And so I'll begin with that. I'll do more acknowledgements, but as I said, this project involves the development of an ungulate eradication program. And so talk about a human dimensions issue that we were talking about earlier. And I have to say, I'm completely humbled by the talks that were, preceding this one and the issues and the discussions that we're having. But the ungulate issue is definitely a human dimensions issue that's really interesting. I'm not involved with this part of it, but I'm just sort of leading up to what my role in it was. But there are 8 islands where there are ungulates, and I've just picked the pig as the exemplar icon, except for Aguihan. I put the goat because that's all. Someone decided to name it Goat Island as well. I don't know who would name an island after the thing that eats it, but that's Goat Island as well. But 2 of these islands, there have been successful eradications. And I'll talk about the one on Sarigan. It was done in 1998. And within 5 years after that eradication, the native forests and the native fauna bounced back really dramatically. There was one done on Anatahan in 2003. They started in April 2003. Then the volcano blew its top in, I think, May of 2003. And Stephen actually remembers when that happened. Apparently it was gnarly. And it sort of went intermittently and kind of augmented the eradication. But the feral ungulate problem, so Adrian gave a great talk about this on Tuesday, went over the problems really well. And she talked about how you have this understory elimination, you've got reduced leaf litter depth, you've got stunted tree recruitment. So she gave some really good details about all of those things. And then I'll just touch on some of the things that affect the critters on the ground, because I'm going to be talking about the lizards. And so when you start getting rid of forest canopy and understory, you get the solar radiation going to the ground, and that dries everything out. And that's really bad for small skinks when you're this big, because when it's really hot and it's dry, you die. And it also accelerates erosion and alters nutrient cycling. And then she explained that the native forest is often replaced by grasslands. There's predation on the native fauna, so the pigs will eat. Apparently, they will eat the skinks, I've heard, but they eat the coconut crabs and probably any other thing they can get in their mouth. And then these dung fly infestations, and they're no joke. And then there are likely these wholesale shifts in the invertebrate communities as a result of the way these animals modify the environment. And so on Sarigan, like I said, there was a successful eradication done on Sarigan. And literally within a year, they saw this major recovery of the forest. And within 5 years, they saw lots of understory recover, forest recovery. Grassland was being replaced by native forest again. And the animals started rebounding as well. So they saw increases in coconut crabs, the Micronesian megapode. And then 2 of the most dramatic increases in abundance were the Slevin skink, the Gualic halom tano, and it probably just butchered that to death, but that's Amoya slevini. And then the Akaliha, which is just the Chamorro term for snail, but that's partially gibbon. And so those 2 species bounce back really dramatically, and they think it's because of this increased moisture that came back to the forest floor. And so because there was this positive response to ungulate removal, they're now thinking about doing similar things on some of the other northern islands. And so the Slevin skink is sort of the focus because it's endangered. It used to be on 11 of the 15 islands in the Marianas, and it's believed to be extirpated from 5 of them, and it's believed to be in a state of decline. And it was put on the endangered species list. I think it was 2015, the federal endangered species list, as well as the ICUN endangered species list. So it's gained some notoriety for that. And then also it was rediscovered on Isla Dano following a rat eradication that was done in, I think it was 2010. I'm pretty sure it was 2010. The skink had not been seen on Dano since the early 90s, so over 20 years. So they thought it was extirpated there, but the rats went away, and then suddenly herpers were going out there and they start noticing that, hey, there's the Slevin skink that's there. So clearly this skink responds really well when you get rid of these non-native species. So the objectives of the work that I was involved in, it was a complete privilege to even be able to go up to Alamaga in the northern the Northern Mariana Islands. It was really an honor to be able to go there. And so we were just there to perform a status assessment of the reptiles to aid in the development of this ungulate control program. So this would be sort of the pre-snapshot of what the reptile fauna looks like before they get rid of, before they start controlling the ungulates. So baseline data, as they call it. And so we did trapping transects and visual surveys to document presence, abundances, and distribution, similar to what Trey was talking about or what they did. We took a little bit of we had a different approach. We used a smaller number of transects, but we did replicates because we wanted to learn something about detectability. And so we did these 11 transects on opposite sides of the islands, and these were previously used for lizard and bird surveys. Jill Liskey-Clark, I should have mentioned it. This was a SWIG grant that she actually wrote to do this work, and so she participated in some of these previous surveys as well. So these 7 transects consisted of 7 georeference trapping stations per transect for every 10 meters apart. And then the transects span a range of elevation and different habitat types. We also did ant and rat surveys. We heard a little bit about this. Ants and rats, non-native ants and rats, non-native rats have really bad effects on reptiles as well. So we wanted to get an idea. There were some of these more common invasive ant species there and what rats are there, if any. And then the CNMI DFW people did these intense vegetation surveys. Jose was not happy about doing these, but they did these surveys for all the lizard transects and beyond, and then USDA is doing camera trapping surveys. So they set these camera traps up, and I think they're going back in June of this year to recover the cameras and then get some idea of where the ungulates are and what the densities might be and whether it's even feasible to attempt an eradication. So this island is awesome. It's your classic stratovolcano. So the island is literally the tip of it, and it goes from sea level to the top of the caldera is like, I think it's about 745 meters, so a little over 2,000 feet. But then it goes down to the ocean floor, 4,000 meters, like over 13,000 feet, all the way down to the ocean floor. And it's basalt adenosite, so it's different than the limestone, mostly the limestone type islands that are down here in the south. And this is just an aerial image of it. This is a veg layer. I don't know much about where this layer came from other than I was able to get it and load it in ArcGIS. And so that's the veg layer, and when it's that sort of beige tan, you can see there's mostly swordgrass. So most of the island is swordgrass. Have you ever dealt with swordgrass? It sucks to walk through anyways, but there's swordgrass and there's a lot of old coconut plantation, coconut forest, and then mixed native forest. Oh, sorry. And those dots that you see there are the locations of where all the transects were. And then I had sort of close-ups of it, but it wasn't really gonna tell you very much. But those are the locations of the transect. This one, so we camped on the north side and we spent most of our time up there, and we ended up just taking ponga rides down to the south. And initially we were gonna go and move camp, but it was just gonna take too much time. And the south side here it is like the pig super highway down there. So John, if you need flies, it's like pig restrooms on the pig super highway too. So that's a good place to get flies. It's unbearable. This is what the sticky boards look like. So each trapping station, we had 3 boards. So one on the ground, one at about knee height as a perch, and then one sort of like waist height. And the idea is that you catch lizards with different behavioral ecologies. And then of course they get stuck on the boards, but then you can kind of massage them with canola oil. The lizard comes off, they're alive. 2 minutes left. Mine says 10:22. Okay, general results. There's no non-native species of reptiles or amphibians on Alamagan, which is really impressive. We didn't have any new native species detections. There's only one previously recorded species that went undetected, and that's this blind snake that Trey also talked about. The Mariana skink was pretty common in certain areas. So 7 of the 11 trapping transects, and then 8 if you include a transect where you saw visuals only. They were detected in a variety of habitat types, including areas that had no canopy, so non-forested areas right out on the lava with the pemphis. And then just to give you an idea about density, so these are both abundance charts on the top, so that's the number of captures represented as proportions. And so I'm gonna have to skip through most of this, but the trap captures and the pre-trap visual surveys to show that Amoyas aureliacata, the Pacific blue-tailed skink, is much more common than Slevini possiliplurus, but Slevini is still the second most abundant skink. If you look at capture rates, again, it's sort of the same information. This is by transect. You can see that aureliacata is the most frequently captured skink. We caught 4 different species of geckos. These are all common geckos. Most of those were the Gahyra oceanica and Gahyra insulensis. Those are the most common. There's also Nactis pelagicus and Leptodactylus lugubris. We didn't catch the Amoya atricasata, which is this littoral skink. So in the southern islands, it's mostly found in that lower left slide, so low gradient slope with limestone, karst, and pemphis. We don't have any of that in the northern islands, and so the lizard isn't there. It was documented there, but I think it was a misidentification. Ants and rats, there's no detection of yellow crazy ants or big-headed ants, occasional detections of rats, but it was never in the forest. It was always around camp. We never caught them in traps. And it could be because of the helitide, the monitor lizard, and also there are some cats that live on the island. Take-home messages is Slevini is patchily distributed, are relatively abundant in certain areas in Alamagan, and the capture rates are roughly equivalent to what they were pre-eradication on Surigan. They're most common in deep ravines with accumulated litter, but they use a variety of habitat types. And then last, because of this limited competition with other skinks, there's only 3 species there. If it's combined with ungulate control, it could lead to a thriving population and safeguard against the extirpation of the species. And so just to thank you, these are my funding sources. Thank you to the crew of the Super Emerald for their hospitality. And they just took care of us. The Super Emerald is not the Love Boat. It's not the Titanic either. We got back. And then you guys for putting this on. This is awesome. It gets better every year. I really appreciate it. And then I'll leave you with this photo of Mario because this photo speaks to me in a lot of different ways. And I feel like this is like the way it should be. So thanks. If I have time for questions, I went a little over. Sorry.
So we'll just have about a minute and a half for questions, so we're kind of behind.
Did you find skeletons at different height levels or just on the ground?
We found them on the ground and on perches, so they weren't just exclusively on the ground. We did occasionally see them climbing in the trees, but it was very sporadic. I mean, they're mostly a ground thing, but we did catch quite a few on perches.
So how does Mario and his family feel about ungulate eradication for conservation?
Yeah, so we got, had long discussions with him about it, and he understands the ecological issues very well. But as you're insinuating, it's a commodity and a resource for the Chamorro people, and same for him. And so, as far as I understand, and Jill might correct me on this, the idea is that they get rid of the pigs and the goats, but they're allowed to keep a small herd of cattle that they occasionally cull and really manage. And so it's really just the non-management of these things that it's really the problem. So there's a seemingly a good working compromise to me, but it was, we can talk more. I had lots of conversations about it. The dude's a character. He's a really amazing person.
Sorry, wasn't there an effort to introduce deer to some of the northern Marianas Islands? I know
you're going to get me in trouble. I mean, yeah, there's an incident not so long ago where there were deer put on plug-in, but I haven't heard of that being discussed for Alamagan at all. That's not really a question for me to answer that.
Okay, so let's have a hand for our John.
Well, great. I'm glad because it's, can everybody hear, everybody can hear me okay? So it's 10 o'clock here in Colorado. I unfortunately could not make it there as I intended to. We're having a snowstorm right now. So it's not the most pleasant place in the world, but I'm really excited to be talking about my favorite lizard in the world, Amoya slaveni. And with that, we'll get started. So late last year, we were finally able to trial a method for surveying and monitoring Emoyas slaveni on Dano Island. I'll tell you the main point of this talk right up front is it's not so much what we found, it's that we trialed a method that looks like it's going to work really well going forward. Oops. There we go. Okay, so why study slaveni? It occurs only in the Marianas Islands and relatively little is known about it. This is its historical range. It's now federally endangered. Federally endangered because it's now absent from the larger southern islands. As Jonathan pointed out, it was thought to be extinct on Dano by the early 2000s, but was detected again 3 years later after USDA removed all the rats from the island. So what is occupancy modeling? In a nutshell, just a real simple explanation is it's a method for accounting for imperfect detection of a species at a survey site and determining the probability of true presence or absence. It requires multiple sites and multiple site visits. Why use occupancy modeling? Data collection is less intensive than the methods typically used for population estimation. Occupancy modeling can be used to determine distribution of a species. It's also very useful for identifying environmental variables that are important to the species persistence. And probably most importantly, and for the purpose of this talk, it could be used to monitor changes in occupancy over time. So this study was conducted late last year in response to the presence of the snake, which, yes, showed up quite a few years before that. But that was the discovery of brown tree snakes on Dano was really a wake-up call to action, but it really wasn't feasible for us to do any of the work there until the transects were cut. The map there shows the transects that USDA Wildlife Services had cut there. These are all 20 meters apart. Thank you very much for doing these. These are awesome transects that make it really easy to study things on the island now. On those transects, we imposed a 20 by 20 meter grid cell layer on these transects, and we use these grid cells as our sampling units. We had a pretty brief period of time there. None of us were funded. So we didn't have enough time or resources to survey the entire island. So we focused on the area where we knew slaveni existed in 2017 as determined by previous island-wide surveys. The red dots there show locations of slaveni back in 2017. So based on the 2017 distribution of slaveni, we selected grid cells to survey. These grid cells here are indicated by the yellow dots that indicate the grid cell centroids. So what we did is, as we were out there in the field surveying each of these grid cells, if we observed a slaveni in a perimeter cell, like say one of these cells along here, we then moved out into the next adjoining cell and surveyed it and so on and so forth iteratively until basically we ran out of working days. And this is just a close-up of the grid cells with the centroids and the actual physical transects running down through the middle. Methods, we conducted visual searches in each 20 by 20 meter cell, visited each cell up to 5 times. We conducted searches during the hours skinks were active, which was approximately 9 in the morning to 4 in the afternoon. They have a kind of a short work day. We counted all individuals of all species seen in a 3-minute search in each cell. We recorded the time of day, we estimated percent cloud cover, and we also estimated the percent sunlight patches falling on the forest floor, and I'll explain how we did that in a second. And as another component, we conducted sticky board trapping on a subset of cells, 40 cells, 4 traps per cell, 30-minute durations for each sticky trap, and we did this twice in each of the 40 cells. So Jonathan pointed out that they found Slaveni out in the open. We don't see that on Cocos. The forest floor where Slaveni occurs has patches of sunlight that it and other species use for thermoregulation. So what we did was we estimated the percent of patches of sunlight within each 20 by 20 meter cell using these cards as shown here in the field. The dark green represents sunlight patches, the light green represents shade, and we'll return to this a little bit in a moment. And just an adult Slaveni in the deep forest utilizing a patch of sunlight. You can see in the background, there's a more open area out there that opens up. And again, they don't really seem to occur out there so much. So again, we counted all Slaveni seen within a cell within a 3-minute period. We predetermined that 3 minutes was enough time to visually enumerate all the lizards that were active and visible within a cell. That was a kind of surprisingly low period of time for us, but we checked it over and conferred with each other and standardized and determined that yes, a person can walk around in a 20 by 20 meter grid and basically see everything there is to see that's out and moving about. So we simultaneously counted all Carlia and Amoya ceruleacata because simply it was easy to do so. And we also use these counts as covariates in the occupancy model for Slaveni. These two species were by far the most common species in these cells. So results, this is exciting. These field cells represent all these cells that we were able to survey. The red filled cells are those where we observed Slaveni. Here, this map shows the maximum numbers of individual Slaveni seen in each cell on a single visit. So in some cells, we observed as many as 4 Slaveni on a visit. But the interesting thing here is look at the perimeter cells going along here where there's no observations. So it looks like we pretty well circumscribed the present range of Slaveni on Dano, but it's likely going to be somewhat larger. One of us found a Slaveni down here to the west, quite a bit further. So that was kind of interesting. So the occupancy results, look at the top row for Amoya slaveni. You can see here under occupancy, this tells, we found that 80% of the cells we surveyed are expected to harbor slaveni. So that's good. 97% of the cells we surveyed are expected to harbor Amoya ceruleacata. And unfortunately, 100% of the cells we surveyed are expected to harbor the invasive Carlia skink. Looking over here at detection probabilities, detection probability for slaveni was relatively low as expected, given that it's a rare and kind of secretive species. Detection probabilities for Amoya ceruleacata were much higher, up around 80%, and for Carlia, up around 90%. Here's the model, or I should say the set of models for Amoya slaveni. The top model is uppermost The poorest performing model is down at the bottom. The top model includes, again, the percent of sun patches falling on the forest floor. It also includes the number of Amoya ceruleacata seen as a covariate. So those things, Slavoni and ceruleacata, are positively related. And it also contains the method. The method was just either visual or using glue board trapping. But note that these AIC values are not all that different. So there really is no one model that's that much better than the other. Detection probabilities as a function of percent sunspots are interesting in how they vary among these three species, reflecting their thermal biology. Data for Slavoni indicate, at least on Cocos, that it's mainly a forest species inhabiting well-shaded areas, having relatively low levels of dappled sunlight on the forest floor. Detections of Amoya ceruleacata, in contrast, increase at sites with having somewhat higher levels of sunlight. Detections of Carlia, not surprisingly, are fairly flat, showing its relative indifference to sunlight levels. This is just one reason why this species is such a successful invader. Getting back to the survey method used for Slavoni, these are the results of sticky board trapping, the averages. Note that the visual surveys give better detection probabilities than sticky traps, but these averages are not significantly different. But that's good because it means that visual searches, which are quicker, easier, less costly, and most importantly, they don't pose any risk to the species. They work quite well. So comparison of our Slavoni sticky trap data with historical data are interesting. Our catch per trap hour was much higher than that for Dano back in the late 80s, where this data comes from. We had 0.062 versus Cocos is down there at 0.005 back in those days. So things are better, at least. It was also higher than it was on Almagan Asuncion back then. Looking at the same sort of data for Sarigan, our catch per trap hour on Dano is comparable with that on Sarigan in 1999, 2000, around here. So this is all not too bad for Dano. So conclusions, the survey methods we trialed will be effective for monitoring the Mariana skink going forward. Skink appears to be continuing to rebound on Dano, just partly based on the data here and anecdotal observations. Further occupancy studies are needed to assess trends. Identification of other important environmental covariates will improve detection probabilities, and we'll be hoping to trial some of those the next time around. So in conclusion, I'd like to thank Jeffrey at DAR for expediting our collection permit. We would have been kind of dead in the water and losing days of valuable research time if it wasn't for him. And last, let's all keep working to ensure that everything on Dano stays as it is and gets better. That's it. Thank you.
They're very sympathetic that you're in Colorado in a snowstorm. It's very sunny here right now.
Yes, I'm sure it is. I should also point out that I can only hear the presenter. I can't hear anybody out in the audience.
Oh, we'll get mics. Does anybody have any questions? Let me know if you can hear this question.
Okay.
Can you hear me? This is Margie Palanru. Pushi Palanru, who collected the first specimens of the Moyas Lemni. They were collected somewhat west of the area where you were looking at. I'm wondering if that was perhaps an indication that they were more common at that time with the wider distribution.
Yeah, I heard the question. I don't know, further west than we were, where we were at would have been well into the Loran Station, the former Loran Station property, which was, severely degraded, and is still not really back to where it's still not really come back. But there are some Slavonite down there, but it's really hard. I find it hard to believe that Medjugorje could have found that many down there. The area where we were working is an area of coconut forest that's remained intact and was spared. It was spared from both the Loran Station development, which was, by the way, built twice, and sandwiched in between the resort, of course, which wiped out another good chunk of property. So, at least in my opinion, I've always felt that the main group of Slavonite on Dano has always been located historically right between the resort and a little bit further to the west where the Loran Station is at.
Yeah, I may have got my directions mixed up then, but it was outside of the area where you were looking, which maybe means they were more common back then.
Okay, Tom, it's Jill. Thank you for showing me the only Slavonite I've ever seen in Ano. But looking at your distribution, we're talking about a tiny range on a tiny island. And I just gotta assume that that population has some unique genetics compared to the other populations in the Northern Islands. Just assuming BTS gets eradicated, we're going to assume that's going to be a success, right? Then what would you do for this population of Slavonite in terms of conservation?
Well, I think Robert has some really good ideas about making other parts of Dano more suitable for Slavonite. The far eastern side of the island, of course, was wiped out by a number of typhoons in the past, resulting in mostly being Casuarina growing back, which is pretty much worthless for lizards and Slavonite. So that whole area needs to be rehabilitated. Different trees planted there, in my opinion, and I think probably Robert's opinion. And right now what they're using coconut forest. Coconut forest is not the greatest thing in the world either, but it does produce the debris that forms the debris piles that these guys seem to need for cover. But that's not to say other tree species can't provide cover either. I hope that answers your question.
One more question.
I will say that I don't know how many of you are familiar with the plant, the nursery trade plant pothos. It is going absolutely crazy on cocos right now for some strange reason, and growing into the Casuarina zone and going up the Casuarina trees and shading everything out. And it's actually creating more habitat for Slavonite. But I think the pothos is going to become a problem eventually. It's going to go island wide in a very short period of time.
Hey, Tom, just one question.
Hey, Tom, it's Chaz. Thank you for sharing your passion with us. I wanted to ask a more personal question.
Yeah.
What fascinates you the most about Slavonite? And also, what's your second favorite lizard?
Well, some of the old timers know me as a brown treesnake researcher when I worked back with the NWRC Wildlife Services, USDA, and then more recently running NAFAC's biosecurity program and fighting little fire ants and such. But before that, I was a herpetologist. And so when I was out there living, you know, when Bjorn Lardner showed me the first Slaveni, and it, you know, it had just basically reappeared from being extinct, that just kind of bit me. And I've become fixated on that particular lizard ever since. But my other favorite lizards are probably Scloporus lizards since back on the mainland, since I did all my graduate work on Scloporus lizards. But I like all lizards.
All right. So I'm supposed to give you a question to start things off. And being a teacher here at Guam Community College, I want to excite my students and maybe, you know, turn around their heads, give them something else to, you know, to think about in terms of a career. And I know that each of you have your own choices, like Dr. Matthews said, their favorite skinks or animal. But what would you say to a potential student, intern, or employee of a research assistant, something that might grab them, hook them to come and work with you or go in that same field of study? Anybody?
Well, I can only say what got me into studying creepy crawly things. When I was in college, I spent a semester in the Salmon Study program. Long story short, I ended up at one of the another student's parents' house, and he thought I was dating his daughter. And he took me out running, and he said, "What are you interested in?" He says, "You know, I'm an accountant. I hate accounting. I always wanted to study birds. So, you know, in the wintertime, I go down to Belize and study birds. So if I were you, I would do what you want to do and not do what your mother tells you to do because you have to support your kids." Okay? Now, obviously you have to do what's important for your family, but you only get one choice. And if you don't do something interesting or something you want to do, you will regret it. Okay? There's some things I haven't done that I sort of regret, but I've pretty much done, I don't know what I wanted to do, and I feel very fortunate about that. Sage words. Yeah, I'd echo that. I think we can't do it all, so you might as well do what you really want to do to make that a career. And I think probably most of us have found ourselves really entering into this because ultimately, you know, we really just like being out there and just studying stuff and just learning. You know? So if you enjoy, at least for a career path, if you enjoy just every day coming in and finding something new and being challenged by that, then you know, this is the career path for you.
Yeah, I mean, those are good pieces of advice. Something that I would say is that, like, just because you change paths at some point along the way doesn't mean you can't come back to what you originally wanted to do. When I was 5 till I was 19, I wanted to be a herpetologist and study snakes. And then at some point in college, I liked a fish class, and I was like, "Oh, I like fish." So I did fish for 15 plus years, and then I just recently got back into herpetology. So, and I didn't think that this was a place. I mean, growing up in the Southeast US, like there's herpetofauna everywhere, and like moving to the Pacific Islands, the diversity is very low relatively. So it wasn't something that I thought, oh, I'd get back into herpetology, but sometimes you find a way. So, you know, if you have a passion that's like secondary to what you're also passionate about, you know, don't rule it out in the future.
I mean, I think for me, it's changed over time, but at this point in my life, working in the Pacific it's really an opportunity thing for me. Like so few people in the world get to see these things, to go to these places and experience people from completely different cultures, different languages, different ways of thinking about science. And to me, it's like adding such value to how we do science. It's important, it's more impactful. I'm always amazed. We can go on these trips like Alamaga, and we were there for 3 weeks. And it's pretty friggin' miserable at times. And it's great too, but the relationships that you can build with people that come from such disparate backgrounds over a short amount of time I can't describe the power of it. It changes me for sure. And I will have nothing but fond memories of these trips for the rest of my life. And I'm not kidding when I say it. Every trip changes.
Tom, do you have anything to add to that?
It's snowing so much. It's white out there.
Here I am. Yeah, I would say, yeah, I mean, my working out in the Marianas definitely changed my life, and I got the Marianas bug, and it's kind of my second home, and I think it's just a great place to work, largely because of the people out there and how nice it is working with everybody.
Okay, thank you. Does anybody in the audience have a question for the panelists?
Hi, everyone. So I feel so spoiled in some ways as a bird person because birds are so visible, right? And they're beautiful. And when you tell people you work with birds, they're excited and have questions. So as folks who work with often overlooked species, what do you think the best part or the worst kind of part of that in terms of your interactions with the public are?
Mm-hmm. That's what makes them interesting, is the rarity and the challenge.
Totally, it's the rarity things, and exactly that, going out there and finding something. You're seeing it all the time. It's not nearly as exciting. It's like the rare, a rare beer that you can only get because they don't distribute, so you have to go and get it. It's horrible now.
The challenge, of course, is the funding, right? From a practical perspective, it's just being able to go raise enough awareness that people view it as an issue. Sometimes, I always like to joke, there are probably like 10 people in the world that care about some of the questions that we're asking, and 9 of us don't agree with each other. But for the most part, it's really a closed meeting. We just need to do a better job of, I think, doing exactly this, going out here and meeting with the public, presenting our results out, this kind of forum. So the more we do that, I think the more we can elevate some of these issues to the public consciousness. I think a couple of years ago, I said we have a PR problem because I don't work with charismatic, but I like making fun of you guys. But it is a problem, and for me, it's depressing because I can't even get my kids interest. I can take my kid to the butterfly rearing tent and say, hey, you can help out, I'll clean up. This is the only time you've seen more Mariana 8-pot butterflies than 99.999% of anyone. They're like, oh yeah, when are we going? How do you sell it? Okay, how do you sell it? At one of those CIS conferences years ago, there was a meeting with the hotels, conservations. We want to increase knowledge and awareness. And hotel guys stand up and well, we talk about dolphins all the time. Give me a break. That is a challenge, and it's not something that's easy for scientists to do because we don't know how to do PR. I mean, the best thing I've ever done was make a Instagram page for the small island study. I don't fit more than 20 people subscribe to it, but it is a challenge.
I like that Instagram page. That was great.
Okay, so we have a PR problem, so we need to hire PR people. Any other questions?
I just had, yeah, I mean, that exactly what Kurt was saying is we're not the best, always the best communicators. And so, you know, as far as people thinking about careers that sort of are allied with sciences, but maybe we can use skills from, you know, maybe from outside of it, like communication. I think there's a pressing need for more and more. I think science communication is really essential. And so having people that can do that, so that we can work with, I think that's what we need more of.
I thought I saw a hand of questions.
Okay. We started this meeting with a tribute to Dick Randall, and I'd like to include some advice he gave me to address this question. You know, we biologists tend to be really critical at those people that damage the environment. And I was that way. I came back from University of California in the 60s when students were all protesting, and I was kind of hot-headed. And I was talking, Dick came back about the same time, or came to Guam about the same time. And I was criticizing someone who did something stupid. And he calmly said, I like to give people an out. And that small, quiet sentence changed the way I operated. And several years later, I had moved to Yap Island. I ended up working with one of the worst environmental enemies, who later became a great environmental hero and started a movement that continues today. So be kind to one another, biologists and the bad guys.
I don't have a question, just a comment for Tom. It's kind of awkward because if you were here, I would just pull you aside after. But I saw you had a radius brewing company hat on. I went to Emporia State for 3 years.
What?
Okay, so I think we'll close the panel now. Can we have a hand?
Right, so the next thing on our agenda is we have another panel.
Last one.
Last panel, panel for the panel, to talk about conservation planning. So if I could call up Diane, Christine, Elsa, Henry, and maybe Brad, and for sure Brad. And so we're going to have a conversation about conservation planning, because it would kind of be awkward if we had spent all this time talking about conservation and hoping and wishing, but we have to do, right? We can't just talk about the things we've done either. We've got to take all the ideas we've been stirring up in one another and kind of turn those into plans. And we're going to have a quick little overview of some of the type of planning that has occurred across the Marianas, and then we're going to have a discussion around, has it been adequate? Where's the room for improvement, challenges, opportunities? So we've provided our panelists with a couple of questions, and then we'd also like to open it up to each and every one of you to ask them a question.
I don't know why you're gonna yell break. Sorry, it's an extra. I don't know if the box is around.
Stand up. That's what I was looking, where were you? They didn't send us questions. No, I didn't know. A lot of experts in the room made it flex. Okay, that's okay.
All right, so, okay, so, yeah. Okay. So we, this was an idea that kind of came out of some conversations in November with Manny Pangilinan at CSW and Saipan, kind of thinking about, and it's like something that I think we have had some panels before, thinking about planning across the each island and then across the whole archipelago, and then thinking more broadly across Micronesia. And I think one of the benefits of this conference is that it gives us an opportunity to get everyone together and to think at this broader scale. So it seems like a good opportunity to try to figure out where those gaps are and what things we might be more successful at if we coordinated efforts or like thought collectively about how to tackle some of these problems. And so that was the inspiration for this panel. We unfortunately we're supposed to have either Rosea Marquez or Tam Greenstone Alfayo from Micronesia Conservation Trust on here to talk about Micronesia Challenge and kind of bring the larger Micronesia perspective, but they were not able to make it, unfortunately. And then we're still waiting. There's a couple more people from CNMI who are supposed to be jumping on Zoom that will hopefully be on here as well. But I wanted to start this off with just a quick overview of the foundational plans that exist that I can think of and that we've kind of brainstormed and just to kind of set the stage for where and how conservation planning occurs now. So I'm going to start kind of as kind of small scale in some angle and then try to get more broad spatially, at least over time, spatially or taxonomically. So taxonomically, there's a variety of species level plans for our endangered species. These are just two examples of those plans that might drive research efforts or conservation efforts for endangered species. There's lots of individual landowners or managers who have plans that guide the actions on their land. So like the Guam National Wildlife Refuge or the Integrated Natural Resources Management Plan for Joint Region Marianas are a couple of examples. There's a lot of other individual landowners that have those particular plans that guide their own actions. There's watershed management plans. These are a couple of the examples, in Saipan, the Garapan Integrated Watershed Management Plan, the takatsukasae Integrated Watershed Management Plan. This is one from Guam. There might be some others. One of the wonderful things is like many of you who wrote these plans are in this room. So it's really we have a wealth of experience right here. But these guide watershed managers, agency partners, and community stakeholders. So this is across multiple different land owners or managers guiding those actions. And then the guiding document for our wildlife agencies are these wildlife action plans. And every state across the country and territory is required to come up with a wildlife action plan. You can see Guam's here and Guam and CNMI's. And every 10 years that has to get renewed. And that really guides a lot of the actions for our wildlife agencies over the, and kind of what guides also their funding requests and research actions. And then similarly, on the forestry side, there are forest action plans, which are required for every state or territory. And you can see the Guam and CNMI Forest Action Plan, also 10 years, but on the opposite 5-year schedule. These guide, they give you an analysis of what the current state of the forest is and make a roadmap for forest management. And then collectively, you can kind of pull them all together at the national scale and see kind of where conservation, what the priorities are for conservation. You might have heard, if you were here Tuesday morning, there's a new habitat conservation plan that's in the works right now through Elsa's talk. She's here, that would be a way of thinking about how to protect and conserve threatened and endangered species in their habitats for future generations. And it's a lot to do with listed species and trying to think about how we can maintain those species while also facilitating economic development and regulatory streamlining. So then you can get to land use plans. This area is a little bit fuzzier to me, is exactly what the current status of all of these are. But the land use plan for Guam, there's a master plan that was passed sometime in the 60s. And I don't think anything has actually been passed since then, but there've been a variety of efforts. So this is one in the middle that was in the mid 90s that came together. And there's a North and Central Guam land use plan that was 2009 or so. There's a lot of community effort that went into all of these, but I don't think any of them have been actually adopted since then. And there's a variety of people in the audience that could speak to that. CNMI's land use plan was created 87, maybe something like that. And there's a law that says every 5 years it has to get updated. And so that guides a lot of the public land use in CNMI. And I'll just mention two other initiatives, the Micronesia Challenge, which started here in Micronesia with this idea of protecting initially 20% of the nation or of the waters of Micronesia by 2020. And then we increased that to 30% and then had 20% of the terrestrial lands by 2020. Now it's moved up to 30% by 2030. And America the Beautiful Act kind of is a federal initiative to also mirror this 30 by 30. This concept has really gone global, but started really here in Micronesia. So those are the main ones that I will focus on. And I ask all of our panelists to, if there's a gaping hole, please make sure you mention that, or in the questions if there's something big they're missing, but kind of laying the landscape of some of the major documents that guide all of our efforts. And I'm going to turn it over to Stephen now for questions for the panel. Yeah. Okay, I'll start that. So then I think what we'll start with is I'll have each panelist introduce yourself and your position and kind of what plans kind of drive your thinking and your, like, what do you look to, either things you've created or things that you are responsible for creating right now that are guiding conservation.
How much time do we get for that?
Just a couple of minutes. Let's go across. Yeah, you can start with Dan and then we'll grab it.
A minute.
Just like, yeah, a minute or two.
Hello, today, everyone. My name is Christine Camacho Farron, and I am the Forestry and Soil Resources Division Chief for the Guam Department of Agriculture. It's really weird for me to talk sitting down when I'm in front of a group. So what drives us? I've had many conversations, several of you in here talking about the vision, the plan for forest and soil resources from our division, from our department. And what we're looking at is a full ecosystem management approach. So it's not about the individual species, which we all know there's barely an understanding where these populations lie and whether or not they're thriving or not. But it's also just remembering that with forestry, it's everything. It's soil. We're growing our habitat. We're protecting and growing potential carving, potential harvest for our carvers, for our zomphi, for our potential timber, small scale timber industry. Let's harvest, grow, and harvest locally. So there's a lot we can do. And we actually pull from many of the local plans that you saw. So the big master plan for North and Southern Guam, I do believe that's being updated. And that's happening through the Bureau of Statistics and Plans, Guam Coastal Management Program. So if you have questions about that, you can contact Edwin Rages with that department and that program. So most of the Guam Forest Action Plan, all of these plans are created with community and stakeholders. So whether you're professionals, your research, your in-agency, your nonprofit,
you name it, we try to put those representatives in the committees so that we can get a more broad scope and review when it comes to what are the goals, what are the expectations, and bear in mind, we are all of our agencies beholden to funding pots, which have priorities, national priorities and goals. So, but we don't just do that. We live here. So we address what we need to see happen here on the island. Micronesia Challenge, for example, I was so proud to be able to be a member of that team for terrestrial measures. It's been going for decades, and we've blazed the trail for all of these new initiatives that you see. Oh, big scale, G3 is great. I'm not crashing them, sorry. But you know what I mean? The big umbrella initiatives, a lot of that started here in Micronesia with the Micronesia Challenge. And for Guam, we realized early on that it required an additional piece for Guam. So Guam added a cultural piece, a recognition that across Micronesia, not only are natural resources being protected, but they perpetuate and live their culture. And here on Guam, there was a bit of a struggle. And so our Micronesia Challenge incorporated culture in there. I've seen it get a little lost over the years, but it was part of the first initiative. I'm going to stop there.
How much time do I get? You have more? No, there's a lot. There's a lot.
There's more questions, don't worry.
Yeah.
I don't even want to follow that up.
Whatever. You have a great vision.
Section. The state wildlife action plan was mentioned, and it's very similar, I think, in context to the forest action plan. It has plans, very high level, general plans for species conservation, both aquatic and wildlife. So it kind of, well, we work on that plan specifically for the state wildlife money, and they make you make the plan, but it really just puts into words everything that our division strives for all the species. And on a project side basis, of course, I'm Queen of Cocos Island. I've been working up there for a long time when you're talking landscape and the holistic. Of course, I cheated because I took a Brown Treesnake free area with no vigilance out there, and we reintroduced the Guam rail and took out the rodents. And now we have and we were demonstrating what Guam could look like without brown tree snakes. And now it has brown tree snakes. So now we move to the next challenge, and now Cocos will be a demonstration for site for how we can eradicate snakes from an area and watch the wildlife recover.
Diane, can you say your name and agency?
Oh, I know all these people. I'm Diane Weiss. I'm with the Division of Aquatic and Wildlife Resources, and I'm the wildlife supervisor. And we're looking for staff.
Same.
My name is Elsa de Molenaar. I'm the Associate Director for Natural Resources at the Center for Island Sustainability at the University of Guam. And I want to kind of go from species kind of to habitat to like island-wide kind of conservation all at the same time. So I studied the Hudson Lago tree, which is a Serianthus nelsoni name for Guam, but it also occurs in Buddha, which is from Guapi. And that encompasses a lot of the things we're talking about, because like what I found through genetic research I did is that Guam has a unique genotype compared to Rhoda. So, but there's only one tree left on Guam. So, but given that the species is conspecific, we could try to actually make a combined, a population that combines both. So that was a pretty big challenge to kind of try to find ways to make that happen. So I wrote a proposal to see if it gets funded. But I think genetic research is going to be a foundation if we want to look across our archipelago, because we have to be careful if these, there's inbreeding happening for our Hudson Lago, for sure, but we also want to make sure we're not causing outbreeding. So I think genetic research is for sure going to be a basis if we're going to think archipelago-wide.
I also think the cultural aspect is really, important. Thank you for mentioning that, Christine. And that's what's driving conservation. I think, on our islands, we really want to preserve not just the species, but the cultural aspects. So I always say biocultural diversity. Let's protect that. And of course, species are, part of an ecosystem. And one thing that I think is really crucial is that we protect or look archipelago-wise what our really high quality habitats are and try to protect those first. And that's also one approach that we have in our habitat conservation plan that I presented on. We really want to, when there is development happening, we really want to ensure that the development doesn't happen in those really high quality habitats. And what we're hoping to do there is like the tiered permitting fees to kind of discourage developing those high quality habitats, and when there is low quality habitats, to try to mitigate in conservation areas that Christine and all of you are working in. Like have that reserve network from the forest system plan, from Micronesian challenge, and try to restore, protect the forest, restore, and also our invasive species knowledge. We all mentioned many challenges with invasive species. Try to kind of look island-wide and archipelago-wide what we can do about it to actually make those habitats stronger and protect them.
So my name is Henry. I am the co-PI for the Washington portion of the Mariana Fruit Recovery Project. So I think my project sort of influences purview over island-wide and sort of archipelago-wide. Conservation is much more narrowly scaled or more narrowly focused than the others who would have been invited onto this panel. But for us, we're mainly driven by the draft recovery plans. Right? That's sort of our guiding. So what we use for all of our grant applications and funding for justifying the research that we would like to do in the future. Something that we have been sort of running up against is the idea that, or I guess the fact that this document was written in 2005, when the species was still, all the species was built on Guam, and so it is outdated to the degree that has made addressing the challenges that the population is facing now. It is outdated and made it difficult to address the challenges that the throw is facing now. Island-wide, there's, because our project is based on rhoda and we have sort of permanent nature to a degree, and the crow is sort of one of the more threatened species on rhoda. We do all our data is shared with the USDA, the U.S. Fish and Wildlife Service, and it is getting incorporated into these larger island-wide plans. The U.S. or the National Park Service is doing a special resource study to gauge the significance of rhoda's natural resources and whether they rise to this threshold and warrant federal funding and federal support, and whether the community of rhoda, whether actually funding that concept is feasible or of interest to the people of rhoda. So that's something that more as a side project, and as like, I'm not tied to the Park Service in any way, but as the community of rhoda is very valuable to me and our project, and we are attempting to serve the community in any way we can find. We have found ways to volunteer to communicate and advocate for the people of rhoda when they're faced with processes that are not being carried out in very culturally appropriate ways. And so, yeah, I guess that's a way in which we sort of live outside of the narrow focus of our project to serve these greater wide.
Today and good afternoon. My name is Brad Eichelberger and I'm the Chief Geographic Information Officer for the Division of the Fish and Wildlife for the Commonwealth of the Northern Mariana Islands. Our main priorities with the Division of Fish and Wildlife. Our main priorities are to protect and conserve our natural resources in the CNMI, which includes delisting the listed species as well as preventing further listing of our other native species. A lot of our research and priorities are governed by the State Wildlife Action Plan, as Haldre had mentioned, as well as the recovery plans that are recovery plans that are essentially used to help recover the listed species. I think we do have some interest in doing something similar that Guam has done with the kind of more holistic habitat conservation plan, and also in the future maybe looking at like a conservation movement.
Thanks. And then we do have Zachary Williams and Marlon Appetit from the Division of Coastal Resource Management in Saipan, who just zoomed in with us. So Zach and Marlon, if you'd like to answer the prompt.
Hi everyone, how's it going? My name is Mariana Fruit Dove. I am the Coral Reef Initiative Communications Coordinator here at the Division of Coastal Resources Management. I basically build upon existing communication efforts to increase awareness on the importance of our coral reefs. The plan that I use to guide my work is the Strategic Communications Plan and also the Coral Reef Initiative Communications Plan.
And my name is Zach Williams. I'm also with the Division of Coastal Resource Management here under our Coral Reef Initiative, and I'm the watershed coordinator. So we're guided by both the national, NOAA's national coral reef priorities or pillars, whatever vernacular they use, which includes, among other things, land-based source of pollution. So that ties my work into our coral reef initiative here. And then a lot of my work is involved in the development of our watershed management plans for our local identified priority watersheds here, as well as implementing in those plans. So we have 4 priority watersheds: the takatsukasae on Rhoda, and then here on Saipan we have Garapan within the West Apache watershed, Lalao Bay, and most recently the Zosterops watershed. So it's making that connection, that ridge to reef connection with our management, and taking this holistic approach in cooperation with different government agencies and nonprofits and community groups on island to manage our watersheds. Thank you.
Thank you, Zach and Marlon. So we're going to start with a pretty easy question. What are the gaps? And I know I say it's easy because we tend to always think about, oh, I wish I could do this, I wish I could do that, or I can't do this, I can't do that. So that's why I think this is the easy question to start off with. And so who am I going to let's go with Diane. What's the gap?
Let's try not to make eye contact.
That's why I picked you.
What are the gaps? Can you repeat the question, please?
It's just, what are the gaps in island conservation? If you have to pick one, just to give everyone some time.
It's easier to talk about what we do because we do a lot, but you're right. What are the gaps? I mean, we have a lot, we would do a lot more if we had more funding, but you want something like biological. Yeah. On Guam, what I would like to see is areas where we're talking about ecological restoration in a landscape scale. And we have several projects going on. Pupus Island being one. The military has their, I went to Kapuas yesterday, if anyone, for their restoration center Kapuas. Highly recommended. And forestry has their areas, and I guess the gap is a little bit of coordination, because I'm focused on wildlife, and yeah, I'm focused on wildlife, and I do my best to partner. Well, I focus on fur and feathers a lot, because that's the history of DAR. The snake came in, took out all the birds, so we're working to restore the listed species. So I work, I work with birds, with snails and butterflies. I'm trying to bring in those other species, but really the main thing is they need habitat. So there's nobody preserving with pristine limestone.
Well, that sounds like a perfect segue into Christine. Maybe you can answer, and maybe your gap is that you don't have furs and feathers.
That definitely impacts our effort. So for forestry, I'm just going to say this, like everybody can feel this. It's a capacity challenge. So we have these amazing programs. I'm telling you, I can sales pitch forestry for days, which I know very well. It sounds like. When you said more money, I'm like, I don't want any more money right now. I need the people to manage these programs and get them moving. We have wonderful projects planned and vision that goes beyond for our grandchildren. But unless we have the people committed to working for the government of Guam, people, for managing those resources, we're going to struggle with these capacity challenges which perpetuate the gaps. So what I mean by that is reforestation projects needing monitoring. We know that we need to get back into the plantations, into the project areas, and start thinning, which we do. We open at just enough for native to thrive. Then we underplant wherever else we can throughout the forest, because as long as those trees are in place, they are keeping fire out. They are keeping they're keeping grasses out, keeping fire out, keeping further intrusions of invasives into our forest out. So monitoring is needed. That is a major gap. Analysis of data, once that comes in, that will be a gap. I've been meeting with Ashley Lehman, Forest Inventory Analysis Team. We're going to be starting that in 2023. And there's a lot of data already available, 20 plus years of data. And we have an amazing guy, Julian Denby, doing the analysis work right now. But there's a lot of data coming out of that FIA work, and we need people on the team willing to commit to looking at that data and helping us answer some of the questions we don't know we have. That's a gap.
Yeah, so I think an overall problem was invasive species, right? So, we didn't talk that much about plants and everything, but I think implementing, once research done and we know how to, for instance, I'm working on Pipex removal, once we know how to do that, how we're going to implement or really do this large scale for Guam, even with the snakes, right? How we're going to do this large scale. So, because like I mentioned the high quality habitats, but I think it's also important to not just, people, volunteers, homeowners can be part of, also, really preserving our biocultural diversity, right? And be part of it, but how we're going to find possibly funding and resources to teach all these methods so it can be implemented and, our ecosystems overall can be more supportive of our native species, because ultimately we need to be able to connect all these habitats. And, if you look at a lot of these endangered species, it was mentioned by Matt, for instance, we need like 20 populations of butterflies. We're not going to meet that goal if we just keep it to the high quality habitats. So I think that's for sure a challenge, how we're going to, battle this island wide and archipelago wide, right?
I think that's a good answer. I want to change gears a little bit and think about gaps in a different sense. I mean, maybe I've been living on Rhoda and I'm not as aware of the dynamics on the other islands, but for me, there's this combination of a credentialing problem where for entry-level positions, requiring degrees that are pretty difficult to attain. We don't have a great pipeline in an education system to, recruit and retain people who are from these islands and include them in the work and are them to to stay and stick around, and rather than move to the States or somewhere else, that there are more job opportunities. I think on Rotary, the scale is a little smaller, so it's been easier for us to put in the extra effort and the extra work to make sure those entry-level positions are accessible to people who don't have trees. At the same time, I suppose on the other end, beyond lowering barriers to entry, also I think it's really important, and this is probably something more for Guam and Saipan to really try and bolster the education system specifically for this field and make it a viable career for the people who are from these islands. I guess, put another way, try and sort of free our field of a dependency on temporary positions that are filled from grads from the States, and try and create more permanent positions and fill them with purpose.
I think in order for us to make informed management decisions, in order to make informed management decisions, we need the appropriate data, and working the 14 islands in our island chain, having accessibility to some of these islands creates a knowledge gap. And that's probably a huge issue that we're facing, and it kind of leads into the next challenge is time. So it takes time to collect that data, and also we could be running out of time for certain species if we're trying to make sure that there are going to be differences.
Thank you. So I am going to pitch the next question to our panelists on Zoom, and given our time, I'm going to try and wrap as many of our planned questions into one. And so, we've heard about these gaps, and I think throughout today we've talked a lot about, so our gap that I want to pick on is like data limitation and personnel. And Henry was alluding to this wealth of personnel that happen to be, right around the corner from us. They're our neighbors, they're our friends, they're our family. And so what are maybe some ways we might integrate the community at the village, island, archipelago scale to overcome these gaps in data and personnel and in equity, right? So we're going to start on Zoom. I know, Christine, you're itching, but we want to practice some equity in real time. So Zach and Marlon, not to throw you on the spot with like the combo question, but I appreciate your
Hi. I totally agree in terms of sort of getting more local residents involved in the work we do. We have positions we're currently trying to hire for, that sort of thing, in those jobs. And a lot of, I know people from our agency, most of the people from our agency are from the island, which is honestly fantastic to see. And I think a big contributor to that has been our coral internship we have here. I mean, a lot of people in this office started out as interns through that program, including our former director, and have since joined the agency, and it gives a early opportunity to get a foot in the door with the natural resource management and conservation field. Marlon can definitely talk more about that because it's a big program that she manages. But I see that sort of early job opportunity, just providing early job opportunities in the conservation field is a good way of getting people into this line of work.
Yeah, so reflecting on what Zach said, personally, I was an intern under Haldre when I was in college. So opportunities like that, education and outreach opportunities or internships, I think they play an important role when you want to involve the local community. Just having that opportunity for them to get involved and to see the work. I can honestly say from my point of view that all the internships I've been through, especially the one under Haldre, helped me solidify my decision to remain in the conservation work. So, yeah, definitely more opportunities for local residents.
Thank you.
Thank you both for that answer. And I am also a product of that very internship that they mentioned. I think Amanda also did the CRI internship. So the proof is in the pudding right here in the room. So let's give it up for that amazing idea that actually kind of works. And so, Christine, now you can have the floor.
You know what? Anthony Tornado was our Micronesia Challenge intern as well. So that's a win. Got a biologist sitting right there. That's awesome. So I was jumping out of my seat. I apologize. But one of the grants that we're going for right now is the Inflation Reduction Act funding. And so I'm going to be submitting that hopefully this next week. But incorporated in that is to address, right? We can talk about these gaps all we want, but if we're not making any movements towards addressing them, then we're going to be frustrated forever. So one of the things we're proposing is to support Guam Trades Academy. Now, degrees, you hit it right. We're asking for these degrees, and we want to give people an opportunity to enter and go for higher learning and education through their departments, because departments investing in their people hopefully keep people. We hope. I've never been one. You probably all heard people say, I won't train them because then they'll leave the minute they're trained. That's despicable. No, we will. Our leadership has invested in supporting team members to go and get higher education. We will support you with our funding as much as we can through that process. So again, Trades Academy, entry level certifications for tree care, tree pruning, landscape maintenance. This is entry level into industry. So we're going industry level, not just training in-house staff and partners, but growing an industry where people coming out have a potential to have basic tree care, tree knowledge, ready to go. Can you hear me? No? Okay. Oh, I'm sorry. So that's one. So that program will be developed and hopefully funded if we win this because it's competitive. The next route is supporting Department of Parks and Recreation. And this is the team, local team that's managing all of these green spaces, these other public spaces. And to really address, we all stay on the islands, disadvantaged communities, we all fall into that. But we can prioritize the communities that we want to reach based on Gura homes, okay? Gura housing, for example, we can start there, really grow the access to education, to more information and getting involved. There's more, I'll stop there. But the Trades Academy thing has me really excited because if we can get high school students and returning adults to go to trades and learn the basics, and when I say basics, tree care, tree maintenance, that can really do a lot for us in forestry, people coming in with that entry level and into landscaping. No more bush cut disease trees, no more ponytailing those palm trees, So. Oh, anybody else?
I'm personally curious to hear from Henry and else, just given the CIS, Guam Green Growth.
Yeah, Guam Green Growth, I want to mention for sure because I've in the recent year hired people that were trained through the Guam Green Growth program with the Conservation Corps, and they're very motivated and they they learn a variety of skills for 6 months. You know, it's very diverse, and I have 3 people on my team currently. I'm looking into hiring for fun, and it's also then they get interested in science, and if they want to study at the same time, that's a possibility. You know, working at a university, we can, you know, do part-time, you know, study part-time work. But another way that we get people interested in STEM, you know, and really do this kind of research is we have an NSF grant, includes where we have interns every year, 5 undergrads, but it's kind of a mentorship idea. So those undergrads get trained by mentors, so, you know, this is me. But then in the summer, 4 high school students are paired up with each student, and they get, you know, to learn some science. And it's, you know, really been very inspiring for those students, and they then want to start a career maybe in biology or, you know. But I do believe there should be like several possibilities, or, you know.
It's filling that internship gap by giving people a 6-month opportunity.
Right.
And we can do certifications as well, and college degrees and scholarships.
And some of those people are in the meantime already pesticide, you know, trained, and, you know, and they get their certifications working for us. And then, yeah, it's really cool to see. I actually really enjoy it.
In the past, if there wasn't there was an attempt in the past to look into ways to connect our research program with UW, like UW's academic campus in a more meaningful way, to try and set up people who work for our project with credits, undergraduate credits at University of Washington. I know that there was an effort passed, it didn't work out, and it's something that we should revisit. Most of our efforts have been really put forth on a small scale and like a face-to-face manner. One of the biggest lessons we've learned is that hiring on Rhoda is not the same as hiring elsewhere. A lot of intention, a lot of effort needs to be put forth to you know, approach people within the community and make sure it's understood that these positions are available and we are interested in community members filling them. So what that means is talking to the directors of the divisions of various offices on Rhoda, talking to community leaders, you know, people in the school system, going to all the stores on Rhoda and posting flyers up, physical flyers, tapping into you know, social media leaders, you know, local leaders on social media that have large followings, and asking them to amplify, you know, our job postings. And people have been, you know, really helpful in doing that. And that effort and that intention is really due to the number of applicants we're getting who are from Florida. So that's been a really big thing. Removing barriers to entry, not requiring a degree, putting extra effort into on-the-job training, understanding that there's going to be a greater investment on that front end, but also greater reward on that back end. As you know, I think that we were talking earlier about climbing the lattice or moving on the lattice, and rather than up a ladder. If we train someone from Rhoda, you know, to collect biological data to monitor the growth, it's not a loss if they leave our project and work for another conservation project on Rhoda and take those field skills, and, you know, do CRP work. They're, you know, moving laterally, those skills stay on Rhoda. They're still serving the island, even if they move to Guam or Saipan, still within the Marianas. And it's much more likely that that's going to happen for someone who's from Rhoda than for someone who's from New Jersey. So yeah, we're really like focused, you know, we're looking at the ground and, you know, really focused on very small scales, providing, you know, basic field gear technician. We applied for State of the Birds grant, Fish and Wildlife Service competitive grant, for funding to redo the specimen survey, the public perception survey that I mentioned during my talk. But we thought that the research does this could also mentor students in the high school. Rhoda High School has a co-op program for the entire school year. Every senior is paired up with a business or organization on an island, and every day for a few hours they shadow or help out that organization. And we're trying to get in. We're trying to do that with our base funding for monitoring population augmentations research. We don't have enough resources to contribute hours to that program, but we built participation into this Rhoda High School co-op program into that Save the Birds grant. Well, actually, it was a recovery challenge grant, and it was not awarded, and we reapplied and tweaked the proposal and reapplied for State of the Birds funding. So we're waiting to hear back on that, and hopefully we get good news. But yeah, so there is this local program that we're trying to get in the high schools, greater contact, more meaningful contact with the youth on Rhoda. That's where we're at.
I can speak to at least the data gap. I think we live in a pretty exciting time. We have a lot of access to ways to collect data remotely, so we can use drones. We hired Kaeli to kick off an acoustics program that's going to deploy ARUs to collect data remotely. There's a lot of opportunities to use things like data traps and things that collect a lot more data than we previously collected, and hopefully we can use that to answer some of the questions that we have.
All right, thank you, Brad. And thank you to all of our panelists. Can we give them a round of applause? So I think we should all be super jazzed that we have this great idea of really doubling down, investing in our local communities and encouraging them to work across the archipelago, as Henry was mentioning. If you have a talented employee and they're kind of thinking about maybe I might want to go explore different pastures, not necessarily green, but just different. I know that Christine is looking for someone to work for, right? And let's cross-pollinate. Let's keep it rich, keep it local. Well, thank you.
Local funding. I've got positions that will be opening, 2 forestry positions currently open. So where are you?
All right, so you know where to find the jobs. All right, so now we're going to transition into the very last thing, aside from the banquet, which all of you are going to show up to tonight. It's going to be lots of fun. Is our awards, right? So one thing we do at the conference is we hand out awards for a bunch of different categories.
So we're going to start with our student awards. But before we do, I want to actually first have everyone in the room who is are there any high school students in the room? Okay, how about college? I think we have some college students. Do you mind standing up if you're a college student? Or you can I want to recognize that we have students from, I believe, we have some Guam students, we have CNMI, any CNMI students over here. We have some Palau students, right? I think we might have some Marshall Islands students over here. Awesome, it's a long way. Any students from FSM? Yep, FSM students here. And then is there someone from American Samoa? Amazing, so let's give all these students a round of applause. And I think there's been a lot of opportunities for jobs and graduate school here. So come up to anyone. If you're interested in any of the things you've heard, feel free to talk to any of the folks who've been speaking about kind of, maybe it's not even this year, maybe it's next year or in 5 years, like something that might sound interesting. I also want to thank the judges for the student awards before we hand those out. So there are some anonymous judges. We really appreciate your time spent judging these. And that means that I'm going to turn it over to Anne Marie to give out these awards.
Yeah, thanks for that, Haldre. And yeah, just want to second that. Thank you so much to the anonymous judges who filled out the rubrics. We have really excellent prizes. So to encourage your students next year, please present. This year we are giving away signed copies by the author, Julian Uggan, signed copies of Julian Uggan's No Country for 8-Spot Butterfly, as well as a $50 gift certificate to Southern Mountain Gear, our wonderful outdoor store, which you should definitely stop by. They're a wonderful supporter of this conference. And so I'll go ahead and get into it. The winner of the best poster is Alexander Tudela. Thank you so much. Congratulations. And I've actually just been messaging with the winner of the best oral presentation because he left before knowing he won an award. And so the winner is Matthew Putnam. And I'm going to go ahead and share it with Elsa to give to him. Yeah, thank you so much. Our next award is the Environmental Group of the Year. And this award goes to a group that was started maybe a year and a half ago, 2 years ago almost, but has really, I think, blossomed in the last year. And that is Friends of East Landano. This was really a grassroots effort. There was a need in the community to do something about the snake problem, and it was really difficult for a lot of the agencies to be able to move quickly. Like there's a lot of structural things that make it difficult to move quickly, and this was a grassroots effort of getting people out to capture snakes. And I think there was some question about whether people would voluntarily go out and catch snakes night after, like multiple times. And it turns out they've had 175 individual volunteers, 62 search trips, over 1,100 search hours, and then have captured 31 brown tree snakes in that time, which is a significant portion of the brown tree snakes that have been captured on Dano to date. And so I think it really just showed that the community can be a major player in conservation efforts, and it's super successful, and it's run off a lot of heart from all of the organizers, and then as well as DAWR has been a central figure in the in keeping it running and funding it. And so I urge all of you, if you haven't been out on a Saturday night, to go out and catch snakes. It's a fun effort. And I don't know if any of either Olympia or Martin is here, but we're going to make a donation to Friends of East Landano for their award. So let's give one more round of applause.
All right, so our next award is for the Environmentalist of the Year, and this award is given to the individual who, through their actions, words, and leadership over the past year, have brought attention to important environmental issues affecting our islands. This year, we're awarding it to two individuals. One of our awardees uses the power of the pen to inspire and invite people from across the world to engage with issues affecting the Marianas. The other took action through the governance process, becoming one of the youngest serving members of their local legislature, and used this platform to launch onto the global stage. In concert with each other, these two amazing individuals shine a light on the polycrisis of climate change, colonialism, and biodiversity loss affecting and taking place on our islands. And they also just so happen to both be Obama Foundation Fellows. So we're very excited to present to you our Co-Environmentalist of the Year award to Julian Uggan and Representative Sheila Jacobalta. And so Joni's going to give a little blurb about Julian, and then I'll give one about you.
So what's in the turn of a phrase? When words speak the truth plainly and deliver a warning in powerful, terribly beautiful prose. That is what transforms a turn of phrase into a thought, an action, and a movement. This is the power of our awardee. So last night I was given some homework, and I was kind of looking around preparing for my turn as moderator and award presenter, and I came across this Guardian article, and because I Googled a spot butterfly, and I'm halfway through the article, I'm reading it, and I realized that the style is not in the form of a news article, or written in that style, but it's more like an essay. And then it's, it sounded a little bit familiar, the tone, the style. And so I scrolled up, and sure enough, there was his name on the left side of the screen in much smaller font than the title. And this was the title. In Guam, even the dead are dying. The US military is building on the graves of our ancestors. So that is such a Julian Uggan title. even the dead are dying. So our ancestors are suffering another death, and that is a desecration of their sacred resting places. This word identify a core aspect of our Chamorro culture, that of respect for our Saina and those who pass away, our Tautamona, those who came before us. So Julian is an accomplished author and Pulitzer Prize winner, as well as an attorney who at 28 opened his law practice, Blue Ocean Law. This was to defend the defenseless. And these defenseless range from indigenous people who are outweighed by powerful nations and entities with massive financial resources to beings with a tiny whisper of a world view. And that tiny whisper matters. I liken it to the phrase Black Lives Matter. So when detractors claim all lives matter, it's a form of erasure that keeps people blind to the truth and resistant to change. With his genius, Julian calls attention to our precious ancestral connections and the tiny butterfly that is unaware of its species' fragile existence as it concerns itself with mating and reproducing. He brings to the world the truth that massively destructive events threaten to silence our tenuous connections to our past and our natural world. So that when that is allowed to happen here on our little Pacific island, and in the aggressive way that it is happening right now, what's to stop similar destruction from happening everywhere eventually? But his message, if I may be so bold to interpret, is that when humans can care about the most vulnerable of beings, we can then care about everything. Igaga siha to itaka siha. With that, I am honored and pleased to present one of MTCC's Environmentalist of the Year award to author, philosopher, activist, and defender of the defenseless, Mr. Julian Uggan Esquire. Thank you. Sorry. Oh, sorry.
then I'd like to now introduce our co-environmentalist of the year, Representative Sheila Jacobalta. She was the youngest serving member of the CNMI's legislature, and she's if you can name an NGO that's based in Saipan, Sheila is either on the board, she helped started it, she's advising them, she's spending all her time, she's selfless in that way. And I wish I could have more words to describe what she's done. And it's nice to kind of circle back, as we often do. And we started this morning with some remembrance of some of the heroes we've had in the conservation world, and mentioned losing Cinta. And I think we are so blessed to have the next Cinta in every way possible in Sheila here. So I'd like to give Sheila the opportunity to share some of her words with each and every one of you. So Sheila, thank you and congratulations.
Thank you, Stephen. And wow, thank you so much for this honor. I am deeply humbled to share the stage with the one and only Julian Uggan. Congratulations, Julian, as well. Thank you to the organizers of this conference. It's so important to gather, to be around like-minded, passionate individuals who are doing good work so that we can share with one another, share your findings, your challenges, your wins. It all matters, and it raises the vibration of the field. And so I hope that you experienced a renewed sense of enthusiasm and curiosity in the past couple of days. So congratulations to the organizers. As a young girl, I was always outside, and I always enjoyed being outside. Born and raised on the island of Saipan, I was always swimming at Pau Beach, exploring the jungles in Asteo, or playing under the lemai tree, the breadfruit tree outside of my house, catching frogs with my cousins. That's something I wouldn't dare do today. But as a girl, the natural world around me was so interesting and so beautiful and big, and there was so much to discover. It encouraged me to explore and expand, to exercise my muscle of courage, because courage is a muscle. We all already have it, and it sometimes just needs strengthening. And so having access to the natural world really shaped who I am as a daughter, a friend, and a leader in my community. And so I am completely and just absolutely grateful for all of the work that you do. Undangkulu nasizus maasi, giliso, for gathering data, for sharing your knowledge, building local capacity, protecting the Marianas and her beauty. And so I'd like to dedicate this award to Auntie Cinta Kaipat. Thank you, Stephen, for bringing her name into the space. Auntie Cinta was a true environmental champion, community advocate, former CNMI congresswoman. We had shared values and we had a lot in common. And so it's very kind of you, Stephen, to say those words about me and Auntie Cinta. She was a leading voice against the destructive military training and testing activities on Pagan and in the Mariana Islands. She was born and raised on the island of Pagan. She led efforts for the Mariana Trench Marine National Monument. She was a founding member of numerous nonprofit organizations as well. And she was a dear auntie and mentor to me. And because of her guidance and love, I was able to fly and represent the Marianas on many different platforms, in many circles, internationally with world leaders. And so I ask each of you to mentor some young person in your community, in your home, in your personal and professional capacity, because there's so much work to do and there's so much beauty to protect, and our young people have the energy for it. And we must use their energy for the natural it is a natural resource that it is. And so I'm so grateful for this award. Thank you very much for the recognition, and I have so much gratitude. Thank you, Stephen.
Oh, thank you, Sheila. We do have a physical award for you, and I'm coming home on Tuesday, so I'll find you.
I just want to note that this is an image from Zia Kreitzner, who's maybe not here, she's a biologist on island, and the 8-spot butterfly was from CJ Paulino, an image taken there.
All right, and so then the next award we're going to be giving is to our distinguished environmentalists. So while we just highlighted the amazing actions and how world-changing 1 year can be in someone who dedicates their life to protecting places and people they love, or sorry, imagine what you could do if you commit your whole life to it. So.
And our distinguished environmentalists that we're choosing this year for the first time, because we kicked Anne-Marie off of the organic committee. And it was just Stephen and I and others that we consulted. And we have selected Mike Gaul as our distinguished environmentalist. So we have a little bit of a history of your experiences. Mike's life started in this tiny pool in Chicago and took him to the largest ocean in the world. His career was really, career in biology began at Yale, where he focused on wildlife ecology, and where, if you've talked to Mike very long, you might find out he was classmates with George W. Bush. From there, he joined the Peace Corps and was stationed at the University of South Pacific in Fiji.
Where Mike taught and did research and developed teaching materials on marine biology from January 1970 to July 1973. He found and described a new fish species of Siganus, uspi, was it USP, which he named after USP, the University of South Pacific. From there, he went on to the beloved University of Guam in 1973 to learn coral and algae taxonomy under Dick Randall, who you mentioned earlier this morning, who Mike had discovered a species of soft coral in 1976 and named it after Dick. And then in a spin of serendipity, 2 years later, a soft coral was named after Mike.
He also worked in the EPA and coastal zone management programs in Guam in the mid-1970s, and working in part on the land use plan that I mentioned earlier. And from 1977 to 1980, he was the only environmental planner for the entire U.S. Trust Territory of Micronesia, which covered everything except for Guam. And in the early 80s, he decided he needed another master's, so he went to the University of Hawaii for a thesis on marine resource development planning for Pacific Islands, and then was the first chief of marine resources for the new FSM national government from 83 to 91.
It's actually singularity Gaul.
They've merged. So,
okay.
Yeah. Always thinking about his work. From 1991 Mike had worked for the government of Guam as an environmental planner in the Bureau of Planning, and then spent 5 years as the chief planner for EPA from 2003 to 2009. He finished out his career with about 10 years at the National Park Service. And through all of this, his passion was to help connect people to the beautiful places they live in. After retirement, he was committed to being present at every scientific event on island, including this. And we even had a joke at the beginning of the day 0 for us for setting up. And we were like, should we just put a name card for Mike right at the front? You could see, you saw him at one meeting where he stepped out during lunch to Zoom or to jump on a Zoom seminar before rejoining after lunch. He's the busiest retired person you could know.
And so I think Mike's depth of experience across Micronesia and commitment to these islands is just really incredible. He, both from a scientific perspective about his understanding and contributions in soft coral research has been impressive, as well as all of his work in the environmental planning efforts throughout especially the 70s to 90s. I think there's a lot we can learn from knowing more about what happened back then. And I think he loves to learn and teach others. So if you ever are kind of want to learn about corals, just ask Mike to take you out snorkeling. It's amazing. He just tells you all of these wonderful things about corals. It's so fun. I highly recommend it. And if you want to learn a little bit more, he gave a great talk at the last MTCC in 2021 that's recorded. And you can listen kind of a bit of a history of the last 50 years or so in Micronesia, environment and conservation. I think one of Mike's most important accomplishments, though, is being father to our very own Anne-Marie Gaul, who has been Dr. Anne-Marie Gaul, who is an incredible addition to the scientific community here. So good work on that. So overall, I think Mike is one of the most positive and encouraging individuals I've ever met, and I think we're exceptionally pleased to be able to present him with the Distinguished Environmentalist Award. Congratulations.
I'm really surprised at this. I didn't expect it. And I'm glad I'm still here. I've been really fortunate to have the career that I've had and to be able to learn so much about our natural resources here and all through the Pacific Islands, and to share the knowledge and to participate in training people and developing training materials, things like that. And so again, I am really pleased to be here, and thank you for your respect for NAFCO. And I think I'll quit. Oh, by the way, sorry, another thing to do. I'm due for a meeting at 6 o'clock on the fisheries management down in the canyon. So I hope to see you later.
So now we just have a few kind of last closing announcements. The first is one more kind of recognition of our sponsors, particularly University of Guam West Pacific Tropical Research Center, Guam Community College, and then our consulting firms, ARC Environmental and EA Consulting. Southern Mountain Gear, go check them out. They're right next to the museum. I think they might even still be open. I'm not quite sure what time they close. Bellowings, Aviation, and then tonight we'll soon get to taste the Guam Brewery contribution. I also want to recognize all of the folks who helped to organize. So we have the kind of board of TTT, which you've seen quite a bit, even some that are not here. So Tim Harvey Samuel has been really helpful at organizing the career fair from afar, though he's in the UK. And then for field trips, Anne-Marie, as well as Christiana and Maya, thank you all. The website, Geraldine Callor back here, did some work on. Career Fair, Harmony Yomai, who also wasn't able to be here, but she helped organize all of the folks for the Career Fair. We have some GCC support for the IT, Jason Oliveras and Angela Carrera. The wellness break, Francesca de Oro, thank you so much yesterday, it was beautiful. Food and just general support in the back. There's been a whole bunch of people. I probably have missed a few because I've been up here the whole time, so I apologize. But Christiana, John, Debbie, Lindsay, Shaylin, and probably some more. So thank you all for just getting the food out and keeping everything organized, the registration back there. And then timekeepers, Alan Aquino and Claudia Gold, and then Christiana as well, have been really wonderful up here. So thanks to everyone in the organizing committee. I want to give a little shout out to the University of Guam's Conference on Island Sustainability, which is April 11th to 15th, and their abstracts due tomorrow. So if you want to learn more about island, or if you want to, a lot of the talks that we presented here, I think would also work really well there. There's a lot of inspiring presentations. So I encourage you to check it out and to submit your abstract there. Next year, we'll go to Saipan for MTCC. Last time we were there was 2019, just before COVID started. So we'll revisit and have the conference again there. I think it'll be wonderful. Looking forward to it. I also put some parentheses around the tea here because there's some discussion of adding a marine component. There's a lot of folks who do marine work, and the marine and terrestrial worlds are intricately intertwined, and so it doesn't really make sense to keep them separate. And so we've been in part strong encouragement from our marine biologist board member here. I'm thinking about creating just a Mariana's conservation conference. Also some conversations about broadening out to include Micronesia more broadly as well. So feel free to chat with us tonight at the reception about about these changes. Things that we'll need from you, we'll send you a feedback form. That's really helpful for helping us figure out kind of what worked, what didn't, and please fill it out and get it back to us. That'll be on email hopefully tomorrow. Panel discussions and sessions for next year. We'd love to get more feedback or more suggestions for those. One of the things I'm hoping to have next year is a panel on the Northern Islands. And so I'm kind of seeding that idea now to think about what that might be and how we could share more information about research that's been done on the Northern Islands in a panel. But I'm open to other, we're always open to other ideas. So if you're thinking about this would be a really cool panel to have, we'll get these people together or this session, please let us know. In Saipan, we'll need some volunteers for field trips and other organizing needs. And then we're always looking for more sponsorships. We're somewhat able to cover the cost of this one. I think we're going to hopefully apply for some funding to support organization of this conference every year, so that it's not quite as much in all of our volunteer time. But if you are in a organization that can sponsor a conference, we definitely would appreciate that. And I'll just close it out by saying we have one event. There's no announcements. There's nothing happening there except for eating great food and drinking and talking to people and being in a really cool place, the Guam Museum. So it's from 6 to 9 p.m. tonight. We hope to see you all there, and it should be great. Thanks, everyone. Well, there's lots of food left over. Time to gluten.
Probably on that.
Yeah.
And then like reach out to everyone that wants to like remind that they think your job here.
Yeah, nice.
I'm trying to stop by the museum. It's okay. I'll try to go to the museum. Thank you. Yes, tonight. Congratulations. Yeah, thank you so much. You guys are very nice. Oh, God, it was great. I mean, just the ones I caught today were great. Yeah. I only caught, I don't know, but the rest was what? 11?
Yeah.
Yeah, that whole afternoon, yeah. It was good. But I missed the last one in the morning about the relocation of the bird.
Yeah.
Can we, or later or something?
On that.
Yeah, I need to research the specific aspect of that project because I'm writing something else.
Oh, nice.
Okay. Okay. Okay.
And you can also, the video will be like uploaded to, so you can.
Oh, yeah, okay. Okay, I think I'll just still say hello to them again. Okay, can I call you?
Okay, so I'm gonna.